Good morning to esteemed participants, distinguished guests, and speakers. I, Shalini Sharma, welcome you all to the International Online Conference on Drug Discovery, Development, and Lead Optimization. Good morning
As
to
the sun
esteemed
rises on a
participants,
new day, so
distinguished
too
guests,
does the
and
opportunity
speakers for
I
collaboration, learning, and inspiration. I hope this morning finds you refreshed and ready to embark on a journey of discovery with us. Today, we all are gathered from every corner of the globe, bound together by a shared thirst of knowledge and a commitment to innovation. Over the course of this event, we are privileged to host a diverse array of sessions featuring keynote addresses, panel discussions, presentations covering a wide spectrum of topics spanning various disciplines and industries. As we embark on this exciting journey together, let us embrace the spirit of curiosity, open mindedness, and collaboration. Let us seize this opportunity to learn from one another, foster new connection, and catalyze positive change in our respective fields and communities. Now, I may please request Dean School of Pharmacy and Emerging Sciences, Baddi University of Emerging Sciences and Technology, to inaugurate and share the brief about the conference. Over to you, Mishra sir.
Thank you, Dr. Saleh Sharma. So I welcome all the participants to our 2 days international online conference on drug discovery, development, and lead optimization in association with our technical partner, HIM Science Congress Association, CRIKC Chandigarh, CIPET Baddi Himachal Pradesh, RM Chemicals Baddi. So the School of Pharmacy and Emerging Sciences is the premier center of pharmaceutical education and research center located in the largest pharmaceutical industrial hub of Asia and have been awarded as the best emerging school in pharmacy in North India by AICTE-CII survey for consecutive 2 years in 2015 and 16. School has state-of-the-art infrastructure for training students on drug discovery, drug development, production, quality control of drugs and pharmaceuticals. So our objective of the conference to furnish attendees with awareness on where the pharmaceutical sciences and drug research and development sectors are heading over the next decades so that they may align their professional goals according in order to elevate themselves significantly. In our 2 days conference, 24 speakers are going to share their views on different topics which are going to our participants in enriching their knowledge in the area of pharmaceutical drug discovery and development. Now I request our assistant professor, Mr. Pansul Jamwal, to play the video of Baddi University to know about us, our school, as well as the university. Over to Mr. Pansul Jamwal.
Hello, Panshul sir, Raman sir, Tarsim sir. Hello,
Hello.
are you with us?
Yes, madam.
Okay, sir, please share the video of Baddi University.
Wait a second, madam.
Okay, sir.
शाली मैम,
Sir?
स्क्रीन दिख रही है मेरी?
No, sir. Only audio is allowed, I think so.
जस्ट अ सेकेंड मैम. Is it visible now, ma'am?
No, no, sir, it is not visible.
So now I request Professor Dr. T.R. Bhardwaj sir to deliver the welcome address of today's conference.
Hello, good day.
Good day, sir. Good morning.
Good morning, sir.
Good morning. On behalf of Chancellor of Baddi University, Vice Chancellor, Secretary, Faculty of School of Pharmacy and Emerging Sciences, and students, I welcome all the participants, specifically our chief guest, Dr. Montu Patel, President of Pharmacy Council of India, our guest of honor, Professor Roop Krishan Khar, and all other participants, including those who are going to present their papers, oral or posters, all are welcomed. Friends, as we all know that India has already emerged as pharmaceutical competitor and has earned third position worldwide with respect to bulk drugs, API, as well as the formulations and generic drugs. It's a matter of pride for all of us that today, 1/3 of the pharmaceuticals, formulations, APIs are being manufactured in India. And it's again a matter of pride for us that 1/3 of that, India's share, is being manufactured, produced in our neighborhood, Baddi, Brotiwala, and Nalagarh areas. And we are located right in the center of this pharmaceutical hub. As we all know, that the Baddi University of Emerging Sciences and Technology is committed to provide future-ready professionals, pharmaceutical professionals, maybe professors, teachers, scientists, chemists. They are prepared as per need of the universities, and for
Sir, kindly unmute yourself. Hello? Mishra sir? Sir, kindly unmute. Yes, sir, you are audible.
Shalini?
Yes, sir, now you are audible, sir.
May I repeat now?
No, sir, I
I'm
think,
to start.
yes, sir.
As we all know, that's a matter of pride for all of us that 1/3 of world's pharmaceuticals, formulations, generic drugs, as well as APIs, they are manufactured in India. And it's a matter of pride for us that 1/3 of that also is being produced in our neighborhood, Baddi, Brotiwala, and Nalagarh area. And we are rightly located in the center of the hub of pharmaceuticals. Our commitment towards the profession is that we are to provide future-ready professionals as per need of the university and industry, pharmaceutical industries. And for that, we have experts from the industry who are member of our board of studies, as well as they are also resource persons delivering lectures on the related topics, latest status of the area is provided to the students. So friends, for that, we have organized this international conference on drug discovery, development, and lead optimizations. These kind of conferences further motivate as well as provide art of the status, present art of status of the area of concern to the professionals so that they can modify their curriculum, they can modify their approach with respect to the area of specializations as per need of the industry. So I once again welcome all the participants, all the invited speakers, specifically Dr. Hector Jose, who is going to present his presentations right now, and he's already joined. to welcome you all, and I extend my deepest of wishes for the grand success of this conference to the organizers, specifically Dr. Ramesh Mishra, Dean, School of Pharmacy and Emerging Sciences, and HOD, Dr. Bhartendu. Over to Salini.
Thank you, sir, for the inauguration and brief introduction of Baddi University. Without further ado, I'm taking the opportunity to introduce our first speaker, Dr. Hector Jose. Dr. Hector is from Puerto Rico, is a distinguished scholar with a robust academic background from the Pontifical Catholic University of Puerto Rico, holding a PhD in clinical psychology, a BSEE in education sciences, focusing on early childhood and elementary education. He embodies a multifaceted and dedicated professional. Beyond academia, Hector is a seasoned clinical psychologist specializing in trauma intervention and rehabilitation counselor in all the scope of practice of the profession. His research endeavors focus on the prevention of suicidal behavior and delve psychosocial dimensions of chronic illness and disabilities and employees' well-being. Welcome, Dr. Hector. Over to Dr. Hector.
Thank you. Good morning. Let me see. This is the first time I'm using Teams, so let me see if I can share my presentation. Okay. Can you let me know if you see my presentation?
No, sir. It is not
Yes,
visible.
sir. Yes, sir. It is visible to us.
Yes?
Yes.
Okay. Okay, so again, good morning. For you, I appreciate the invitation to this important conference. You might be questioning why a clinical psychologist and a rehabilitation counseling is presenting in a conference dedicated to drug discovery, development, and lead optimization. So I am going to discuss with you a little bit on how we can elevate the role of rehabilitation counseling with a crucial insight into drug discovery, development, and lead optimization. Our objectives for today's presentation is first to understand the scope of practice in rehabilitation counseling, and then try to understand how we as rehabilitation counselors can influence and optimize each stage of the journey in an interdisciplinary collaboration. So you might find some challenges when you conduct clinical trials and when you are developing different drugs to treat different disabilities and conditions. Some of them has to do with this research design, some safety concerns, not only from the developers, but also from the participants of the clinical trials. Maybe there are some lack of efficacy of how the process is conducted, unexpected results for both from the researchers and also for the participants, insufficient funds, and moreover, many clinical trials have difficulties in patient recruitment and retention. And sometimes most of these challenges are related to psychosocial factors in terms of disability and chronic illnesses. Also, other challenges are the animal models often cannot recapitulate an entire disorder or disease, challenges related to heterogeneity of the patient population, and greater emphasis on human data, might lead to improved target identification and validation. And there is a lack of validated diagnostics sometimes when we are trying to recruit some participants. So, 1 in 6 people might be diagnosed with any disability of a chronic illness worldwide. That means approximately between 16 to 20% of people around the world right now is living with a disability or a chronic illness. So we as rehabilitation counselors, we use a systemic process to assess individuals with various types of disabilities, including physical, mental, developmental, cognitive, and emotional, to achieve their personal, career, and independent goals. Through the counseling process, we build rapport and fostering trust through communication with our clients. We establish personalized goals with them. We implement psychosocial, vocational, social, and behavioral strategies, and we empower individuals to advocate for their needs and their rights. Through the rehabilitation counseling process, individuals with disabilities and chronic illnesses can navigate challenges, overcome barriers, and enhance their overall well-being, ultimately contributing to their successful integration into society. In our field in rehabilitation counseling, a diverse array of techniques and modalities are deployed to support individuals with disabilities on their journey towards achieving personal and professional fulfillment. This approach, while we have a varied approach, are united in their dedication to addressing multifaceted needs of each individual. For example, in terms of assessment and appraisal, and that might be very useful when developing clinical trials, at the outset of the counseling process, a thorough assessment is conducted with each individual, and we delve into the unique strengths, limitations, and needs of the individual. This comprehensive understanding forms the bedrock upon which personalized intervention strategies are crafted. Another aspect that you might be interested is individual and group counseling interventions. Acknowledging the profound impact of disability on both medical and psychosocial fronts, we as rehabilitation counselors offer individual and group counseling interventions. Through these interventions, individuals find solace in shared experiences and receive support in adapting to the myriad challenges they face. Also, we can conduct case management, referrals, and service coordination. The journey towards rehabilitation often needs access to a myriad of services and resources. We, as rehabilitation counselors, serve as navigators in this labyrinth, facilitating connection to essential services, coordinating referrals, and ensuring seamless service delivery. And finally, in terms of consultation services, we collaborate. Collaboration is in the heart of rehabilitation counseling profession. We engage in consultation services, liaising with diverse stakeholders, including healthcare providers, employers, and regulatory system. Through these collaborations, barriers are dismantled and pathways to access are widened. So you might be asking why I am talking about rehabilitation counseling. Well, let's set the stage by exploring the profound intersections of rehabilitation counseling and drug development. Because as rehabilitation counselors, we can collaborate with you in terms to understanding the needs of the people, informing target identification and validation needs, we can develop a collaboration that is patient-centered when we are developing drugs, and we can optimize clinical trials and design. As rehabilitation counselors, we possess a nuanced understanding of the intricate web of individual patient needs and complexities, transcending the confines of addictions, disabilities, or chronic illnesses. Our proficiency extends to evaluating a spectrum of factors encompassing psychological, social, and environmental dimensions. This holistic approach enables us to glean profound insight into the multifaceted experiences of patients grappling with various medical conditions. Moreover, rehabilitation counselors bring to the table a wealth of experiential knowledge gleaned from their interactions with patients from various backgrounds. This rich tapestry of experiences enriched the drug development process by providing developers with first-hand accounts of patients' journeys, illuminating the real-world impact of medical conditions, and offering invaluable perspective on potential treatment avenues. So in essence, the collaboration between rehabilitation counselors and pharmaceutical developers serve as a catalyst for innovation and patient-centered care. Collaboration with rehabilitation counselors contributes significantly to the identification and validation of drug targets across a spectrum of medical conditions. By leveraging counselors' profound insight into patient experiences and the underlying mechanism of illnesses, pharmaceutical researchers gain a unique vantage point for pinpointing potential targets for interventions. We as rehabilitation counselors, we possess a deep understanding of the intricacies of very medical condition, as I mentioned before, informed by our extensive interaction with patients. Through a holistic assessment, we as counselors uncover the intricate interplay of biological, psychological, and social factors underlying these conditions. This multifaceted understanding provides researchers with invaluable clues for identifying novel drug targets that address the root cause of disease. In essence, the collaboration between pharmaceutical researchers and rehabilitation counselors represents a synergistic partnerships that harness the power of interdisciplinary collaboration to drive innovation in drugs discovery and developing. The shift toward patient-centered drug development applies universally across medical conditions in interactions with patients. By involving rehabilitation counselors in drug development teams, pharmaceutical companies gain access to a wealth of patient-centered insight. These insights enable researchers to design clinical trials that are more inclusive and representative to the target patient populations, leading to more robust and generalizable findings. In terms of optimizing clinical trial design, rehabilitation counselors play a pivotal role in refining clinical trials by offering insight into patient recruitment, retention, and adherence across various medical contexts. Our deep understanding of patient-related barriers and facilitators aid in designing studies that are inclusive and representative of the target patient population. Clinical trials are the cornerstone of medication development. Yet, they often face challenges in recruiting and retaining participants. Rehabilitation counselors bring a unique perspective to the table, drawing from their experiences working with individuals from diverse backgrounds and medical conditions. By collaborating with us as rehabilitation counselors, pharmaceutical companies can design clinical trials that account for the diverse needs and preferences of patients. The collaboration between pharmaceutical companies and rehabilitation counselors in optimizing clinical trials design represents a pivotal step toward advancing medication development. So what are the key components and strategies that we rehabilitation counselors can bring to the table? First, a holistic approach to rehabilitation, addressing physical, psychological, and social aspects. We also can bring to the table evidence-based techniques as motivational interviewing and cognitive behavior therapy to promote retention of clients, of patients during clinical trials. A patient-centered approach are paramount in drug development, placing patients at the forefront of the process. By prioritizing patients' perspective and needs, pharmaceutical companies can develop treatments that are more effective, accessible, and aligned with patients' preferences. Patient-centered drug development fosters a deeper understanding of the lived experience of patients, including their symptoms, treatment challenges, and quality of life. These insights inform decision-making at all stages of drug development, from target identification to clinical trial design and beyond. Furthermore, a patient-centered approach enhances patients' engagement and empowerment, fostering collaboration partnerships between patients, researchers, and healthcare providers. Incorporating patient perspective in preclinical and clinical studies is essential for developing treatments that resonate with patients' experiences and preferences. Patient-reported outcomes, qualitative research methods, and patient advisory boards are valuable tools for capturing patient perspective and integrating them into study designs. And we as rehabilitation counselors can contribute in these aspects. Moreover, in terms of when you are developing a research and including vulnerable populations, there are ethical considerations that we might examine. First, ensuring informed consent and protecting vulnerable populations are critical components of ethical drug development practices. Informed consent involves providing patients with comprehensive information about the nature of the study, its potential risks and benefits, and their rights as participants. This process empowers patients to make autonomous decisions about their participation in clinical trials, promoting respect for their autonomy and dignity. In terms of vulnerable populations, as people with disabilities and chronic illnesses, including individuals with disabilities, mental health conditions, and substance use disorders, require special protections to safeguard their rights and well-being. Rehabilitation counselors play a pivotal role in advocating for the rights of these populations and ensuring that they are treated with dignity and respect throughout the drug development process. Another aspect that we should consider in terms of ethical consideration is stigmatization and bias, because they pose significant barriers to effective research and counseling in the field of drug development. Stigma associated with certain medical conditions such as mental health disorders or substance use disorders can deter individuals from seeking treatment or participating in research studies. This stigma can also influence the attitudes and behaviors of healthcare providers and researchers, leading to bias in the delivery of care and the interpretation of research findings. Rehab counselors are uniquely positioned to address stigmatization and bias in research and counseling. Through our advocacy efforts and patient-centered approach, counselors work to destigmatize medical conditions and promote understanding and acceptance within the healthcare community and society at large. In summary, ensuring informed consent and protection of vulnerable populations, as well as addressing stigmatization and bias in research and counseling, are essential components of ethical and effective drug development practices. By collaborating with rehabilitation counselors and adopting patient care and patient-centered approaches, pharmaceutical companies can promote respect for the patient's rights, enhance patient safety, and foster more inclusive and equitable healthcare systems. So there are opportunities in this synergy between pharmaceutical professionals and rehabilitation counselors. Precision medicine and personalized treatment approaches offer exciting opportunities to revolutionize healthcare by tailoring treatments to individual patients' characteristics. These approaches leverage advancements in genomics, biomarkers, analysis, and data analytics to identify optimal treatment strategies for each patient, based on their unique genetic makeup, disease profile, and treatment response. Rehabilitation counselors can contribute valuable insight to precision medicine initiatives by providing holistic assessment of patients' psychosocial and environmental factors. By considering these factors alongside genetic and molecular data, healthcare providers can develop more comprehensive and personalized treatment plans that address the individual needs and preferences of patients. Moreover, interdisciplinary collaboration between rehabilitation counselors and other healthcare professionals presents numerous opportunities for innovation and advancement in patient care. By bringing together diverse perspectives and expertise, interdisciplinary teams can develop holistic and integrated approaches to addressing complexities of medical conditions and improving patients' outcomes. So, what are the opportunities that we have to collaborate? First, integrating rehabilitation counselors into precision medicine initiatives to provide holistic assessments and personalized treatment plans, collaborating with researchers and clinicians to develop innovative interventions that address these psychosocial and environmental factors influencing health and well-being. Third, partnering with communities, organizations, and support networks to enhance patient engagement and access to care. And finally, engaging in research and advocacy efforts to promote policies and practices that support the needs of individuals with disabilities and chronic conditions. So in conclusion, addressing the physical, psychological, and social aspects of patient care is paramount in the field of drug development and rehabilitation counseling. By adopting a holistic approach that considers the multifaceted needs of patients, we can optimize treatment outcomes and improve overall well-being. Recruitment, retention, and adherence are critical components of success clinical trials and patient care. Collaboration between rehab counselors and drug development teams is essential for designing studies that are inclusive, representative, and patient-centered. By incorporating insights from rehabilitation counselors, we can enhance patient engagement, retention, and adherence, ultimately accelerating the development of new medications and improving patient outcomes. Evidence-based counseling techniques serve as the foundation for effective rehabilitation counseling interventions. By utilizing techniques such as motivational interview, cognitive behavioral therapy, and family therapy, we can empower patients to overcome barriers, achieve their goals, and lead fulfilling lives. In closing, I call upon all stakeholders in the field of drug development and rehabilitation counselors to continue collaborating and advancing the field. By working together, we can harness the power of interdisciplinary collaboration, precision medicine, and personalized treatment approaches to transform patients' care and improve the lives of individuals with disabilities and chronic conditions. Let us commit to ongoing collaboration, innovation, and advocacy as we strive to create a healthcare system that is truly patient-centered and inclusive. Thank you.
Thank you, Dr. Hector. Your insights on... Hello? Thank you, Dr. Hector. Your insights on rehabilitation counseling have broadened our perspectives and sparked new ideas that will undoubtedly influence our future endeavors. Now it's time to start Q&A session, and we have with us our panelist, Professor T.R. Bharadwaj sir, advisor to Chancellor Baddi University, Dr. Avinash Mishra sir, and Dr. Bhartendu Sharma. Attendees can also raise their hand to ask any question, and Panchul sir will let him or her in one by one. Over to Bharadwaj sir.
Yeah, it's a very, very interesting collaborative area of research. The medicinal chemist identifies the pharmacophore required for the drugs used in the rehabilitation process, and clinical psychologist, giving some thereafterwards feedback to develop into the desired molecule having best efficacy and least side effects or toxicity. So since the drug discovery is again multidisciplinary area, it's really very good to hear from you that what kind of collaborative research could lead to the drug discovery, development, and of course, lead up to medicine can be taken care of. Thank you very much, Dr. Hector Jose, for your very, very educative, very, very informative talk. Thanks. Over to Dr. Avinash Mishra.
Thank you, Dr. Hector, for connecting the drug discovery and rehabilitation counseling into the development of new molecules, which are going to help the community to get toxic-free medicines. Because nowadays, most of the drugs have side effects, as already we have seen in case of our COVID-19 pandemic, where only rehabilitations or prevention techniques helped a lot to all the patients. So we would also like to know from your side, can you give example of some medicines which are we can means, or in any therapy which plays a role in like having the medicine uh, means instead of you prescribing the medicine by rehabilitations, they can means get rid of the some ailments or the symptomatic treatments.
Okay. Well, it's important to clarify that we as rehabilitation counselors, we don't prescribe medications. We work collaboratively with most of the time with psychiatrists in if we have clients with mental illnesses or any physician, depending on the disability we are working with. I, as a rehabilitation counselor and as a clinical psychologist, one of my courses was in pharmacology. So I have general knowledge in terms of how medications works within the body. So what I do as rehabilitation counselors, for example, I have an app that I can review the interaction between medications. I can review side effects of medications, short-term and long-term side effects. So when here in the United States and back home in Puerto Rico, we have the same healthcare system. So often physicians does not take time to explain these details to clients and to patients when they are going through a treatment, and moreover when they are in clinical trials. So when I, as a rehabilitation counselor, explain these details to clients, I can develop in them more adherence to their treatment. And actually, when they understand the risks or benefits of being a participant in a clinical trial, that allows them to make an informed decision and stay in clinical trials. I had a few of my clients back home in Puerto Rico that they were going through clinical trials in psychiatric medications, and in the front they experienced a lot of fear, of uncertainty. What's going to happen with what if this doesn't work, and I want to recover. I want to get better. So in our field, we work with those fears. It's like valuable information to address the whys this medication don't work. So I don't know if that addresses your question.
No, it's good. Doctor, can you also means let us know about the rehabilitation in case of our terminal patients?
Terminal patients. That's an emerging field in our profession. When we work with terminal patients, we don't only work with the patient who has the chronic illness, right? We also work with the systems around the patient. So actually, that's one of the topics that I am getting more interest now on developing the attitude to cope with the death process with dignity and with humanity. So we here in the United States and in Puerto Rico, we are promoting more in terms of helping patients to decide why I should continue with pharmaceutical treatment and why and when I should not continue, and address all those emotional aspects of when patients decide I need to stop because I need to balance between my physical well-being and my emotional well-being, and my support system well-being. So we as rehabilitation counselors, we address those aspects of coping with that. Again, not only with the patient, but also with family members or support systems. I encourage, that's not frequent, that we see rehabilitation counselors in death and dying process. So now I am a big advocate for the healthcare system to incorporate counselors, rehabilitation counselors, and psychologists within the healthcare provider system when we are dealing with terminal patients, for them to have dignity while they embrace that as part of living.
Okay, thank you, doctor, because it is the means role of the clinical psychologist in such cases are very important and very crucial. So thank you, sir, for be with us. And now I request our Dr. Bhartendu. Yes, sir, please.
Thank you. Good morning, Dr. Hector.
Good morning.
How are you? And thank you for sharing this valuable information. Dr. Hector, when we talk about patient-centered drug development, in that case, we have to communicate with the patients and to share this information. So I just want to know what are the best ways to communicate the information and to support their decisions, the decisions that they had made that we are communicating. So what are the best ways to communicate and to support the decision in patient-centered development?
Yeah, in terms of patient-centered treatment, one of the best practices I encourage is not only to have one person with the skills to communicate with assertiveness and with compassion. Okay, so we need first to develop those skills, compassion and assertiveness. When we are communicating a difficult information in terms of patient care, my approach and what have worked for me first is, first, the patient should not be alone, because the support system is part of a big decision. So when someone is making a decision, it not only impacts one person, it impacts many people around that person. So yes, my approach is, when I communicate difficult information, I never do it with only the client. Okay, and that's part of the informed consent. When we are clear with our clients, when we are transparent with them, and allowing them to know upfront that at some point you might bring others to support the decision-making process, that is very helpful. In terms of being compassionate, it's like, I don't want to say that there is going to be a perfect time to communicate a difficult information, a hard information. I believe that maybe no one is never ready to get hard information. The difference is how we said it, and the making us available to support the person who is receiving that information. That we provide information with sensitivity, and also providing them options to buffer the harm that the information can cause. Having a mental health professional when you communicate hard information, for me, should be kind of the best practice. Not only the healthcare provider, but also the mental health provider should be there, because that's our training. It's in how we can help people to cope with pain, sadness. So yeah, that might be. Besides that, And going back to my first suggestion is that not only have one person trained to communicate, because when you communicate, the patient will need a lot of support from the medical provider to the nurse to the administrative assistant in a healthcare office, from the researcher to if you are in a clinical trial. So everybody needs to understand the how to be compassionate and how to sometimes get in the other person's shoes to feel their feelings and their experience.
Thank you. Thank you so much.
Yeah.
Thank you to our panelist. Thank you to our panelist. Attendees can also raise their hand and ask their question one by one. Is there any question, attendees? Hello, Komal, are you there? Hello. Hello.
Hello, good morning.
Good morning.
Komal, you can raise your question.
Yeah, ma'am. Thank you so much. Good morning, Dr. Hector. I hope you are doing well. I wanted to ask you about how can counseling insights enhance drug discovery and lead optimization? Could you share your insights on this topic, please?
Okay, in terms of how we can enhance what I discussed, I am going back to some points of my presentation. First, we as counselors, we have first-hand information from people who are suffering from different conditions. So providing that information to researchers on how people experience their lives, how people experience the symptoms of a medical condition, might be important when you are trying to discover these molecules that works to treat any type of symptoms. So having that first-hand information from clients and patients who are using the medications, that's very valuable in the process. Secondly, if we, as rehabilitation counselors, if we have the chance to be incorporated in clinical trials, we can help the development of drugs in terms of the target population. We have contacts in the community. We know services, we know resources available. So when drug developers are developing a medication, we can address them to the specific population who are in need of that drug. So we can shorten the time for identification of possible participants in those clinical trials. Once we identify the participants, we can be there to retain the participants, because at some point and often they develop psychosocial symptoms in terms of I feel fear, I feel sad, I am experiencing uncertainty, I don't know what is going on. So we as rehabilitation counselors, we can handle all those emotions within the participant of a clinical trial to avoid them to abandon the research project or the clinical trial. So those are kind of our primary aspects on how we can help the drug development and optimization. And besides that, if clients if we are able to work with you to retain clients, then we should be able to develop in then adherence to the treatment. So in terms of long-term research, you should be able to obtain better information on how the medication works in long-term use.
Okay, thank you so much, sir.
You're welcome.
Thank you so much. Thank you.
Thank you, Komal. I hope the answer satisfies you. Thank you, Dr. Hector.
Yes, you're welcome.
Thank you for sharing your knowledge and expertise with us. And we look forward to the opportunity to collaborate with you in the future. Thank you.
I will be more than happy to collaborate with you.
Thank you so much, sir.
Thank you again for the invitation, and I hope you have a blessed day and a very enrichful day for you during this conference.
Thank you.
Thank you. Thank you very much.
Have a good day.
Thank you so much.
Same to you. Thank you.
So, participants, now it's time to welcome our second speaker of the session, Dr. Zayabing Fan. Dr. Zayabing is at UMES Department of Pharmaceutical Sciences. Prior to this, he was assistant adjunct professor at University of California, Los Angeles, and he has completed his postdoctoral fellowship at Howard University. His research focuses on developing novel therapeutical approaches for bone repair and age-related skeletal disease treatment. He has broad research background in stem cells, tissue engineering, drug or gene delivery, biomaterials, and molecular pharmaceutics. He has received UCLA, the Outstanding Mentorship Award, and the Princeton PACRI Award, and published over 40 peer-reviewed articles, and several are published in high-caliber journals. We welcome you, Dr. Zayabing. Over to you.
So thank you for introduction. Can I share my PowerPoint?
Yes, sir, you can share your PPT.
PPT, just the click the Windows or click screen. I don't know how to share. Can see. Sharing.
Panchul sir, are you with us?
Share Windows. Yeah.
Yes, ma'am.
Sir, can you please help? Oh, okay.
Can you see my PPT now?
Yes, sir, it is visible. Your PPT is visible to all of us.
It's okay, right? Okay, sounds good. So so everybody can see my the whole PPT, right?
Yes, sir, it is visible.
Yeah. Okay, great. So thank you for your introduction, and it's very nice to meet you everyone virtually. And also I want to thank this Baddi University for this opportunity to share my work. So today I'm going to talk about one specifically the research project. The topic is not very broad, but it's mostly related to my previous research. So I'm going to talk about the generation of the exosomes mimetics for the bone regeneration. So my talk will cover how to generate exosome mimetics and engineering exosome mimetics for bone repair and regeneration, particularly under osteoporosis disease model. So let's start. So that we all get older. So aging populations increase globally, and aging can cause different type of the chronic disease. So I want to point out that aging is one major factor contributing to the one type of the chronic disease called osteoporosis. So osteoporosis affected over 34 million people in the USA alone, and mostly affect the female and aging over 15 years older. And also the treatment of the osteoporosis need a high medical cost. Unfortunately, so far, there's still no effective treatment about the osteoporosis disease. And also the osteoporosis can induce severe side effects like a bone fracture. I believe that many of you may know your surrounding people, particularly at an older age. So one day when they fall down, they have their bone fracture. So actually, I have one friend, so she told me recently, her mom and the age is 74 years older. She told me her mom suddenly fell down and her mom have severe craniofacial bone fracture. So why are some people easier to get a bone fracture, other people are not? So studies show that there are some people at the older age, they may have their osteoporosis disease. So this osteoporosis disease can cause their bone fracture. So the osteoporosis that induced the bone fracture is a major cause for the disability there in the aged people. So there maybe you know this drug is called Alagilate. This Alagilate drug is the first line drug for the osteoporosis disease treatment in clinical. But the patients still need long-term use this drug, and also this drug have limited efficacy in the clinical. So studies show that this drug may also cause one significant side effect called osteonecrosis. Osteonecrosis means that the patient may have the death of the bone tissue. This is a very severe side effect for this drug. So in addition, the BMP2 is also commonly used for the bone the reconstruction in clinical. So actually, BMP2 is first discovered by Dr. Urster and UCLA in 1965. So now BMP2 is FDA approved growth factor and commonly used for spinal fusion and tibia hernia maxillofacial surgery. However, the high dosage BMP2 is still required in clinical. So high dosage BMP2 not just increase the medical cost, but also may induce life-threatening side effects like ectopic bone, cyst bone formation, tissue swelling, and tumor risk. So in my previous lab, we successfully use high dosage BMP2 to induce this side effect in the animal model. So we use this high dosage BMP2 you induce hollow bone and cyst bone formation. So you can see this hollow bone. So the bones, the middle of bones most often is empty. And also there's some bone, cyst bone. So the cyst bone, they filled with fat tissue. And that means that this high dosage BMP2 generated this bone tissue have limited quantity. So this is in general. So this BMP2 induce this side effect completely affect the BMP2 the wide use in clinical. So besides the BMP2, I believe that you already know the stem cell therapy, right? So stem cell therapy is also regarded as the promising therapy for the tissue repair and the regeneration, particularly for bone regeneration. So when we mention stem cell, we also talk about the mesenchymal stem cell. So what are mesenchymal stem cell? So mesenchymal stem cell is a multipotent ability that can differentiate into different type of cell. Also, mesenchymal stem cell can be harvested from different human tissue like bone marrow, fat tissue, and even tooth. So after we harvest this mesenchymal stem cell from the human tissue, and then we can proliferate them and get enough lumbar cell, and then we can use this stem cell for the bone regeneration through local injection or systematic infusion. Another approach, we can combine this stem cell and mesenchymal stem cell with scaffolder to generate this stem cell and scaffold complex for the bone defect repair and regeneration. However, the mesenchymal stem cell treatment so far, they still have some limitations. For example, they may induce unwanted immune response Also, maybe they can induce like a cancer risk, and also they can bring some disease, because some stem cell they're collected from their like a different donor, right? So the donor, this patient may bring they have some potential disease. So this stem cell therapy still have limitation now in the potentially the clinical application. So based on the challenges, these stem cell challenges, right? There's still a lot of challenges. So MSC derived exosome is regarded as another alternative approach for the bone regeneration. So what are the exosomes? Exosomes are nanoscale vesicles produced by cells upon the fusion of the intracellular polyvesicle with the cell membrane. Also, the diameter of the exosome ranged from 30 nanometer to 150 nanometer. And also exosome composed of different protein, mRNA, and microRNA. So provide this promising biology drug for potential disease treatment. So recent years, I know the Americans, so they have many like biopharmaceutical company that focus on developing this exosome-based product for potential disease treatment. So this slide shows the ideally the exosome-based therapy. So first, we can culture stem cell or other type of cell. So after we culture this stem cell or other type of cell, we can collect the growth medium, culture medium, and this we can use the appropriate method to isolate this exosome. So after isolation of the exosome, we can characterize and analyze this exosome. But for the company, and in this stage, they may use they can do COC test. So after that, we can manufacture this exosome to form this exosome product, and then we can use this exosome product for potentially clinical disease treatment. So this is ideally the approach to use or develop exosome-based product for clinical application. So far, they already have been many clinical trials about exosome-based therapy for different disease treatment. So this is a graph just summarize. So the several key disease related to the exosome treatment in clinical trial. For example, like acute respiratory disease, like COVID induced this type of acute respiratory disease, diabetes, like cancer, eye disease, or other stroke, particularly for neuron degenerative disease. So this is now so they already have many clinical trials related to the exosome-based therapy. So I believe they follow the progress of the exosome related to the research. In future, there will be more the exosome related to clinical trial. And so so globally, there are over like 15 the biopharmaceutical company focus on developing the exosome-based product. So the exosome-based product, they have different area. Some product related to exosome for the disease treatment, and some company develop an exosome product for some disease diagnosis. For example, exosome can be used for some cancer biomarker, so they can use it for disease diagnosis. So another exosome product related to the vaccine. So exosome can be used for create some the vaccine, and also exosome can be used for some drug delivery vehicle. So it's a very exosome now is developing can for broader application. So my talk, I really also include my lab. I'm mostly focused on developing some exosome-based therapy for some disease treatment. So for the exosome-based therapy, I think currently they have two major challenges. One is no production yield. So although they already have many methods developing for the exosome isolation, but the production yield is still very low. So it's really challenging now. Second challenge is non-specific function. I already introduced like exosome include different type of the small RNA protein. but the exosome have very broad application. That means they lack non-specific function. So this is two challenges for exosome-based therapy now. So this slide just summarize currently they already have different methods developing for exosome isolation. This ultracentrifugation method is regarding as like a golden standard method. Including my lab, I also use this method to isolate exosome. Also, they have other new technology to isolate exosome, like immune affinity method, polymer precipitation, microfluidic method, and also the acoustic separation. But I want to point out that all of these methods, they have the common limitation now. So they only can isolate a small volume of sample. Also need a high medical cost and time consuming, and also they only have their low production yield. So they are based on these limitations, particularly for the low production yield. So our lab, we developed one novel approach called the extrusion approach. So we use this approach to generate one type of the exosome called exosome mimeticus, EM for short. So we directly use this extrusion method to generate this exosome mimeticus from the mesenchymal stem cell. I already mentioned that in previous slides. So then to generate this EM. So it's a very simple and convenient method. We generate this EM. And this we want to evaluate the production yield, right? We want to increase production yield. So our result is very promising. You can see the generation of the EM can increase the over 10 times production yield when compared to control, like traditional method for exosome isolation. And this we want to compare the exosome mimeticus with traditionally the exosome about their morphology, size. So our results show that these two types of exosome show similar morphology and also similar size. And this we also study their cell internalizations. So results also showed that these two types of the exosome show similar cell internalization. So result is good. So the generation this exosome mimeticus show the similar property as the traditionally the exosome. And this we also compare these two types of exosome about their orgenesis. Through this LP staining. LP staining can check the early organic marker like LP activity. So through this result, you can see these two types of exosome show the similar orgenesis. But again, although our exosome mimeticus can increase the orgenesis, but we ask one question. I know this is a broad, this is universal the function. So could we further enhance this orgenesis of EM for the bone regeneration through a modulation of the endogenous small RNA? So to address these questions, first we want to find the interest in the small RNA. So the previous study also showed that BMP signaling, I already mentioned that BMP2, right? Also BMP the signaling play important role in the bone repair and regeneration. But BMP signaling also regulated by BMP antagonist like login. So this login can bind this BMP growth factor and interrupt this BMP growth factor, further bind this receptor so they can inhibit this BMP activity. So previous study, including our previous study, we found that if we lock down or decrease or inhibit this login protein, they will maximize this BMP signaling. They release this BMP growth factor so they can maximize this BMP activity, so further can increase this orgenesis. So okay, good. So we find this small RNA, called login siRNA, and then we ask a question: could we enhance endogenous increase of this small RNA in the exosome mimeticus? So for this approach, we developed another combined approach to generation of this small RNA modulated EM. So how can we achieve this goal? So we constructed the lentivirus encoding this logging shRNA, and then use this logging shRNA transfect the human MSC, and then we generate this logging suppress the MSC, and then we further use extrusion method to generate this exosome mimetics also the increase with logging siRNA expression. So also EM-NG for short. So this is our engineering this small RNA modulator EM through this combined approach. After that, we want to measure the logging siRNA level, right, in this modified EM. If logging siRNA doesn't increase, that means our strategy doesn't work. So the interesting our real-time PCR, we developed some real-time PCR. We can check this logging siRNA expression level. So you can see the modified EM-NG can increase this logging siRNA about 3 times further compared to traditionally the controlled EM. And this we also use this modified EM to treat the MSC. We want to see whether this treatment can significantly decrease logging protein. So through this Western blot, you can see the modified EM treatment can significantly decrease the logging protein when compared to control EM treatment. So this result further indicated that our strategy successfully increased the endogenous logging siRNA in this EM, and also this modified EM works, right? They can decrease the logging protein in the cell level. And this, based on this result, we further want to study their orthogenesis in vitro. So how can study this orthogenesis in vitro about this modified EM? So we can treat MSC cell with this modified EM called EM-NG, and after treatment, we can use ALP staining or unnecessary red staining to check the organic marker. So result is also very promising. You can see the modified EM treatment can significantly increase the ALP activity as well as this mineral deposition through this unnecessary staining. So when compare this control group, right? So this result further indicate that our modified EM can enhance the orthogenesis in vitro. So after that, we want to further study their orthogenesis in the 3D environment. So then we can combine this modified EM with injectable hydrogel to form this complex. And then we study their ALP activity and the mineral deposition. So similarly, so our ALP staining and unnecessary staining also show that the EM-NG, the hydrogel, this complex group should significantly increase the ALP activity and the mineral deposition. So this study further indicates that our modified EM can increase orthogenesis in 3D environment. So based on this 2D and 3D result, we want to further study their bone regeneration in animal model. So then we inject this modified EM and the hydrogel complex into the mouse clavellary defect. We create this 3 millimeter critical size clavellary defect in the mouse. So after the injection, after this 8 weeks, we use micro CT to check the bone regeneration outcome. So you can see the EMG hydrogel, this group can significantly increase the bone, new bone formation in this critical size, the bone defect when compared to control group. So this study further indicates that our modified EM-NG can promote the bone regeneration in animal model. So as scientists, we also like to ask why, right? So at that time, when we get this result, we ask how do this modified EM called EM-NG mediate the bone regeneration? And this we already know microRNAs play important role in the exosome for their function. So based on this hypothesis, we want to use a microRNA sequencing to screen all of the microRNA expression in this modified EM and control EM. And this we use bioinformative analysis we want to find the potential mechanism. So through this like volcano plots and a Venn diagram and a heat map, and then we found they have several key microRNA significantly upregulated or downregulated in this EMNG group. So we already have these several key microRNA, right? And then we want to identify one specific microRNA related to this the mechanism. So then we also use LP staining to screen these several key microRNA. So results show that our modified EM can mediate organogenesis through inhibition of the microRNA 29A. So you can see this EMNG treatment can increase, alone treatment can increase the LP staining compared to this control group. But after adding this microRNA 29A mimics, you can see after adding this microRNA 29A, so they can decrease LP expression compared to this EMNG group. So the decreased LP, the level is similar to the control EM group. So this study further indicated that microRNA 29A play important role in this modified EM, the function, right, for organogenesis, and also provide indirect evidence to show that this EMNG mediated organogenesis through this inhibition of the microRNA 29A. So this is we get this mechanism. So now there is a summary for the above study. So we successfully use this extrusion method to generate this small RNA modulated EM from this no-gain suppressed MSC. And this we also combine this modified EM with hydrogel and this injectable animal model, bone defect model. And our result also show that this the EM, the NG, the landed hydrogel can promote the bone defect repair in this animal model. And this we also use the microRNA sequencing assay. We found that this modified EM mediated bone regeneration through the microRNA 29A inhibition. So we know that when we inject the exosome into the animal body or the human body, they can distribute everywhere, may distribute every tissue and organ. That maybe induce some unwanted side effect. So it's better we can develop some like a bone targeting EM, right? So can specifically target the bone tissue. That will be good. So based on this hypothesis, we also developed the engineering approach to generate this bone targeted EM. So we first use the metabolic engineering approach to generate this enzyme modified MSC from this human MSC. And this we further use its extrusion method to generate this enzyme modified EM. And also during this approach, we also added one like organic small molecule drug called the SAG drug. And after that, we further conjugated the bone binding ligand called the allogeneic molecules. So we can use the clinic chemistry approach to conjugate this molecule into the surface of the enzyme modified exomimeticus to generate this allogeneic modified EM with this SAG drug loading. So the short name is ALD, EM, and SAG. I know it's a little bit complicated. And after generated these engineering exosomes, mimeticus, and then we study the bone regeneration through the systematic administration, or study the bone defect repair through their combination with the scaffold. So overall, our engineering exosome mimeticus, we achieved the two goals for this project. First, our engineering exosome mimeticus can deliver this small molecular drug called SAG that can promote bone regeneration. Second achievement, we can our engineering exosome mimeticus have the bone targeting ability through the conjugation of this allogeneic by the clinic chemistry approach. So after for generation this engineering exosome mimeticus, we want to test their bone binding ability. So how can we test this bone binding ability? So first we can labeling. this engineered exosome mimetics with Nile red. And this we also harvest the bone tissue from animal, and this incubate this deboned exosome mimetics and the bone tissue. So after like 24 hours, the incubation, you can see the engineering exosome mimetics show very strong signaling. That means they have a large amount of the exosome mimetics can binding with the bone tissue. And this we also can inject this modify the engineering exosome mimetics into the animal body, and we use the optical image system to look at the signaling. So you can see after 48 hours injection, they have very strong signaling in the bone tissue region. That means that our engineering exosome mimetics can also the promoter can show the very high binding ability with the bone tissue. So and this after finishing this work, we also the study this engineering the exosome mimetics with the cycle drug loading for the bone healing in the animal model. So we combine this engineering exosome mimetics with a scaffolder, and this implant the animal effector model. And this we use micro CT to study their result. The result is very promising. You can see. So engineering the exosome mimetics with this drug loading can promote this the bone healing in this animal model. So based on this micro CT, we also use the histology analysis to confirm this result. So this histology analysis including HE staining, mesotelioma staining, and picoserous staining. So you can see, so this engineering exosome mimetic treatment group, they can increase this new bone matrix in this bone defector regions when compared to the control group. And so lastly, I quickly briefly introduce one ongoing project in my lab. So we are working on the project targeting TRP3 for bone regeneration in osteoporosis. I already mentioned in previous slides, right? Osteoporosis affects the millions of people globally, and it's a challenge disease in clinical. So the pathology showed that the bone marrow from this osteoporosis patient looks like they already show like a fat tissue called a fatty marrow. So this fatty marrow can cause the bone loss. So also the previous study showed that MSC, the look at in bone marrow, they have balance. So MSC, they have the balance between the adipocyte differentiation and osteoblast differentiation. But in the osteoporosis patient, MSC already become abnormal. So MSC focuses more on adipocyte differentiation rather than osteoblast differentiation. So this this result show that if we reverse this abnormal MSC differentiation, may promote the bone regeneration. And also our previous study showed that TRP3 can promote the MSC for the osteoblast differentiation rather than the adipocyte differentiation. So based on this study, we suggest that targeting TRP3 is promising approach for osteoporosis. And this also we want to develop the engineering approach to develop this exosome mimetics with endogenous increase of TRP protein. And this will be found we one small molecule drug can increase the MSC to increase to treat this MSC can increase this TRP protein. And then we can use the extrusion method to generate this engineering exosome mimetics called EMTRP3. So after that, we further use ELISA assay to check this TRP protein, the level. So you can see this modified the engineering EM, they can show the significant increase of TRP protein when compared to the EM control group. So there, and also we do some pilot study. So we want to inject this EMTRP3 into the animal model called the OVS mouse model. OVS mouse can mimic the osteoporosis disease. So after IV injection, the 12 weeks, So we can use a PET micro-CD to check the bone density. So you can see this OS mouse, this is an osteoporosis disease model. So you can see they have decreased the signaling in the spinal region, spine and the non-bone region when compared the animal. But by contrast, the EMTRB treatment can increase this signaling. So that means they can increase the bone density in this spine and the non-bone regions. So this result further indicate that our engineering EM with the endogenous increase of TRB protein can promote the bone regeneration in the OS mouse. So this summary, so several of the key takeaways from my several projects. So we successfully use extrusion method to generate these exosomes. So our results should be very promising. So we generation this exosomes can show the significantly increase the exosome production, right? So we compare the traditional method. So we increase over 10 times production yield. And also we developed several engineering approach to develop this modified EM with endogenous increase of small RNA or protein. And this our result show that this modified EM with endogenous increase of small RNA or protein can promote bone regeneration in animal model. And also we developed some of the metabolic engineer approach to develop this EM with specifically the bone targeting ability. And finally, we also engineering some EM show some potential in the osteoporosis the disease treatment. So this is the summary for my several other projects. So what's next? So we already know, right? So the bone fracture is a significant side effect in the osteoporosis patient. So we are working on one project about we try to implant this EM-TRB scaffold complex into the mouse calaveria defect model in this OS mouse model in osteoporosis disease model. And this we want to check their bone regeneration ability. So if we're successful in completion of this project, we can maybe develop some potential translation approach for use this combined approach for bone fracture treatment for osteoporosis patient. So I'm the scientist, and also I have some medical background. So for exosome-based therapy, I think there's still need lots of work to do. For example, the mechanism of how exosome work in vivo is still the large unknown. So how the exosome working on the in vivo immune cell or progenitor cell. And also there has still lack study about effective the exosome delivery, dosage, and pharmacokinetics. And also the producibility and manufacturing about the exosome is still a challenge. And another challenge is exosome the stability, preservation, transportation. That's still a big challenge. And also they still need improvement on develop some like a rigorous regulator to evaluate the clinical trial or the preclinical trial about exosome-based therapy. In general, exosome is very like a hot topic in American. I believe globally. So they already have many companies there. Now that they switch from stem cell therapy to the exosome-based cell-free therapy. But there's still a lot of challenges. So my lab, we measure the work. We still focus on this exosome-based therapy. So before I wrap up my talk, I want to thank my lab have 4 PhD students and 1 postdoc and other several technicians for their hard work and dedication. And also I want to thank my colleagues and the UMS, the School of Pharmacy, and also I thank my department. I joined this department just 2 years ago. They helped me to set up the new lab. I really appreciate the department and school support. And also I thank several other grant support, including NIH, NIDCI, Osteoscience Foundation, and Princeton University PSCI, their program for their great support. And yeah, thank you for your attention. I want to take question now. Yeah.
Thank you, Dr. Zayabing. A heartfelt thank you for the leading learning session. And now it's time to start our question and answer session. We have with us our panelists, Professor T.R. Bharadwaj sir and Dr. Ravinesh Mishra. Over to Bharadwaj sir.
Thank you, Dr. Jivang Fan, for your very informative and knowledgeable lecture. As we all know that these days, on account of the average age of persons have changed, enhanced, you see. And on account of this enhancement in the average age, the number of patients having osteoporosis has tremendously increased, more so in female specifically, you see.
Yeah,
the area is really very, very specific, need of the time. My question is, what is the present status of exosome mimetics in bone regenerations? At what stage, clinical stage, your compounds or this therapy has reached?
Yeah.
Is there some clinically used formulations or exosome therapies being used clinically also, or is in the clinical trials? So I think
the American FDA includes stem cell therapy. The FDA still put a very strict regulation because they have lots of limitation, like immune rejection or cancer risk, right? Also, the different resource have different stability is not very good. So there are lots of limitation on the stem cell therapy. So that's why so many biopharmaceutical company. Before I stayed at UCLA, I stayed in Los Angeles. I know they have multiple biopharmaceutical company. So they developed a now switch from stem cell therapy into the cell-free therapy, like exosome-based therapy, right? So I already checked the FDA, government website. They already have clinical trial website. So I checked they have over maybe 15 exosome-based therapy globally, right? So they use it for different type of disease, like cancer or diabetes and wound healing and even ulcer or other disease. But for the also for skeletal disease, they have 1 or 2 clinical trial about the exosome for the osteoarthritis disease treatment. But for osteoporosis, I think so far they have no any clinical trial. But that's why it's worse. We can working on this area, so, right? So I think in near future they have clinical trial regarding this exosome-based treatment for osteoporosis. But the osteoporosis is really challenging because most of people, they don't know they have osteoporosis. It's a silent disease. I make an example, like my friends, the mom and 74 years older, she suddenly just slightly fall down, have big craniofacial fracture, right? For young people, maybe it doesn't matter, right? It's okay. But most of people, particularly at old age, when they fall down, they realize I have osteoporosis. But this is too late. I think in the near future, the exosome, maybe they have the exosome-based therapy for osteoporosis in clinical trial, I believe. Yeah, because I know that several companies also working on this area, use exosome for osteoporosis treatment. But particularly for engineering exosome is a very hard topic. How to modify this exosome for different disease treatment.
So in clinical use of these exosomes for bone regenerations, do we have to make use of immunosuppressant, or it's not required, it's biocompatible?
Yeah, that's a good question. For stem cell, they have, but for some drug, I think exosome have the advantage is the exosome show very high biocompatibility. That's one. And second, they already have some immune suppressed function. They don't induce so strong immune response. Third one, this exosome, they can, for example, my lab, we work on engineering exosome, right? So we can use exosome to endogenous, increase some protein or small RNA. That's a very smart approach. If we directly put like exosome with protein together, that's really challenging now. But if we modify like a cell level, right? We modify cell, and this we collect this exosome or exomimetics from cell, they can naturally increase this like protein or small molecular components in the exosome. I think that will the minimize side effect in clinical trial. So that's why I don't think that they use the immune suppressive drug. They can directly inject. Yeah, this is advantage. If we use the chemical drug or other resource like allergenic stem cell, they need an immune suppressive drug. But exosome have multiple advantages compared to stem cell the therapy. So that's why there are so many companies now move to this area. But still challenging. I mentioned then still need a lot of work to do. But I want to point out engineering exosome is the direction now. Many industry, academic, many the scientists are working on this area.
Okay, thank you. Over to Shalini. Thanks.
Thank you, sir. Over to Mishra, sir.
Thank you, Dr. Fan, for delivering such a beautiful and informative talk with respect to the application of exosomes in osteoporosis. So during your presentation, there is a number 34 million affected cases in United States. So as we all are very much aware about, means the country where we used to abide all the means good practices, but why there is a means rise in the number of such cases? So can you let us know about what are the factors, or it may be linked to some neurodegenerative parameters which are going to means affect such a huge number of population? Yes.
Yeah, that's good questions. I already mentioned in the background. So this osteoporosis is a silent disease. So mostly affect the people and age like over 15 years older, particularly in the female, right? But now we still have male. Be careful, male still have osteoporosis, but doesn't mean the male is safe, right? But mostly like the female, they stop many pause, and then the estrogen is decreased, right? After the 15 years older, so the estrogen is decreased, so they start to have osteoporosis. So that's why it's most often they're affected. Like also American, there is now aging, average age is increased now, right? People lifespan is longer, so the society they have more like older people now. So that's why affects 3-4 million people in a risk for osteoporosis. For other countries like Asia, I'm from Asian countries, so I think follow out more like aging increase, right, globally. So each country, they have problem. They have many people will have this osteoporosis disease. But some people don't know, don't care, because looks like I'm okay, right? But when one day they get a bone fracture, they realize, oh, I have the osteoporosis, but it's too late. And that times because their bones already lose a lot of bone tissue, and it's very difficult to treat that bone fracture with osteoporosis patient. That's why some people like old age, they fall down, they get bone fracture, they get a disability. Yeah. So that is most related to female and Asian population increase in our society.
Okay. Thank you. And what will you suggest to our means participants? How means they can? Is there any kind of preventive measures that can be taken care of?
That's a good question. So they have like components like vitamin D, right? You take more vitamin D to some regular exercise, right? Also, you don't want to stay like from the computer every day, stay office. That's not good, right? We want to exercise, we want to need the sunshine, right? But particularly now, the new generation, people, they every day stay office, like just in front of their computer, so never go to nature, right? That's a big problem. Yes, so they need some trash food, right? They don't want to like eat more vegetable, more balanced food diet. That's another issue. Exercise, right? Exercise is very important. So we need to more like a good nutrient. So that is a multiple factor. It's not only related to one factor, right? And yeah, so it's if we should have good like health lifestyle. That's very important.
And sir, you will be pleased to know that here in our lab at pharmacy department, under the leadership of Dr. Bhardwaj, presently we are working on targeted drug delivery system, basically for the colon cancer. So we have also want to work on extending our lab if you support us in case of some exosomes. So that is again kind of a good studies, and we can also analyze our regional areas and some data with respect to the osteoporosis and how we can do better this your hydrogel therapy part, that is your ND and GM part one is.
Yeah. So that's why I
yes.
my colleague encouraged me to attend this conference, and I'm very happy because we want to build a potential and collaboration opportunity. So for this one, I'm totally open. I'm research we need a collaboration, right? We need different expertise. From your institute, I believe you have more advantage. You maybe have clinical data, right? You have more large population have this health issue. But USA, my lab maybe have more advantage. We have engineering approach. We have more like new technology, how to develop better this exosome, right? So we can combine both sides of the expertise to see the potential collaboration. I'm totally open for this potential opportunity, particularly for collaboration. So that's I'm very happy we can talk more after conference.
Yes. Okay. Thank you, Dr. Fan. Over to Madam Salni.
Thank you, Mishra sir. Is there any question from attendees? Dr. Akriti, are you with us? Dr. Akriti, would you like to ask any question? Ms. Kritika? Ms. Kritika, are you with us? Okay. Dr. Zayabing, I have also a question. What are the regulatory bodies or the guidelines that govern the use of that bone regeneration techniques in clinical practices?
That's a good question. So now, so I already point out in the last slide. So it's there's still no like a standard method to evaluate this like a cell or cell biology product, right? This is belong to like a cell or biology product. So there's still a challenge. I think they still have some the criteria. For example, we can use micro CT to look at the bone density, right? So that is a very important part. And also they can check the like osteoporosis. You can use a serum. They have check like ALP or other like bone mark. You can use ELISA to check this one. Yeah, most of it is true, right? CT and serum to check the other outcome.
Okay. And what could be the major precautions specifically for females that should be taken care on daily basis to prevent that osteoporosis? Because it is good to take the estrogen level on higher note or on means limits on right limits. But after taking the vitamin D content in regular basis, the body being adapted to the vitamin D. So what could be the precautions that the female should taken care of in daily basis?
I think daily basis maybe long term is always a lot good, right? we should balance. And I think diet is very important. Healthy diet you can take from foods, right? Rather than always rely on the specific need like nutrients, like vitamin, right? So also do exercise, like regularly exercise, like yoga or some. I play the Teji now. I play with my dean at my school. So exercise is very important. Always control your weight, right? We don't want to like is overweight. That's also cause a lot of problem. So also we control other chronic disease like diabetes, right? This is also high blood pressure. So this is a multiple factor we need to consider. It's not only like rely on the take like a vitamin D. I think it's not enough. We should be maintain our body is very well through the multiple factor. Yeah. Mm-hmm.
Okay, thank you, Dr. Zayabin. We have another question from Kritika Bakshi. Kritika, you can raise your question. Kritika Bakshi.
Hello.
Yes, hello. You are audible, Kritika.
I'm audible. Good morning, Dr. Zayabin. Thank you for sharing your valuable information regarding generation of exosome mimetics for bone regeneration. So I just want to know that what are the main advantages of using exosome mimetics over other approaches such as direct cell therapy for bone regeneration?
Yeah, that is a good question. So stem cell therapy, we already, I think globally, including the many countries, right? So like they already have some stem cell products for different skeletal disease treatment, but stem cell they have some limitations. So one of the limitations, the stem cell can maybe induce some immune response, right? If, for example, I donated my stem cell from myself, right? For the you use my stem cell, maybe if we implant my cell to your body, maybe have immune rejection probably. That is the potential risk. Second, stem cell, they also can maybe cause cancer risk, right? Stem cell, they can, if we implant the body, they can maybe differentiate into other cell we don't want, right? They maybe also grow like uncontrolled. They can proliferate, they can become cancer, the tumor, another risk. And also the stem cell, they also can breed some potential disease. For example, some people donate this stem cell. That stem cell maybe also brings some disease from that donor, right? And this implant this stem cell to the patient maybe have this type of disease transmission. And also the stem cell is not stable, very difficult, some different resource. For example, these people A, maybe younger age, right? 20 years older, maybe donor B have 15 years older. So they donate this stem cell maybe function is totally different, right? So the stability is not is very difficult to control. The stem cell therapy make a consistent result. That's why the FDA now is very difficult to approve any stem cell therapy. So that's why we want to use the exosome. Exosome, so the animal study showed that the exosome also derived from stem cell, right? Stem cell can produce exosome. So this exosome also include information from stem cell, so they have similar function. But stem cell, the exosome have some other like advantage. They have show high compatibility and no like the toxicity, no immune response, and also they can easily inject our body. They can easily uptake by cell. They can provide important information. More importantly, exosome can deliver some small RNA or can endogenous. For example, I presented my topic today, right? We have different approach. We can increase like endogenous expression of some protein, specific protein. For example, we want to like a bone regeneration. We want to increase one like the protein like TRP3. I mentioned that TRP3. So we can use exosome or exomimetic, so we can endogenous delivery this protein. But stem cell is very difficult. Maybe you can transfect lentivirus, right? But if you transfect cell like a virus to the stem cell, that may cause other safety concern. So the exosome, they have lots of advantages compared to stem cell, but also the exosome have similar function as stem cell. So that's why this is a very the popular area now. So there people want to see the cell-free approach. Cell-free approach means that we don't want directly use stem cell, but we still use this type of the particular. They still have stem cell function, right? Because this still come from stem cell. Also, this is still the biology product, right? So they show very high biocompatibility. If we inject the human body most only have less like immune response, so easily accept this type of the biology product. So they have multiple advantages use this exosome-based therapy.
Thank you, Dr. Zabik. And Kritika, hope the answer satisfies you well.
Yes, ma'am. Thank you.
Any question? Anyone question from the attendees? Okay, I think there is no more question, Zayabing. And it's time to thank you once again for your time, for your expertise and engaging presentation. I look forward to apply what I have learned and continue to grow in my understanding of the subject. Thank you so much, Doctor.
Yeah, I also thank you your school to organize this. It's amazing the conference. Yeah, I really enjoy this the talk, and hopefully look forward we have potential collaboration in future, or you can visit us, right, USA. we can talk.
And there is one more request, that please share your presentation with us also.
Okay, thank you. Yeah, look forward to the future opportunity.
Madam Salni, just a minute. Doctor, when there is a question from our audience, Mr. Sunil Korde. He has asked how is the efficacy level of exomal therapy to need evaluated.
So for which type of disease? For the bone disease, for osteoporosis disease, right?
Yes. How is this efficacy level is evaluated in such therapy?
Yes, the therapy there are both only, for example, the osteoporosis, right? The micro CT is like a PET CT or micro CT is very like a common image technology to visualize the bone density, right? So after if treatment, first we want to use this advanced image technology to visualize the bone the density. That's really important, right? So the second, we can use the serum, right? They have different enzyme or protein. So for example, osteoporosis patient, the ALP, the increase in the serum. But after you get a better treatment, the ALP, the level should be decreased, right? So also they have other calcium like level in the serum. For osteoporosis patients, the calcium in the serum increase because they already have the bone cell, like osteoblast cell or bone broken, right? So they release the calcium to our body, right? The serum. So if we get a good treatment, so they increase the bone density, and definitely they will decrease the calcium level in the serum. So I most only the evaluate this also other factor, right? We want to see is any like cause some side effect, right? Is like immune cell response, right? Immune cell that is still can check from the serum, the level. But for bone density, most only use the image technology. We cannot harvest the bone tissue to histology, impossible, right? That's the human, the body. Yeah. So most of the image technology is very also serum uses different the bone marker. We can check the different bone marker level.
Okay, and there are popping number of questions like, is there any kind of markers which are using for this purpose?
Yeah, I already mentioned that ALP, right? ALP, yes. And another calcium, like the calcium, yeah, that the calcium in the But for animal study easier, we have multiple marker, so we can for research for animal is easier. We can do micro CT, we can do PET scan, and after we harvest the bone tissue, we can do histology analysis, like HE staining, like muscle trichotome staining. Also, you can immunohistochemistry staining. You can the stain some like a bone marker like OCN, osteoblast marker or osteoclast marker, right? So they have osteoblast, osteoclast cell. They have imbalance in their osteoporosis patient. So for animal study is more easier, but for human patient, I don't think that you can take some bone tissue from human body. It's very impossible, right? So most often they use the image and take the serum to measure the marker.
Okay, thank you.
Yes, thank you.
Thank you, Dr. Zayabing. And dear participants, now it's time to take a short breakfast break. Stay connected with us.
Yes, thank you again.
Thank you, Dr. Zaya. We will join after 1 hour break, and it is my request to all the participants to please join on time.
Okay, see you. Bye. Thank you.
Bye.
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 Hello. Welcome back, my dear participants. We are going to start our next session with Dr. Arif Noor Mohammed. Please keep your mics off during the session. Now, I'm taking the opportunity to introduce our next speaker, Dr. Arif Noor Mohammed. Dr. Arif is a visiting scholar, Division of Genomics and Transcriptomics, the Joint Research Center for Human Retrovirus Infection, Japan. He is a member of the American Society for Microbiology, United States. He is a member of Indonesian Young Academy of Science Indonesia. Also a member of European Virus Bioinformatics Center, Germany. He is a founder of Jalan Tengah, Indonesia. He was selected as a keynote speaker in international conference in Uttaranchal University, Dehradun, India. He was awardee of PMDSU Scholarship Batch 3, Ministry of Education, Culture, Research and Technology of the Republic of Indonesia. Dr. Arif, welcome to online international conference. Over to Dr. Arif.
Thank you. Thank you so much for the invitation.
Dr. Arif, you can share your presentation.
Okay, thank you. So, could you see my screen?
Yes, sir, it is visible.
Okay. Okay, thank you very much for inviting me in this international conference. Thank you for Professor Misra that I met in Uttaranchal University in Dehradun. Our title about the global perspective in integrating bioinformatics and biotechnology for traditional medicine advancement and the case of jamu development in Indonesia. I'm Arif from Postgraduate School Universitas Airlangga, Indonesia. Okay. Universitas Airlangga is, sorry. Okay, Universitas Airlangga is including in the top 500 in the world by QS and Times Higher Education, and number 3 in domestic rank. So for the undergraduate and master student, you can join the Amarta program at my university. You can choose many things, subjects that you want, and of course about the jamu development. You can try many things of including the tropical medicine aspect. And also, if there are many researchers in Baddi University that want to conduct research in my university, we have a program called Aranga Postdoctoral Fellowship, and we have so many members from India, of course, and you can join in this program and like a short visit in my university, and many things you can do online, and you can do the research and publish the research in Scopus Index Journal. So in my city, in Surabaya, we have Taman Husada Family, or Taman Husada Medicinal Plant Gardens. This initiation developed by Universitas Erlangga and Intiland Development, and we have so huge medicinal plant garden that consists of many families of medicinal plants like Leguminosae, Zingiberaceae, Lamiaceae, Compositae, Apocynaceae, and many things. This is the pictures of the flowers of medicinal plants. And we have more than or almost 500 species in this medicinal plant garden. So you can visit and develop with us together. Yeah, this is Surabaya, my city, that's so beautiful with the mangrove forests and the huge bridge that connecting from Surabaya to Madura Island in Indonesia. Yeah, as the vlog before, and this is my profile that I'm a member in European Forest Bioinformatics Center and many institutions, and then I'm also the faculty adjunct faculty member in Uttaranchal University. So I also develop many things as an editor. This is my profile in European Forest Bioinformatics Center, and I'm an editorial board member in many journals, in a Scopus Index journals, and also reviewers. Yeah, this is my profiles in Scopus, and I just selected as a top 2% scientist worldwide by study by Elsevier and Stanford University, and I'm focusing on Garcinia mangostana or mangosteen. That if you know about the mangosteen, yeah, we develop it in Indonesia, and my president and Narendra Modi, Prime Minister of India, is a best friend, and I hope we can develop many things together between India and Indonesia. Yeah, Indonesia is a mega biodiversity country. The second biggest biodiversity in the world. A high number of indigenous medicinal plants. Indonesian tropical forests consists of almost 150 million hectares and home to about 80%, yeah, 80% of the world's medicinal plants. And Indonesian tropical forests have more than or almost 30,000 plant species. It's a huge number. And Dictionary of Indonesian Medicinal Herbs stated that more than or almost 3,000 plant species can be developed for medicinal purposes. It's so wonderful that we have so many medicinal plants that we can use as a potential drug discovery. National Agency of Drug and Food Control, or BPOM in Indonesia, stated that almost 300 plant species officially registered and already known about their medicinal use. Indonesian people, especially in rural areas, they use traditional herbal medicine known as jamu to treat many diseases. Jamu, a word in Javanese tribal language called traditional medicine that originated from plants or medicinal plants, yeah. And also jamu gendong, the type of jamu like this kind of traditional jamu sold without a label and freshly prepared, not preserved, and consists many plant materials. This is a woman that sell jamu gendong. The sellers bring the jamu from door to door, and the word of gendong, yeah, jamu gendong itself means to carry something on the back of a body, like this. It's ethnomedicine about the jamu gendong. And nowadays, production of jamu developed on an industrial scale. The Indonesian government, industry, and academia, they do a collaboration to further development of jamu and extensive research required to establish the safety and efficacy about the many traditional jamu preparations. And about 110 companies production of Indonesian traditional medicines. Almost 90 are classified as IoT or industry obat traditional or traditional medicine industry, and almost 750 as IKOT or industri kecil obat tradisional or small industry of traditional medicine. So Indonesian government divided the medicinal plants as 3 categories. First, jamu, and then standardized herbal medicines, and the last is phytopharmaca. All preparation have to meet that basic safety criteria of the jamu formulation. The therapeutic effects of jamu supported by the empirical data. The efficacy of standardized herbal medicines be proved in preclinical trials, standardization of active ingredients required for the efficacy of phytopharmaca. Clinical trials have to be carried out. Yeah, it's mandatory to assess the medicinal plants' efficacy. Yeah, this is the economical perspective in jamu industries. The company called Sariayu Martatilaar, they developed a postpartum herbs that consist of these plants from the rhizome and folium, herb, floss, radix, and many things about the parts of the plants, of the medicinal plants. And these components can be relieves abdominal pain after giving birth, is excrements and vaginal inflammations, stimulates blood circulation and improve appetite and digestion, as well as strengthening and promoting health generally. So they have so many benefits for our health. And also Fapros, they developed the product called Menstralax. This compound consists of from this plant, from the cortex, radix, rhizome, floss, and many things from this species, and regulates endocrine gland secretion and menstruation, promotes ovulation, reduce menstrual clots, and many things. And also another industry, Deltomed Laboratories. They developed the strong bass ginseng that consists of Zingiberaceae, Panax ginseng, of course, from the radix, and it can be increased vitality, relieves backache, sore muscles, fatigue, and many things. Also, they also developed the Antangin GRG that consists from Zingiberis rhizome, Panax ginseng extract, and then many things about many species. And this product can be effectively combats as cold and alleviates its symptoms such as fever, nausea, bloating, cold sweat, and fatigue. And in Surabaya, in my city, we have Jamu Ibu Jaya. Jamu Ibu Jaya developed hypertension and diabetes. It can be used to treat the mild hypertension and treat the diabetes mellitus. They consist of Dinospora caulis, Andrographidis herba, and many things, many parts of any medicinal plants. Also, the Petrochemical, they develop Sentia that can be treat stomach pain and diarrhea, and also Puspo internusa, they develop Pacakap diabetes to treat the diabetes mellitus. Now, biological activity of the most common plants in Jamu. Jamu consists of many plants, especially Zingiberaceae, the family of Zingiberaceae, like Zingiber and Alpinia and many things of genus. And also Curcuma domestica. Curcuma domestica, the well-known about the curcumin, and curcumin have so many benefits to our health to treat many diseases like hemorrhoids, diarrhea, asthma, and many things. Also, the new study developed that curcumin, gingerol, and any compounds can be used as an antiviral development. We developed the study based on the bioinformatics and biotechnology against the virus from the compounds of Indonesian medicinal plants. So, Zingiber officinal, or in Indonesia we call it jahe, and they have many compounds like gingerol, paradol, and many things. It can be used to treat headache, anorexia, cholera, influenza, anemia, malaria, coughing, and vertigo. As a tropical country, Indonesia have so many infectious diseases like malaria, dengue, and other parasites. So we can develop it many medicinal plants as drugs against the agents of tropical medicine or infectious diseases. Yeah, this one is Alpinia garanga from its rhizomes. With the oil extraction, we can reveal this compound, and with the aqueous extraction, we can develop this compound, and these compounds can be used to treat anorexia, dermatosis, malaria, gastritis, and so on. And one of the most popular medicinal plants is Cymbopogon citrates. The leaves can be used to our dish and many things. And these compounds, limonene and geraniol, can be used as to treat dysuria and then rheumatism, gastritis, and many things. Yeah, this one about the Andrographis paniculata that have so many parts as medicinal medicinal plants, like the aerial part and then roots and leaves. They have many compounds like andrographolide, xanthone, and andrographapine. These compounds can be used to treat many diseases, like antidote for many poisonings, fever, diabetes, and then eczema, appendicitis, common cold, epilepsy, syphilis, and many benefits for our health. So, one of the important medicinal plants is Philanthus. Yeah, we developed Philanthus as a product of phytopharmaca that developed by one of alumni from my university. They developed a product to induce our immune systems that I will told you in the next slide. So from these whole plants with the aqueous extraction process, we can develop or reveal many compounds like alkaloid flavonoids, terpenoids, tannins, lignans, quercetin, and many things. And we can develop it to use against asthma, epilepsy, malaria, constipation, lowering blood pressure, diarrhea, and so on. Aloe vera is important too. Induce many activities against diabetes, tuberculosis, anthelmintic, hemorrhoid, and so on. And then Orthosiphon aristatus, it can be used to prevent diarrhea, colitis, menstrual disorder, and others. Yeah, Jamu goes to international because jamu just designated in the end at 2023 as intangible cultural heritage. Yeah, it's so important to improving many things about jamu and share many things about the benefits of jamu to the world, from Indonesia to the world. And jamu or Indonesian medicinal plants already invented by our ancient generation, and they create a relief in a Borobudur temple, the biggest temple in the world. You can visit Borobudur temple in Indonesia, and you can find this relief that we can see that this is the seller of jamu, and how the king or queen from the kingdom in Indonesia previously in the ancient era, they also use jamu and develop and consume the jamu as a daily use. And this is the old photo about the jamu gendong seller around 1800. Yeah, jamu goes to international. Indonesian herbal industry enters the global market. This is already published by Statistic of Indonesia, the data from Ministry of Industry. They stated that export destination countries such as Australia, Taiwan, Singapore, India, Hong Kong, Japan, Malaysia, China, and other countries. And export of Indonesian jamu are increasing during the pandemic because the Indonesian traditional herbal products can be used to treat the COVID-19. Yeah, jamu is a traditional medicine from Indonesia, which is made from natural materials such as roots, bark, flowers, leaves, and fruits. Also the seeds of the plants. And the benefit of maintaining health have been known since ancient times that I told you before in the previous slides. Indonesian jamu and herbal supplements are made using natural ingredients that have passed several tests and using the latest technology. Indonesian cultural heritage continue to be maintained during this process. Based on the statistic of Indonesia, they stated that total export value of jamu in 2021 will reach almost 50 million US dollar, and increase more than 10% compared to 2019. This is the jamu gendong in the modern era. You can see here mostly made by Zingiberraceae family of the medicinal plants, and the cinnamons, and this one is turmeric and ginger and things like pogon. Yeah, this one they also they how to create jamu, the process. And then as I told before, jamu ibu jaya from Indonesia, they develop many products that we can serve as a nice drink, based on the ginger, based on the turmeric, and so on. Yeah, this one, the products. This is aloe vera, mangosteen, and then ginger, and rosella, and so on. Also, the red ginger is so famous in Indonesia. They can develop to treat many problems in a stomach, and also sometimes they add honey and orange and others. Yeah, Stimuno Forte or Stimuno, it can be used to maintain our immune system. This product already developed as a phytopharmaca, and then this product already sent to many countries. And Stimuno, the name of the product, is made by famous niruri that we can develop it. This one of the plants. Also Herbamuno, almost the same components with the Stimuno, but this one from Mustika Ratu. And Tolak Angin is made by ginger. Yeah, this is the jamu products from my university. Diabet Call is made by mangosteen or Garcinia mangostana, already formulated to the capsule, and we can directly consume it without any doubt. And this one about the Mediti. Mediti is green tea extracts that they have so many compounds like epigallocatechin and many compounds to treat cancers and other diseases, including the infectious diseases. Yeah, Kriya Herba by Jamu Ibu in Surabaya. We can see the store that serve jamu as a modern drink that we can enjoy in a cafe. So, Borobudur Extraction Center is the new industry to develop the Indonesia herbal medicinal plants. And they process many things in high quality products, and they serve the best quality to producing herbal extract of Indonesian species. Yeah, jamu or Indonesian herbal medicine, they have many or three stages or three categories. First, jamu, and then standardized herbal medicine, and then phytopharmaca. What is the difference between jamu, standardized herbal medicine, and phytopharmaca? Here is the details. Jamu is fulfill the quality requirements and empirically proven efficacy. And then for the standardized herbal medicine, fulfill the quality requirements, efficacy proven scientifically or preclinical, raw material use standardized. And then the last category, fulfill the quality requirements and then efficacy clinically proven. So many things that include in phytopharmaca already have already passed the clinical trial. Raw materials used standardized. So this is the process for jamu from the raw materials directly to manufacturing, and they directly use or competitive jamu and sell to the directly to Indonesian people. And the second category is standardized herbal medicine. From the raw materials, they come into the manufacturing process, extraction process, preclinical tests, stock formula, and then they directly sell to the product. And then phytopharmaca from the raw materials move to extraction process, preclinical test, stock formula, and clinical tests. After that, they develop as a phytopharmaca product. The phytopharmaca product have so many long process if we compare with jamu. Yeah. Trust area for botanical research. We know that plant biotechnology, marker analysis, traditional medicine, organic farming, and then natural products, lead compounds from medicinal plant, system biology approach, genomic technique, evidence-based research, formulation and development, reverse pharmacognosy, they can become one to induce the development for medicinal plants. And the laboratory of herbal product, they have some many categories like phytochemistry and phytochemical analysis, herbal pharmacology and toxicology, pharmaceutic and processing, herbal analysis, and standardization. So standardization is the most important thing to make sure the product is safe and easy to consume. So this is from the raw materials. We know that source of chemical diversity, pharmacophore-based virtual screening using bioinformatics systems, and the conservation and propagation is so important to prevent the extinction of the species. And molecular pharmacognosy, it can be combined with biotechnology and bioinformatics, biomarkers and pharmacogenics for quality, multi-target herbal therapy, combinatorial phytotherapy. It's so important to produce the compounds. Yeah, we can see in this slide that medicinal plants, and we can identify the single compounds and then test the single compounds in vitro, in vivo, and clinical trial in human, and or molecular phenotyping. It can be inspired new medicines with a bioinformatics and biotechnology application. Biotechnology application have many important things like transcriptomics, metabolomics, phylogenomics, and then this one to improving the natural product biosynthesis and metabolic engineering to enhance the yield of the compound. We can see here that molecular genetics of specialized metabolism in model plants. And from the traditional herbal medicine, we isolated the quinine, and we can test and using the application of bioinformatics and biotechnology, we can develop or we can manipulate the production of the compound of the single compounds, and also the adaptive functions of plants, specialist metabolism can be used to prevent the plants extinctions because of the weather, because of the global warming, and many things. Yeah, here is the medicinal plants, and to create anti-COVID drug discovery and development, we need to identify the traditional medicines, herbal preparations, functional foods, and many thing aspects, and then we propose the mechanism of actions. We can do any clinical trials, and after that we can reveal about the anti-COVID drug discovery. From these plants, from the lithospermum, from the young leaf and callus, we can develop the plant cell and subculture and cell suspension culture, and then we move to the cell culture in bioreactor. From the bioreactor, we can develop the downstream processing, and we can develop the natural medicine. This is the process that we can induce by biotechnology. Also this one, and also this one from the hairy root culture, cell suspension culture. We can develop many things, and we can reveal the bioactive compounds, specific bioactive compounds. Yeah, in Indonesia we have this website, IJA Analytics, so we can use for free for this web server, and we can analyze the compound and plants and the proteins target of the mechanism with a bioinformatics way. So we also have the Jamu Herbal Medicine Research Center in Krida Wacana Christian University, specific research center for Jamu. And we also have a Journal Jamu Indonesia, or Indonesian Journal of Jamu. Yeah, it's so interesting to develop our thinking about Jamu. And Jamu already found in Scopus almost 300 documents. This one is one of the important paper by Professor El Fahmi from ITB Bandung, Indonesia. They developed many things about the artemisinin process to reveal the single compound using plant cell culture, or maybe using other species like Saccharomyces cerevisiae. They manipulate the biosynthesis process in artemisinin products. Also, this is my friend Dr. Johan from Surabaya University. They developed the ginseng or Panax ginseng products using plant cell culture and bioreactor. And this university already developed with the industry skill and enhance the scale up the product directly to industry. And this is for my university, the products of jamu. And this is my lecturer, Professor Mangestuti, that developed jamu in Indonesia. And this is my friend from ITS Surabaya, Indonesia, and member of Indonesian Society of Young Academy of Science. Yeah, this is our scientific publications about the antiviral development using Sonchus arvensis with N-hexane fractionation with the collaboration of Chulalongkorn University. Yeah, we analyze the chromatogram and analyzing the using GCMS analysis about the compound, and then after that we perform or identify the bioactivities. After that we do the bioinformatics research or study, and then we develop and suggested that this plant can be used or potent against the SARS-CoV-2 virus. And this one, the nanoparticle of Anonas squamous leaf extract against the cancer cells. Yeah, this one. It's so potent chemical that still under process in my laboratory to scale up these compounds. Yeah, this is my study about the mangosteen against the SARS-CoV-2. Yeah, from alpha mangosteen, beta mangosteen, and gamma mangosteen from the compounds against the papain-like protease and then main protease as a target proteins. So we also developed the herbal combination of Moringa oleifera and Curcuma longa, and we can see we have so many international collaborations with the Netherlands and then Russian Federation. Yeah, this is the compounds, and this is the targets of protein. Yeah, they are so potent as anti-inflammatory and antiviral. Yeah, this one is the result. And also, we developed the anti-dengue virus. We test the Ananas squamosa leaf extract against the dengue virus type 2 that isolated from Surabaya, Indonesia. We have a collaboration with Filipina and then China to develop these medicinal plants. Also, the quercetin as an anti-cancer candidate. We already published it, and you can enjoy our publication here in Karbala International Journal of Modern Science. And this one is a finger root or Bosenbergia rotunda as atherosclerosis, anti-atherosclerosis. We also developed the topical administration effect of sargassum and garcinia extracts. So this combination can be developed as induced wound healing process in diabetic. We test in vivo, and we are in process to develop it in human trial. This is the results. And also we develop many kind aspects of Garcinia mangostana, especially from the carcinosantons against SARS-CoV-2. Yeah, this is my undergraduate thesis project that I finished in 2019, 2016, yeah, 2016, about the renal protection by Garcinia mangostana pericarp extract in streptozotocin-induced diabetic mice. And we developed the narrative review about the mangostin and the potential of gamma mangostin as antiviral agents. Yeah, we developed it. So, the conclusion of this talk and future directions are medicinal plants potential to be developed as herbal medicinal products or as sources of new therapeutic agents. Plants in jamu investigated scientifically for their biological activities. We need more investigation, more research to explore many things about Indonesian herbal medicine. Jamu makers or industries should be standardize the formula of jamu in order to assure the efficacy and safety. Efficacy and safety is the two main part about the standardization. It's so important to develop this kind of efficacy and safety. A challenge develop jamu into standardized extracts and phytopharmaca. So this one is the challenge to develop jamu into standardized herbal medicine and phytopharmaca because we need more long research and need investment from Indonesian government, pharmaceutical industry, and academia to develop jamu to phytopharmaca products. Qualitatively better, safer, and more effective products, it can be contribute to the quality of healthcare in Indonesia. Of course, it can be contributing to the enhancement of healthcare in Indonesia. Maybe it's so similar with the Ayurveda in India. Further research, we should perform to evidence the traditional use of jamu scientifically. Exploration of medicinal plants also cover ethical issues, efficacy, safety, IPR or intellectual property rights is so important, benefit sharing, and biodiversity conservation. Biodiversity conservation is so important to prevent the extinction. Okay, thank you very much, and back to the committee.
Thank you, Dr. Arif, for sharing your expertise with the jamu formulation. We have with us our panelists, Professor Bharadwaj sir, Professor Tejveer sir, Professor Avinash Mishra, and Dr. Bhartendu to start the question and answer session. Over to Bharadwaj sir. Sir, unmute yourself. Sir, please unmute yourself.
Yeah, am I audible now?
Yes, sir. Yes, you are audible now.
Dr. Arif Noor.
Yeah.
Yeah, for your very valuable. You see, we also in India have numerous traditional medicines
Of course.
Used since long. You see, traditional means for the years they have been used, and still we are in using those compounds. And to know about the jamu, it's again very amazing that it has so many components, so many herbs in it, isn't it? I would like to have one query regarding clarification rather, that in one of your slides you have shown that the cinchona bark, which has from where the quinine has been isolated, isn't it? It's identified, and of course there are other four stereoisomers, isn't it?
Yeah.
That means four compounds: quinine, quinidine, cinchonidine. These are the four components, main components, and quinine, of course, is having the maximum antimalarial activity. So, have you used one of these for the standardization of herbal drug, and similarly for others also identified? And if they are used, have you gone for the HPLC also, or only the TLC or other kinds of standardization procedure which have been used? What are the approaches with respect to the herbal standardizations which have been used in the standardization of jamu?
Okay, yeah. Thank you, Professor. So, in jamu development before, in traditionally in our community, we develop the jamu, and then in industrial scale, we scale up the jamu to the phytopharmaca product and standardized herbal medicine products like a stimulo products to induce our immune system, like in my presentation. And based on our based on Indonesian government policy, the standardized depends on the our government, and our government assess the products, and then if the products are already clear and no problems in the products, they can they can sell to the community, and also export the products to other countries, and many countries like Japan, Malaysia, and others.
So in this standardization, only the safety part has been taken care of. That means it is not toxic, it is not having other side effects, unwanted effect. Or is there some scientific equipment, approach, modern ethical method which have been used in its standardization?
Yeah, of course. So from the beginning process, the industry do the many research about the basic research about the jamu components, and then they use the in vitro study, in vivo study, and human clinical trial.
I see.
Clinical trial, of course, do in a phytopharmaca product, especially for phytopharmaca product. That's why phytopharmaca product have a long-term process to develop, because the government, our government policy needs to many types of assessments, like efficacy assessment, and then safety assessment, and so on.
Okay, thank you.
Thank you.
To Shalini.
Thank you, Dr. Arif, for awareing about your traditional jamu preparation that is known globally, and we have seen the export means revenue generated to Indonesia. So can you let us know what is the main purpose of using jamu for everything or for some certain preventive measures or for specific diseases?
Okay. Thank you, Professor Misra, for having me in this international conference. It's so wonderful event. Uh, Regarding your question, so based on our daily use using jamu, Indonesian people using jamu as a daily use, like maybe they drink it in the morning or maybe during in the night, and almost every day we consume the jamu of maybe we can create the ginger with the hot water, and maybe other people using turmeric in the hot water, and other people maybe combine it turmeric and ginger with the sugar, a little bit sugar, like brown sugar, and many thing aspects about the medicinal plants can be directly consumed by our communities. It's so interesting, and previously our team developed the study in ethnomedicinal study in some uh, specific areas in a forest, near the forest. So we revealed that they have unique drink and or unique jamu in every areas, as also different parts between in a city or maybe in other places. Yeah, it's so interesting, and we should develop more scientifically proven about this jamu and other products.
That's good. Now our colleague Dr. Tejvi will interact with you. आप बोलो कहीं से भी आवाज जाएगा कहीं से भी बोलोगे.
Okay, very interesting talk. I would like to ask about the quercetin. Did you check the percentage of quercetin?
Okay, the quercetin, yeah. We developed the quercetin in bioinformatics research. So we are in a process to do more research in vitro and in vivo areas about the quercetin. Quercetin is and is still under development in our lab, in my lab.
Yeah, because it is an antioxidant and used in tea a lot. So that's why I'm interested. We published a paper, and quercetin is known as antioxidant and is a constituent of tea.
Yeah, of course. That's correct. And our team developed the bioinformatics study to know, based on the data and based on the previous study, what should be done in the next or the future study about the quercetin. Because in our study, we develop it as an anti-cancer and anti-atherosclerosis, and also antivirus. Our study revealed that quercetin have good effectivities against the many virus. Yeah, because my study focusing on the virology aspects of this one.
Thank you. Thanks a lot.
So there is one very interesting question from one of our participants. So they have asked, does the efficacy of jamu matched with the synthetic drugs?
Okay. The jamu efficacy data match to the. Oh yeah, of course. In Indonesia, previously, we explore the industries, explore the jamu efficacy, and based on the data of ethnomedicinal data, they developed the synthetic drugs to develop like a single compound drugs, single compound drugs, and the result is match between single compound drugs and jamu, and there are no problems about that. Then one of the single compound products already established, and about the synthetic drugs, I think in synthetic drugs, I think there are so many problems because maybe because the single compound, and in jamu they have so many compounds that can be in use, or maybe they can they can uh, help the other compounds, and then these compounds more than around more than 100 compounds in one or maybe three combination. It can be used to develop and treated many diseases like diabetes and so on. Yeah, it's so interesting.
Okay, thank you. Does this preparation have been approved by international regulatory bodies?
Yeah, of course, for phytopharmaca products already checked by the international board for the development of the drugs, like phytopharmaca products. About the in my slide about the uh, Stimuno is a phytopharmaca products that can be induce our immune system, already export to other countries. Yeah, it's interesting.
Okay. So thank you, Dr. Amir, and we are very glad means your university and you are very much interested. So again, we are going to have some separate discussion regarding the collaboration. Already our senior professor, Dr. Bhardwaj, Dr. Tejveer has interacted with you. So we will sign some bilateral projects so that we can make a benefit of it for the society of Indonesian as well as the Indian community. Thank you so much.
Thank you so much, Professor Misra and others, and other committee for this conference. Thank you very much for having me in this conference.
So, Dr. Bhartendu, do you
Good
want
morning.
to ask?
Good morning, Dr. Arif. Thank you so much for this. I just want to ask one thing, that if we integrate this bioinformatics and biotechnology on the development of Jammu, then what is the socio-economic impact on this local communities?
Okay, yeah, it's so interesting question, because I'm not focusing on the economical aspect, of course, but as I know, the Jammu development, we can enhance the economical value of Jammu in our community, because the Jamu gendong that selling freshly and made freshly by the jamu seller already so famous in Indonesia, and many people can directly buy to the seller in home by home or door to door to our village, or maybe in the city. Yeah, it's so interesting, and our branding to develop the jamu, it's so good to create the better performance about the jamu seller, and also the increasing of economical value about jamu.
Thank you. Thank you so much.
Thank you to our panelist. Dr. Arif, I have also a question.
Yes.
Is there any standard parameter or test that you are using for the evaluation of jamu, specifically for the pharmacokinetic parameters like the metabolism of the jamu in the body or clearance from the body? Because I think when there are so many compounds together in a one formulation, that will create some unnecessary burden on the kidneys to clear the metabolites of that all herbal drugs. So is there any pharmacokinetic parameter?
Okay. For jamu development, we don't use the modern parameters about this development, but for the phytopharmaca products and standardized herbal medicines, we use common policy about the many parameters, yeah, especially just for the standardized herbal medicines and phytopharmaca products, not for jamu. So jamu jamu is a traditional one made by the seller without any parameters about the about the pharmacokinetics and others. Yeah, it's traditionally used. Yeah.
Okay. Dr. Arif, our herbal medicinal expert, Professor V.K. Kapoor sir, has also joined with us. Welcome, Professor V.K. Kapoor sir.
Thank you.
You may please ask your question or you can interact with Dr. Arif.
I'm sorry, I have not heard in completion of his lecture, so I do not know exactly the contents. So I will apologize for that.
Basically, Professor Kapoor is one of the renowned scientists in India and has contributed a lot for the development of your various extraction technique related to the herbal pharmaceutical. And he is the part of the team of Professor Harkishan Singh, Professor Bhardwaj. Means he has received the highest contribution research award from the government of India, like Padam Shri awardee, his mentors and team leader. And part of the team, the drug which is discovered by any Indian university, that is only one drug is chendonium iodide. So I would like Dr. V.K. Kapoor to let you know about his discovery of journey of chendonium iodide to you.
So, I would like to add a few lines here. That Professor V.K. Kapoor has been involved and the person responsible for standardization of herbal drugs. All our pharmacopoeia, including Ayurvedic pharmacopoeia and others, he is the person who has been involved and produced all those monographs. I think more than 20 or 20, how many? Around 20 monographs on the standardization of herbal drugs, including all those drugs which are main, have been again prepared and by Professor V.K. Kapoor and his team. So it's a matter of pride for us that he's with us at Baddi. And in fact, that's why I was asking you that if you can send your jamu, we can again go for its standardization as per Indian system also, you see. The scientific approach, maybe like okay.
Yeah. It's amazing work.
Pleasure.
Nice to meet you, Professor.
Thank you.
Thank you, sir.
Yes, sir.
Yes. Is there any question from attendees? Attendees can also ask their question by raising their hand. Is raise. Archana Chaudhary. Archana.
Good morning, sir.
Yeah.
Thank you for your valuable words. I have one question.
Okay, thank you.
What challenges are faced in the integration of bioinformatics and biotechnology into JAMU development?
Sorry, it's not clear.
What are the challenges that are faced in the integrations of bioinformatics and biotechnology into JAMU development?
Yeah, okay, thank you for the question. Sometimes in the bioinformatics area, the results of bioinformatics research should be confirmed again in vitro study and in vivo study, and also in a clinical trial test. So we cannot reveal direct conclusion in a bioinformatics study. So we need biotechnology to induce enhance many research areas like in vitro, in vivo, and clinical studies like that.
Anand Suman.
Yeah.
Anand Suman, would you like to ask?
I have one query. Hello?
Sir, yes. Yes, sir.
I would like to be knowledgeable about the traditional system which is followed in your country. For example, in India, the traditional medicinal system is Ayurveda. In Chinese, they have their own Chinese system of medicine. Which system of alternative medicine do you follow in your own country? Because if you have to select the drug for investigation of any potential therapeutic compound, which traditional system would you follow?
Yeah, okay, thank you, Professor. In Indonesia, we have our own system. Yeah, we don't follow.
जो सन ले हम क्वेश्चन पूछे थे उसी का आंसर नहीं समझ में आया वही पूछेंगे.
We have our own system, and we don't follow Chinese or other countries. But in Indonesia, based on our research, many traditional products have similarity with India, have a similarity with Chinese or maybe Japan. Yeah, a little bit. Yeah.
Thank you.
It's so interesting.
Is there any documented, composition or compilation of pharmacopoeial type,
Indonesian pharmacopoeia type?
Yeah, of course. Of course, we have the big book, a big book for standardizer. Yeah, like pharmacopoeia for herbal products.
Okay, that's right. Thank you. Thank you.
Thank you.
Thank you, Dr. Arif, for sharing a knowledgeable session. For next proceeding, my colleague, Dr. Priyanka, will take the opportunity to introduce our next speaker, Dr. Vaishali Patil. Over to Dr. Priyanka.
Thank you, Shalini ma'am.
Dr. Priyanka, you are not audible. Dr. Shalini, Dr. Priyanka is not audible.
Okay, sir, I'm checking. I'm just let her in.
Hello. Am I audible? Hello, am I audible?
Yes, you are audible.
Okay, thank you, sir. Good morning to everyone. Thank you, Shalini ma'am, for giving me this opportunity to introduce and welcome our next speaker, Dr. Vaishali Patil. Dr. Vaishali Patil is working as a principal head at Charak School of Pharmacy, Chaudhary Charan Singh University, Meerut. She has more than 16 years of professional experience and her research interest in medicinal chemistry and CAD. She has been confirmed with various prestigious awards, notably Dr. APJ Abdul Kalam Technical University Best Teacher Award. She also has Most Cited Paper Award 2022 and Brand Ambassador for Bentham Science Publisher and has received research grant for government organization. She has delivered several lectures on application of rational drug design across the world. She is commonly member of American Chemical Society and life member of APIT India. She is editorial brand member of Index Journal. Welcome, Dr. Vaishali Patil. Over to you. Dr. Vaishali, are you there?
Hello, Vaishali ma'am. Panchul sir, are you with us? Raman sir, Panchul sir, that same sir.
Yes, madam.
Please.
Am I audible, Ms. Akshita?
Yes, ma'am. Yes,
Chaitanya, sorry, Dr. Chaitanya.
Yes, ma'am, you are audible.
Okay, thank you. So let me share my screen. Bolo.
Bhardwaj sir, kindly unmute yourself.
Yes.
Sorry,
Is my screen visible?
Sir, not unmuted yet. Yes.
Yes, Miss Shalini, is my screen visible to you?
No, ma'am, it is not visible to me. Is it visible to Panchul sir or our IT personnel?
It is not visible, madam.
Now it
is. Yes. Yes.
Okay. Okay. Thank you. So first of all, good morning and namaskar to all the respected sir from Baddi University, Professor V.K. Kapoor sir, Professor Bharadwaj sir. So regards sir from myself, and it is really an honor to be associated with the conference which is being organized by Professor Ravinesh for a long time. I think every year these conferences are being organized and various good knowledge sharing sessions are being provided so that in the online mode also the audience can take the benefit. And thank you to the moderator for introducing me. So first of all, I will like to talk about the topic which I have selected. That is about the drug repurposing methods and challenges associated. So for the last 3-4 years, right after from COVID outbreak, and even before that, we're struggling with various health associated disorders. Maybe it is lifestyle associated diabetes, or it is the mutational, or we can say the complicated cancer, which has been creating havoc among the all over the globe. And these diseases have shown the need that we need to accelerate the drug discovery process, and immediately or based on the requirement, the drugs must be made available. So drug repurposing, it is not a very new terminology. I think most of us must be knowing about that. Like a very common example of aspirin that has been used earlier as an painkiller, as an NSAID, and later at reduced dose, it has been used as it has been clinically approved for use in cases of anticoagulant for for cardiovascular ailments. So it has been there in the pipeline of drug discovery process, but but over the years or over the last 2 decades, when we have seen how it can be applied, or what are the best practices while adopting the drug discovery, while adopting the drug repurposing, some observations have been made, and in my talk I have tried to present all those. So with this, I will like to initiate my presentation or my talk. Oh, my system is not responding. Yes. So in my talk, I am going to cover up about the details about what is the generalized term drug repurposing, what are the various available approaches, then what are some examples of application of drug repurposing in cancer, as well as some cardiovascular diseases, and what are the associated challenges, and how we can go in the future by adopting these practices. So when we talk about drug repurposing, so it is generally the available drug which has been well established in the clinical phase, or well established for the clinical use. It is reused, or it is indicated for some other ailment or some other disorder for which it was not indicated earlier. So that is drug repurposing. So there are some other terminologies also, that is reusing, rediscovery or reprofiling, drug repositioning, that right from cancer to some diabetic or in vice versa. So the new indication is not earlierly prescribed, or it was not earlier suggested. So that is in a simple term, drug repurposing. And in this process, the main objective is to shorten the drug discovery process. As as we can see, the common drug development or drug discovery timeline is very lengthy. So right from target identification, or we can say identification of the disease, first incidence of the disease, then later on we identify the compounds or we identify the drug first of all. For that we do experimental work, like the traditional synthetic approaches are taken, or we can go for computational drug screening, where we can screen the large data sets with the help of computer-based softwares. Like in that we can take help of ADMET screening, where we can check pharmacokinetic and toxicity parameters. We can go for molecular dynamics as well as simulation studies, and we can identify the potential hits. Then we can take them directly for evaluation. So that is preclinical phase, which is almost which also takes more than a year, where we can go for in vivo or in vitro studies. Pharmacological, kinetic, and toxicity testing is also required to be done. And then we could come out with some mechanism, or we can establish some efficacy of the drug. Then later on we go for the clinical phase, which is having various phases. 3 phases are there, and that phase is not also even very small. It also takes 4 to 7 at a minimum, or many a times the drug fail in either of the phase, and we need to stop the project, and again the work starts from the scratch, or the problem needs to be identified why the drug failed in that particular phase of the clinical trial. And if there are a few candidates, like very few candidates could pass into the clinical trials, and then later on it goes for the review and approval by the FDA, which is again a lengthy process, and it may take years to be completed. And later on, if the drug is successful there, then it is being made available in the market for clinical application. There also, it is still under observation, and if any side effects or any adverse effects are encountered, the drug need to be taken back from the market. Again, it need to be properly analyzed, the reason behind it. So this drug development or discovery, it is a very complicated process, but being complicated, it also takes lots of time. So much manpower is required to be put up, and the chances of success are very less. So in sense of that, if we talk about drug repurposing, so it could help us to shorten this process, as well as we could utilize the available drugs.
Vaishali ma'am, you are not audible. Vaishali ma'am. Hello, Vaishali ma'am.
अभी हुआ था जब मैंने कॉल उठाया. अब ऑडिबल है?
Yes ma'am, now you are audible.
Okay, okay, thank you. So before going for drug repurposing, there are some important points which one need to be considered. That is, first of all, to make sure that the drug works in another indication or it is having or some other disease state. So it should be first confirmed. Then second thing, the disease for which it is going to be now newly indicated, so that biology or that disease should be different from the one which is previously reported. Here, the dose may be different. And third thing, it may interfere with current treatment regimen. So that also required to be monitored when we are adopting the drug repurposing strategy. So first of all, there are some drugs which we can consider for repurposing or the can be classified into generic, patented, or protected or failed drugs. And from these baskets, we can select the drug to undertake the to adopt this approach, like the generic drugs or the patented drugs, or the third category will be the failures from the pharmaceutical phases or the drugs which have been discontinued in various phases of development. So those can be selected for conducting the for using them or for reusing them for some another indication. So first of all, how to select a drug for repurposing? So for that, there are various methods. First of all, that we need to see that when the drug is administered among patients, so we need to observe the patient that what are the clinical changes he is reporting. So so that is going to help us to select a drug whether we can take it for drug repurposing or not. So the first point is to observe clinical symptoms among patients who are administered with this drug. Then mechanism of action and biology for the existing drugs. Then the third point is to study what are its off targets. So based on these three criteria, we can decide whether we can select this drug for further repurposing studies or not. And next, when we come for the drug repurposing methodology, so there are three phases when we go for this approach. So first of all, that is selection of drugs. So based on the clinical observations or off targets, we can identify the drug candidate. Then we can assess during the preclinical phase. And the third will be the evaluation of drug efficacy in clinical studies. Like we can go directly for phase 2 as safety is already established for these compounds. Then when we talk about this drug discovery, this drug repurposing process, so what different approaches we can apply. So for that purpose, we are having two methods. The first one is serendipitous, means where that is not in our hand, and it is going to be through random screening, so random discovery. While the systematic approaches are of our interest, where the chemist can put up his efforts, and the serendipitous will be mostly by the clinical persons. So when they will be observing the patient with who are under clinic, who are taking the medicine. So based on the clinical observations, they can identify whether the drug may be repurposed or not. While the systemic approach can be will be of use for the medicinal chemist as well as for the pharmacologist, so they can select the candidate and they can perform, they can adopt these approaches. So the first one is by computational methods, and the second one is experimental, where we can perform the in vivo or in vitro studies. So among computational, that is the computer-assisted methods, we can go for text mining approaches. The second one is bioinformatics, and the third one is artificial intelligence, which is getting very popular these days. And In the experimental, we can go for target-based high-throughput assays, animal studies, or drug-based phenotypic screening can be performed. And based on these methods, we can select the drug candidates. So the first approach, that is the text mining approach, which helps to identify the lead or the candidate for performing the for selecting it for some other indication. So this text mining approach, it is a simple literature-based methodology where various search engines, the literature search is performed using PubMed or Medline. And through the literature search, we can identify the candidates for various reported indications as well as its reported clinical observations. And based on that, the candidate may be selected. Then the second one is about bioinformatics. So in that, these computational methods are of help where we can perform chemical similarity of of the reported drug or the of the potential drug which is clinically proven, and its chemical similarity is checked with the other drugs, and based on that we can identify the drug for study. Molecular docking is again a very popular approach. Molecular dynamics, it has been adopted very well. So, and it helps to provide a real life interaction of drug receptor. Then comes network analysis, genome-wide association studies, and disease target association studies. So these computational approaches help us to identify the drug molecules. Then comes clinical analysis, which is a retrospective study where we need to carry on the post-marketing surveillance as well as the data which is obtained through clinical studies is properly analyzed, and based on those observations, the decisions can be made. Then in the second step, we perform the mechanistic assessment of the identified drug, where target-based assays can be performed for checking the binding of the candidate with the identified target, where we can select a variety of enzymes may be selected, and in addition to that, cell line assays may be performed, and or animal studies can also be carried out, where we can derive the dose, we can select the route of administration, different animal models may be selected, then the duration of drug exposure, various biomarkers which we can identify. So all that planning can be done for mechanistic assessment of the selected drug candidate. Then comes stage that is evaluation of efficacy in clinical trial phase 2. So in this case, this phase 2, that is first of all proof of concept. So we can check the efficacy of the drug in selected patient population, or we can perform the drug safety evaluation, as well as use of drug at series of doses of varying strength can be performed here. And assessment is generally based on specific clinical endpoint or marker. So it helps us to give a proper rationale behind selection of the drug candidate. It helps us to justify that. And phase 2B, that is about the efficacy. So among the selected group of patients, we can find out the drug efficacy. Now comes the various strategies which we can adopt for performing the drug repurposing methodology. So the approach here usually utilizes, like in the drug repurposing, we utilize the public databases to search the drug candidates, and data from various primary and translational research or from clinical trials that can, or we can even select the off-label uses of the drug. We can check the published human data information, which is available at various platforms. And for this purpose, we can even adopt artificial intelligence algorithms or other bioinformatic tools, and we can investigate between the interaction of the drug and the protein targets. So here in silico drug repurposing is, we can say it is a very powerful technique. And this in silico or bioinformatics is having various advantages, like it is cost effective as well as it is very speedy process. So it helps to reduce the time duration of all the experimental work. Secondly, there are, if we talk about this drug repurposing approaches, there are three approaches: computational, biological, and experimental. Or we can say we can adopt a mixed approach where we can use both computational as well as experimental parallelly. If we first of all, if we talk about the phenotypic screening, so in phenotypic screening, we need to it can be used to discover molecules and biologics, which are approved by the regulatory bodies, and these do not include pharmaceutical or biological information, and therefore their use is like very less to find out the mechanism of the drugs, and it mostly depend on serendipitous identification, where it is performed specifically for some specific disease and medicines. But if we talk about this phenotypic screening, so there is one advantage that it is having high chance of application for many diseases or many drugs. So phenotypic screening is one for all, we can say. So it is more adoptable. But if we talk about the traditional phenotype-based screening methods, so there we do not need prior knowledge, and the repositioned drugs are just serendipitously tested. So here the integrated knowledge and elucidated drug action mechanism, they have complex nature of modeling methods. Then comes after phenotypic screening, the next popular approach is target-based approach. When we talk about target-based, so here we need to have knowledge about the targets, like the protein structure, its 3D conformation. So all those should be already reported. And in addition to that, it also we need to have target about the disease condition, the drug, the adverse effects, in the regulatory approval, record of various clinical trials, then what are the biomarkers of that particular disease, what may be its potential targets, or what are the different disease pathways. So all this information should be reported. It should be available in the literature, and that need to be assessed. So this target-based methods, they help the researcher, or they will help us to quickly screen any number of drug molecules. So, but the only condition is that the structure should be known to us. So target drug repositioning, it will include, like, we can perform further in vitro or in vivo high throughput methods. We can do that screening for a particular identified protein or biomarker of that disease condition. Then in silico screening of drugs or compounds from drug libraries. So where we can talk about the ligand-based drug screening, that can be also performed. We can go for docking studies. So when we compare this target-based with the blinded method, so here target-based is having advantage that it improves the chances of discovery, and the identified drug can be directly related to the mechanism, disease mechanism. So it is having its own advantages, and it is the possibility of finding therapeutics is more with target-based approach. Then comes the knowledge-based methods, where these methods apply bioinformatics approach, and we can include available information of drug target networks, as well as chemical structures of targets and drugs. We can have the information provided from the clinical trials. We can have the information which is approved by FDA. Then signaling, yeah, metabolic pathways. So all those studies can be will be helpful here. And the information which we are getting from blinded and target-based method, it won't be sufficient to identify new mechanisms beyond the known targets. But knowledge-based methods, they help us put information into to predict unknown mechanism also. So this is the advantage of knowledge-based methods. So here we can go for novel drug targets. We can identify obscure drug similarities and new disease biomarkers. So those can be identified, or those can be discovered with the help of knowledge-based methods. Then comes the signature-based method, where these are in this signature-based method, gene signatures which are derived from the omics data with or without treatment to discover unknown off-target mechanisms is generally used. Here we can use genomic data also, and that can be the genomic data is widely available on the public databases like NCBI or the sequence data, CMAP connectivity is there. Then for various cancer cell line, the encyclopedias have been built. So using the artificial intelligence tools, this data can be assessed, and we can see the molecular level mechanisms, and we can identify the significant changes in the genes. Then the next one is pathway or network-based method. So in this method, we generally utilize genetic diseases data. So again, as well as this data are available on the public domains, and they help us to narrow down the general signaling networks instead of going for a huge number of proteins. So we can narrow down our search, and in this method we can use pathway analysis or network biology methods, and these are helpful to discover essential pathways as well as genetic, genomic, and this proteomic, metabolic data that can be used to find new targets for the repositioned drugs. So here we can like signaling mechanism for various conditions of cancers can be identified, because in case of subtype signaling mechanisms, it is hard to identify the existing cancer pathways or gene signatures. So here, in cases of cancer, these signature-based methods are very popular. Then there is targeted mechanism-based methods, where we can integrate treatment omics, or we can say the genetic data, and this data we can get from signaling pathway information and protein interaction to remove the unknown mechanisms of the action of drugs, and it will be helpful to discover drug action mechanism and to identify off-target or targeted pathways from this omics data. And here drug resistance, which is a very important issue in cancer therapy. So there we need to identify the reason behind it. First of all, like generally it is observed that the patient responds very well to the cancer therapy, but later in a short period of time they acquire resistance, and it successful drug treatment would be possible in such cases. So we need to identify the information about the drug mechanism, and for that purpose, it is better to find the drug target. So there are very few studies on targeted mechanism-based method where we can use the developed computational models to predict the drug effects and their pathways. So there are various difficulties in deriving these deriving these effective computational models to properly put up the mechanism. Then the next one is about the molecular docking approach. It is a versatile tool. It can be used to predict the geometry of the protein, or we can score the interaction of a protein with a small molecule or with the drug. Then this docking can be performed with a known drug into a large set of different target structures or a database of approved medicines into one intended target. We can select one target, and for that we can perform the docking study. And in this docking, if we talk about molecular docking, it is very convenient, and secondly, it is a fast method. We can screen large number of ligands and against a large number of targets also. And there are various sampling options. Then 3D structures are available. 3D structures are already available and the crystallographic data is already available. So that has simplified this process, and accurate scoring function and can be obtained through various recently developed computational tools. So with this different strategies, the drug repurposing can be easily adopted, or that can be carried out in a manner, we can say. So when we talk about cancer therapeutics, so there are various drugs or various like cancer. It is a very complicated. You can say it is having heterogeneous nature, and the increasing number of cancer cases all over the world, as well as its economic burden. So there is requirement of newer and suitable therapeutics. Secondly, as I discussed here about the rapid development of resistance with the available cancer therapeutics is also of major concern. And that's why in case of cancer therapeutics, large work is undergoing to repurpose the available candidates. And a very known example is the use of mustard gas in case of skin cancer. So these are some drugs which were repositioned for use in cancer, like cimetidine, which is already reported as a H2 receptor antagonist. So preclinical studies are undergoing for its effect in lung adenocarcinoma. Then clomiphene citrate, which is an estrogen agonist for glioblastoma. Then imipramine for breast cancer, which was the serotonin receptor, which acts on serotonin receptor. Then itraconazole, which is an antifungal agent. It is for non-small cell lung cancer. So these are some. Then aspirin, which is an NSAID. It is in phase 3 trial for its efficacy in colon cancer. So these are some examples which have been reported in the literature. And when we see that these are very few. If you will go through the literature, there are more than 50, around 100 compounds are being undergoing in the development phase in various stages of clinical phases. for evaluation in different types of cancer. So, if we see about the application of drug repurposing, so there are wider applications, right from cancer to COVID, Alzheimer's, then diabetes, various heart disorders, cardiovascular disorders. So, during their application, some challenges have been observed. So, which includes like aggregation and search and analysis of the vast amount of previously captured data to make efficient and effective use against a new indication. So it is a challenging task to search all the data, to collect it, to analyze it, and to derive some conclusions. So it is a very challenging task. Secondly, we have to go for safety. So safety concern is a major thing. Then comes patient consideration. So if we talk about the knowledge of the domain and the claim which is given by the patient, it may be different. And so patient consideration is an important point, or it is an important limitation to be associated with this approach. Then comes the regulatory considerations. So if the company is marketing some exclusive that drug which is designated as an indication for 7 to 10 years, so then it comes a limitation with that particular drug. FDA offers only 3 years of market exclusively for new use of a previously marketed drug, and there is not enough time for companies to retrieve the investment. So for pharma industry, it is going to be a major burden then. Then the third hurdle is about the organizational hurdle. For example, if the pharmaceutical company is not interested, as the repurposed area is not in company's disease area, so the company won't like to put its interest on development of that particular drug. Then generic drug research area is discontinued, so no R&D support will be there, and there is no sharing of compounds which are still in live development outside of pharma. So because of that, many candidates which are in the development phase, those are not getting proper financial support. So we can say that in the conclusion, or I will like to conclude here that apart from the numerous challenges associated with the or we can say with the drug repurposing and the traditional drug discovery, like in traditional drug discovery, there are various stages. First of all, discovery, then preclinical, safety, then clinical research, then FDA review, FDA post-market safety monitoring. While in case of this drug repurposing, there are only 4 stages. First is compound identification, then its acquisition, then its development, and then FDA post-market safety monitoring. So because of that, it is less time consuming also. The investment is less if we talk about the traditional drug discovery, and here failure also lesser as compared to the traditional drug discovery. And the clinical efficacy, if we talk about, or the safety profile, it is already established for the drug. So because of that, this drug repurposing methodology is going to be always a win-win situation. And if we talk about the other prospects of application of drug repurposing strategy, so we need to find out like a new of use patient for an old generic drug should be utilized. Then we can limit of label use of a generic drug by developing unique formulation and dosage. We can develop some financial, this is the part of the regulatory or the research organizations that they can provide funding for to encourage generic manufacturers to participate in drug repurposing, collaborate with larger pharmaceutical companies, academia, or smaller companies to offer better access to well-characterized compounds and collaborate for preclinical experiments and phase 2 proof of concept trials. Funding opportunities may be made available for specifically for repurposed drugs in collaboration with industry. So that will help us to give more focus on the available identified drugs for their use some other indications. So these are some publications from my group about drug repurposing. The first one is about cancer therapeutics, and in the second one that is about about the COVID polymerase inhibitors, where the favipiravir has been used using computational studies for its anti-polymerase activity. So thank you to the organizers of this international conference, the chairperson as well as the patrons. And I will also like to acknowledge my parent institute, Chaudhary Charan Singh University. So thank you so much, sir.
Thank you, ma'am, for valuable information on drug repurposing and challenges. I am requesting our panelists, Professor Bharadwaj sir, Vicky Kapoor sir, Tejveer sir, to start the question and answer session. Over to Bharadwaj sir.
Thank you, Dr. Vishali, for your very encouraging, informative talk, and that to pertain to the repurposing of the existing drugs.
Thank you so much, sir.
Welcome. So, what we have the tools right now, that is the docking studies or even molecular modeling, 3D structure of the targets, receptors, which can be very helpful in identifying the receptors for the existing drug as well as the new entities also, isn't it? So it's very good approach. I would like to know that with this approach, strategy in finding the repurposing of the existing drugs, what are the drugs which have been, of course, you have already listed some of these, but what are in the maybe most potential, which have not yet been listed in this drug, they are under studies. Do you have some data regarding that also? Other than
yes, sir. Sir, in addition to that, like if we talk, there are around, when I identified a list, there were around 150 drug candidates which have been like they have been under studies in preclinical as well as in clinical phases, and most of them, like I will say around 60 to 70% are for the treatment of cancer. So and in which even the thiotepa, which is an immunosuppressant, which is a very reported drug. Then there is like drugs which are we can say for even Hodgkin's lymphoma. So that is not only for the other disease, but among cancer also those are being repurposed. Like the one which was vinblastine, which is for, I think, Hodgkin's lymphoma. So that is now being evaluated for other type of cancers. Then, in addition to that, there are various antifungal drugs also, which I observed in that listing. So the list is vast. So,
in fact, I would like to say that the role of pharmacist as keen observer with respect to the medicines which are being given to the patients, he or she will be the best observer to tell us about the new, unexpected, unwanted, unreported actions of the existing drugs which are being used clinically in the hospitals or in the multi-specialty hospitals also, you see. So that will give us again another idea, even if the leads are identified, these can be further developed in the drugs. You see. Thank you very much.
Thank
so
you
over
so
to
much.
Professor V.K. Kapoor, sir.
Namaskar, sir.
Namaskar, Dr. Vaishali. I would like to repeat what Dr. Bhardwaj has said. Thank you for very illuminating talk on the very hot topic on the drug discovery. As you have already mentioned, to discover a new drug entity is a very time-consuming and very costly affair. And you have given a new direction to the drug discovery. My observation is that if you take the account of the already repurposed drug, they have all amount of the keen observations, as Dr. Bhardwaj has said it. I mean, it will be only possible to find out if the patients are observed in the hospitals and what is their behavior. I tell you a case of MAO inhibitors as a new class of antidepressants, right?
Yes, sir.
In a hospital, there was a patient who was admitted with TB, and already a drug was given to him, the isoniazid. Then another version of isoniazid, where methyl group was substituted by isopropyl group, that was iproniazid, was given to him. I mean, it was observed by the clinicians that the patient who was earlier was very dull and depressed was now very happy, and it was very apparent that he is in an accelerating mood. And they certainly found that it was not because of his improvement of TB conditions, but then they discovered that it was the effect of iproniazid, the new anti-tubercular drug, which made the patient happier. And when they studied it, they found that it is a MAO inhibitor. So I think it is the keen observations and pharmacovigilance which is necessary for discovering a new repurposed drug. Similarly, we have the case of sildenafil, which was initially, made as a beta blocker or as an anti-hypertensive drug, and it resulted in a drug for erectile dysfunction. So, in my view, it is the keen observation which should be made, and the hospital should be advised to observe the behavior of the patient who are on the long-term strategy of therapeutic. Anyway, thank you very much, and knowing you,
sir.
Our presentation. Thank you.
Thank you so much, sir. Thank you, sir. I think it is the time where not only we need to collaborate outside the pharma, but within pharma also. If the chemist, the pharmacologist, they collaborate with the clinicians, we will be able to help the society.
Yeah. Professor Tejveer Singh is a Emeritus Professor with Baddi University. Will talk to you.
Namaskar, sir. Ji, sir. Namaskar, sir.
Professor Vaishali, I have a doubt. You mentioned that the aspirin can be used as a drug for colon cancer.
Yes, sir.
Does it really reach to colon?
Yes, sir. Like the in vitro studies they are carrying out, and I think the formulation, if I recall the paper where I read about that, so they are preparing a formulation of it. some for colon directed delivery, they are preparing the formulation so that it will reach up to the colon. So in that way, like I think they are not on we can say about a new discovery, but it is about the changing the formulation. So they are working on formulation development of the
I
aspirin.
think for that the approach must be it is pH dependent.
Yes, sir.
Drug delivery system.
Yes, sir. They are working on that.
Most likely the pH of the formulation must be giving ultimate result that it's being released at the colon where the pH is.
Yes, sir.
Because
So
there are
when
some...
I read about it in the literature, it is not only about the identifying for a new indication, but even developing a suitable formulation is also it can be also adopted.
Yeah. Professor Vaishali, I asked this question because me and Professor Bhardwaj have been associated with colon cancer, and we developed a drug which really has a linker to reach to the colon cancer.
Yes, sir. That's really nice to know, sir.
Yeah, over to Vishal. Dr. Ravinesh also, please.
Thank you, Professor
Namaskar,
Vaishali.
sir.
Namaskar, ma'am.
Congratulations, first of all, to you.
Said, ma'am, thank you. It's only because of eminent speakers like
Teamwork.
you.
No, so it is teamwork. Bataiye, sir.
Ma'am, there is a question from some of our participants and the listeners. They want to know, does dose also plays important role in repurposing of drug?
Partially, it is not a major, I will say, a major parameter. If like dose, it is related to potency of the drug. So instead of developing a formulation, a different formulation will play a significant role. But through the things which I have came across, so dose won't be an important parameter. It is, I will say, it is related to potency of the drug, right? I think this will be the better answer.
Yes, ma'am, because few drugs means depending of the doses, their therapeutic action used to be changed, like in sedative, hypnotics, and all. Similarly, few other medicines are also available. So, thank you, ma'am. Thank you so much.
Thank you, sir.
Over to Madam Salni.
Thank you, Mishra sir. Thank you to the panelist. Is there any question from attendees? Attendees, kindly raise your hand.
Yes, one of the attendees has asked what drug Professor V.K. Kapoor said, which is a beta blocker but was repurposed for erectile dysfunction. So this one is sildenafil citrate, and trade name is your like Viagra and other similar kind of formulations. Those are available in the market. Yes, Madam Shalini. Or
yes, sir. Yes. Thank you, Vaishali ma'am, for sparing your valuable time with us.
Thank you so much, Dr. Shalini.
Thank you. Thank you so much, ma'am. My dear participants, I'm taking the privilege to introduce our chief guest, Dr. Montu Patel, President, Pharmacy Council of India. He has just conveyed his best of wishes because he was unable to join due to some unavoidable reasons. but I am feeling privileged to inform you all that our guest of honor, Professor Roop Krishan Khar, has joined with us. Professor Roop Krishan Khar is Director General, Anangpuriya Group of Educational Institutions, Faridabad. We welcome you, sir. Roop, sir, are you with us? Roop, sir. Roop, sir. I'm requesting the participants to please wait for a few minutes because Roop sir is about to join. Roop sir, are you with us?
Hello, Shalini.
Yes, sir.
Hello, Dr. Roop Khar's
has joined.
No, muted. You see, tell the technical persons.
Okay, sir. Okay.
Yeah. And read the message of President of India, President of Pharmacy Council of India also.
Right, sir. Right.
Okay. We got it.
Dear participants, there is a message from our guest of honor. Sorry, there is a message from our chief guest, Dr. Montu Patel, President Pharmacy Council of India. And he conveys his best of wishes because he was unable to join today due to some unavoidable reason. But I am feeling privileged to inform you all that our guest of honor, Professor Roop Krishan Khar, has joined with us. Professor Roop Krishan Khar is Director General, Anangpuriya Group of Educational Institutions, Faridabad. We welcome you, sir. Sir, am I audible to you, Roop sir?
उनका म्यूट है मेरे ख्याल से. We are unable to hear him. Audio.
Sir, I'm letting him unmuted.
Okay.
Roop sir, kindly unmute yourself. He is again requesting us.
Yeah, hello?
Hello, Roop sir.
Oh, thank God it has come.
Good morning, Daso.
Very good morning. आपने एक्चुअली आप लोगों ने म्यूट कर रखा था मेरी तरफ से तो I know something was wrong, it's always like that. Yeah.
Thank you so much.
So, sir, I'm requesting you to please share your blessings on us. Over to Khar.
Thank you. Without wasting any time, let me congratulate all of you. And there are very eminent scientists and academicians sitting out there. Dr. Professor Tilak Bardwaj is there, Dr. V.K. Kapoor, and also the coordinator and the motivator and the main initiator of this conference, Dr. Ravnish Sharma is as well. Mishra, I'm sorry, Ravnish Mishra. Thank you very much for giving me this proud privilege and honor to be a part of this international online conference on drug discovery and lead optimization. Certainly, it provides an excellent platform of interaction and exchange of knowledge between different experts and specialists from different domains. School of Pharmacy and Emerging Sciences has evolved into an institution of excellence over the years, I think, with the leadership and with the brilliance of academic staff and research staff out there who have brought this institution to this stage and working and under the initiation of Baddi University. Well, I will not talk hardcore R&D. I have listened to a very good lecture by Dr. Vaishali, and there was a very good discussion also followed after that. I will give you a very general outlook of the present trends. Particularly, I will talk about the current trends. Secondly, I will tell about the evolution of industrial progress from 1.0 and 4.0 and what the pharmaceutical scientists need to do in order to be in tune and to be in so that we can be in the process. And thirdly, I'll also tell you and try to impress upon you what are the various Government of India initiatives in pharma sector, lest we all do not be behind. We have to all be a part and parcel of what is going on in different sectors, whether it's pharma industry, whether it's Government of India, and what as well as the progress at the global level. Well, we all know, madam, and all of you, ladies and gentlemen, the pharmaceutical sciences by its nature and its intrinsic content of various courses is a wonderful interdisciplinary platform, and it creates a good space for innovation and new discoveries to happen. We all need to catch up with the contemporary needs and future challenges from time to time. And this is rightly in tune with the theme of the international conference. You are doing it now. It should focus on the future trends for at least next 10 years, 2023 onwards. We all know that academia is a fountainhead and a starting point of all the disciplines. We need to sow the right seeds at the appropriate time and to grow into a strong and a vibrant profession to compete our own needs at the national level and equally at the global level. In tune with the theme, I have chosen some topics, some trends, like say, which are the topics of concern, which we should all to know, the contemporary trends for next 10 years, what I feel in pharmaceutical sciences and drug discovery. First and the foremost, which the previous speaker has also mentioned, the computational pharmaceutics and pharmaceutical sciences. It is a new paradigm for drug discovery and drug delivery. Then secondly, the role of three-dimensional printing in healthcare and medicine. It will find a very strong place in the times to come. Thirdly, the precision medicine, what we call as academic dreaming, which is becoming a clinical reality. We need to find out how we can progress and find out our place in the whole game of science and technology. The computational pharmaceutics, as we all know, in the recent decades, drug delivery science has become very critical as it is considered as one of the important drivers in terms of deliverability. We have been given concerns of the quality, the safety, and the efficacy, but the new drugs and the new paradigm which is taking place, we are concerned about the deliverability of the drug. It's a very important place in the current research. That is why it takes a longer time. Otherwise, it relies on these, earlier sort of strategies. We had a traditional trial and error methods. It involves higher cost and less productivity of new molecular entities, NMEs, what we call. But with the exponential growth of computing and algorithms in recent years, a new discipline named as computational pharmaceutics has emerged, which integrates such big data, artificial intelligence, and multi-scale modeling techniques into pharmaceutics and pharmaceutical sciences and drug discovery. It has made a big paradigm shift in the drug delivery science and other sectors. Computational pharmaceutics will involve and provide a multi-scale lenses to pharmaceutical scientists. It will reveal physical, chemical, and data-driven details, which will range from starting from pre-formulation, formulation screening, in vivo prediction in the human body, precision medicine for use of patients, which will include artificial intelligence, machine learning and algorithms, molecular modeling, mathematical modeling, process simulation, and of course, very important and the most important, physiologically based pharmacokinetic systems. So they'll be focused also on, in the next generation and now on, focus on patient-related outcomes, what we call as PROs. That's why physiologically based pharmacokinetic systems would be more important than the traditional pharmacokinetics. Will not be depending on only the blood levels and the urine levels and all those parameters. PROs can be used for drug approval process, certainly in the times to come. There will be a direct relationship between the doctor and the patient through artificial intelligence, through Internet of Things, what we call as most important. Let me tell you some of the latest discoveries in some drugs which have come up recently, which will mark a very important place in the drug discovery process. As many of you must be knowing, Epidiolex, an important drug which was derived from marijuana for pain management. So this, again, a paradigm shift has taken place. Drugs from marijuana, we call it as cannabidiol. Then another drug which I want to highlight is DS8201. It's a monoclonal antibody conjugated drug. It's a future blockbuster for breast cancer after Herceptin. Similarly, another approach based on the technology is from Aprecia Pharmaceuticals, Zipidose technology using formulation technology with 3D printing to create a high-dose medication in a rapidly disintegrating form, Spritam, SPRITAM, for epilepsy. It is made by layer technology without traditional compression and modeling techniques which we generally know. Recently, you must have known, hardly I think a month back, there has been a new discovery of an antibiotic by an Indian pharma industry. It's an antibiotic which is a very strong beta-lactamase inhibitor and a new antibiotic approved by none other than FDA from India. And it is much more efficacious and more, useful than the currently available clavulanic acid, which is used in combination with amoxicillin in the products like Augmentin and others. Similarly, there are many other drugs also which have made a paradigm. Like say we have Debut, which was in March 23. It is an oral solution of trofinetide for Rett syndrome, a rare metabolic syndrome affecting brain development. Then Zavipret, it is a drug by Pfizer. It is a calcitonin gene-related peptide for acute management of migraine. Again, an important drug to be. It is in a nasal delivery system. The story is endless. Ladies and gentlemen, I only wanted to set some leads to illustrate that a lot is happening in the pharma industry, with the academia. This convention is going to open new vistas and identify some gaps to be filled up and which will be carried forward. Certainly, I request all the delegates of this conference, the young scientists, they should take new messages, take heavy messages from academia and pharma industry so that they can create synergy between for better outcomes for future. Well, you see, academia alone cannot do anything. We cannot live in one compartment model of I am a chemist, I am a pharmaceutical scientist. We have to work together for drug discovery process. It is a multi-compartment, it is a multi-speciality role which we have to play. As you all know, you see, the present generation is really very lucky. If we go to the industrial revolutions which have taken place, transition from what we call as 1.0 to Industry 4.0, we have made transition and progress through precisely 4 stages over the years from 18th century onwards. It has been a transition from the Industry 1.0 with the start of process of mechanization only in the late 18th century, which was followed by Industry 2.0 with the use of electrical energy and mass production from 1870 onwards. The stage of Industry 3.0 was the time for automation. It was the time for computers. It was the time for integration with electronics from mid-20th century, precisely 1970 onwards. You all, the present generation out here, the new scientists and the young scientists who are sitting here, you are very lucky to have surpassed all these 3 initial founding steps, which has made a very good, strong platform for all of you for a new drug discovery process. You are exposed to the vibrant, dynamic, and a faster Industry 4.0 in the current times. It comprises of cyber physical systems, what I told, Internet of Things, Internet of different networks, and perhaps, and certainly, the full-fledged digitalization. You have to get tuned to this new setup. I think our younger generation is tuned to this, and they will certainly make a lot of progress in the time to come. You have to be all ready for this change, ladies and gentlemen, and it's a continuous learning. It's a continuous learning and upskilling, lest you become redundant and आवाज आ रही है?
Yes, sir. हाँ जी, आ रही है आवाज.
आ रही है. Okay. I carry on. So, to continue very briefly, let me congratulate the academic institutions. In the recent data, I will tell you, out of 215 drugs which were discovered in last about 20 years, 32 were from academic institutions. And that means we cannot, ignore the academia, the institutions like you, the universities like you, which are taking a very good leading role in propagating the knowledge and in being able to set up such platforms which you are doing today, and having set a process so that people can know what they should do, how they should plan. And let me tell you, ladies and gentlemen, the Indian pharma industry is a sunrise industry after IT industry. The COVID pandemic period has put the spotlight on pharma sector as a vital component of self-reliance and positive growth in the economy. We are happy that Government of India has put in various schemes and efforts for the development of pharma industry under Atmanirbhar Bharat scheme, like establishment of bulk drug and medical device parks from 21st of July 2020, and it has already given these various stimulation of investment in these sectors more than 3,000 crores of rupees. We have to persevere and find out. There are various production-linked incentives for manufacturers, for the manufacture of API, for the manufacture of medical devices. Already more than 200 crores have been released on in February 2023. And if you look at the recent budget 2024, the pharma sector has got 90,000 crores. It is as compared to about 80,000 crores in the last year's budget. Similarly, there are many schemes for academicians and all of us, for promotion of R&D in pharma sector in terms of tax breaks and financial support. I just wanted to highlight that Government of India has taken the cognizance and the importance of the pharma industry, and it is our turn and the responsibility as to how we can make best use of such proactive and positive measures. Academia industry interaction is an effort in this direction. We cannot discover drugs without integrating, without interacting with the academia industry as such. Well, it's something like, I recently read a paper, it is just like a DNA molecule with a double strand, one represented by the academia on the one side and industry on the other side. Only then the drug discovery can happen. And government funding and funding by the pharma industry, in case it is possible, is the central core in the DNA strand. Well, the academia and the industry have, of course, substantial differences with each other. The goals are same. The pharma industry lives and breathes on transformational research and innovations for developing new products. They switch over to a new project if there is no returns, if it does not involve commerce, if it does not involve and give you business. While as the academia has generally no relation to the output. As a matter of fact, by and large, they are not, in fact, focused on the outcomes. They have to perhaps change as far as the outcome is concerned about the research. And the effort of this conference is that only. The academia only maybe wanting to publish very good papers with high impact factors and everything, get promotions and all those things, all the knowledge, I think most of the knowledge remains on the shelf, 80%, 90%. We have to find out ways how to be able to do a piece of work which has a tangible, which has a output, which has an outcome. And that is only possible if we make very, very proactive efforts. And I think Government of India is providing now some very, very important models, what we call as the PPP models, a public-private partnership is possible. I think academia should be obligated to work with the industry if they are funded by Government of India or even by pharmaceutical industries in order to pursue the principle of bench to the bed. Otherwise, no point, spending crores and crores of rupees, doing a lot of research and all that without any outcome after even 10 years or 20 years. There are many instances that the data generated, there's a very important observation I want to make, in many instances, the data generated in academic institutions is irreproducible. It is not reliable. This is a great concern. We call it laptop research, LAPTOP, laptop research. We are interested in laptop research, LABTOP. No, it was, I am not saying of my own. There is a publication I read recently from the Bayer company, BAYER, Bayer company, one of the top companies at the global level. They published a data wherein they said that 2/3 of the targets and the initial basic research conducted in various academic institutions could not be replicated. That's a very, very important observation. We need to find out solution to this, ladies and gentlemen. We need to develop clear communication channels between academic institutions and the industry. And we have to develop joint research projects, encourage joint research projects. It was earlier done under DSIR. Even I had a couple of such projects with the industry, which could result in some IPR and intellectual property rights from time to time. Currently, I think you must be knowing, I think students must be knowing, there is what we have DSIR dash CRTDH schemes. It is known as Common Research and Technology Development Hubs. There are already 18 CRTDH centers in our country. And I think there are also RTDH, and IIT Roorkee has that project. There was one CRTDH project with Dipsaru University and many, many other. I'm just giving you an exposure. It is possible. Then BIRAC was another initiative for contract research schemes. I think it's doing well, and many, many good research grants with outcome-based research related to drug discovery are happening. Then there are similarly incubation grants. So all government is helping us, for innovative ideas, for initial seed money. See, if you have some idea, if you have some IPR, if you have something which is useful, there's no dearth of funds. No dearth of funds. And many startups have come up like that successfully. There's one more scheme. I'll just end up in 2 minutes. PACE, PACE, promoting academic research, conversion to enterprise. That is what we are calling as translational research. There are many islands of success and innovations in our country. I am not pessimistic that we are not doing anything. There are many things which are certainly happening, and we are very, very happy about it. And we should all work together in a way so that there is some useful outcome, useful output, which brings laurels to the academic institutions and which brings strength to our country, to our research, and to our pharma industry, which is certainly going to rise days in and days out. Thank you very much, ladies and gentlemen. I'm sorry there were some initial IT problems. I think that was done. Thank you very much, all of you, the eminent scientists and young delegates, and all of you. Have a nice, wonderful day. Thank you very much.
I may please request Honorable Bhardwaj sir to share words of wisdom with our participants. Please, sir. Over to you.
Thank you, Shalini. Thank you, Professor Roorkee Khar, for your very, very encouraging directional areas of research, funds from where we can get the things, and more specifically, the industry-oriented projects instead of theoretical or basic science, which may or may not lead to the funding, and we cannot be Atmanirbhar on account of that. As you said that we are already having third position globally with respect to formulations, bulk drugs, APIs, or the generic drugs. And out of that also, it's a matter of pride for us, those who are in the Baddi area, Baddi, Brotiwala, Nalagad area, that 1/3 of Indian drugs which are exported or API or generic drugs, they are produced in our neighborhoods. And in addition, it's again a matter of pride for us that the medical diagnostics park is again coming up at Nalagad, and the bulk drug park has been sanctioned to Himachal, which is coming up near Una, Hiroli. This is around 12 kilometers from Hiroli, Una, and that's another area of pharmaceutical hub, bulk drug, which is coming up, so that we are not to be dependent on China for our raw materials, for doing research and other things also. So thank you very much, Dr. Sir, for your very informative, encouraging, motivational talk. Thanks a lot. Over to
Thank you, sir. Thank you.
Professor P.K. Kapoor. He would like to share his experiences, and he would like to talk to you also.
My pleasure, sir. My pleasure.
Hello,
Welcome, sir.
Professor R. My salutations to you. I regard you as my great revered friend. So I will talk in a different tone. First of all, you are a true pharmacist in the sense that unlike the other stalwarts of pharmacy profession, you are the only one who have never refused the request of other institutions either to participate in their activities or to deliver a talk or to be a guest of honor or to be a chief guest. That is a great quality in you which I want to express myself to you, and that really encourages all the pharmaceutical institution in India and is very helpful. For that, my personal salutations and salutations on behalf of the rest of the pharmacy fraternity in India. Thank you. Dr. Har, I'm regarding the drug discovery part. Pardon?
Hello.
Hello.
Vaishali ma'am, kindly unmute yourself.
Dr. Har, as far as the talk on pharmacy or pharmacy research is concerned, like you, I'm a very God-fearing man. I know you are a very religious and God-fearing, so am I. There is a quotation from our Shastra. Matulya yasya Govinda, Pita yasya Dhananjaya, sa api kalvash prapto, kalo hui dhruti kraman. It means that his maternal uncle, Lord Krishna, was there. His father was a great warrior, Arjuna. Still, Abhimanyu got killed. So, Kalo hi Dhruti Kraman. It is the destiny which is the greatest of all. That is to interpret in terms of drug discoveries. Dr. Khar and other ladies and gentlemen, there have been 3 melodies in the world which have, very calamities to the population. First is the infection. Till 1941, the infections in the human being could not be treated properly, so that there was a lot of fatalities. Second was anti-cancer, I mean cancer. It is a disease which has not been cured so far. All the research is going on to find a treatment for cancer, and we have failed. The third is a socio-economic, aberration, and that is the erectile dysfunction, which has destroyed many of the marriages and has been a social disaster. For all these great calamities which human race has faced, the drug discovered are from the divinity. Another name of divinity is serendipity. Drug discoveries by chance. Antibiotic penicillin was discovered by chance, a God gift. Anti-cancer drugs were discovered by chance, by your the ship mustard. That was also discovered. The anti-metabolites, anti-cancer agents, they were discovered by the serendipitous discovery of nitrogen mustard. And third is the cure for the social evil had come also serendipity, sildenafil citrate. So that is my observation, that all the pharmacists should believe in God Almighty and pray that all the diseases, they are cured by the blessings of Almighty. Thank you very much, Dr. Khar, for your very motivating and very informative and very knowledgeable address which you have delivered. Thank you, sir.
Thank you, sir. Thank you very much. My privilege and honor. Thank you. Thank you.
Sir, Professor Tejveer, sir, may I please request Tejveer, sir, to please share your words.
After Professor Kapoor, I do not have anything to share. The only thing I can say, thank you, Professor Khar.
Thank you, Tejveer, sir. Thank you, Bhardwaj, sir. Thank you, Vicky Kapoor, sir. Ravinesh, sir, would you like to share some words with Roop, sir?
Yes, thank you. I always feel glad to listen our teacher, Dr. Khar, when I was at Jamia Hamdard. We are like a fan of Professor R.K. Khar, sir. So it's kind of memorizing our days. So thank you, sir, for sparing your valuable time and delivering such a message to all the participants across the world. Thank you, sir.
Thank you, dear. Thank you. Thank you. All the best to you. Keep it up. Keep it up. Keep it up.
Thank you, Mr. Sir. And thank you, Khar, sir. A very heartful thank you for being our guest of honor. Your presence has added immense value and prestige to this conference, and your insightful remarks truly enthusiast all of us. Thank you, sir. Dear participants, our next speaker, Dr. Alka Ahuja, has joined, and I'm taking the opportunity to introduce Dr. Alka Ahuja. Alka ma'am, are you with us? Alka Ahuja ma'am? Alka ma'am, kindly unmute yourself.
Good morning, everyone. Are you able to hear me?
Yes, ma'am.
Okay.
You're audible.
First of all, I would like to thank the organizing team for inviting me today for this conference. And it was actually my pleasure to listen to Professor Khar, who actually happened to be my guide for my PhD thesis also. And I feel so honored to see my own teachers, Professor Kapoor and Professor Bhardwaj, who are leading your institute with such great leadership. So I'm sure with their leadership, your institute will really grow very high. And thank you so much, my guides, my mentors, and my teachers for grooming me, and so that I'm able to present my work, which I have actually gained under your guidance. Thank you. Thank you, all my mentors and guides, please. I'll start with my presentation now. I would just like to know if you can see my presentation. Can somebody let me know?
Raman sir, Panchal sir, are you with us?
Ji, madam.
Sir, please help ma'am to share her presentation.
It's not that. Okay, fine. Just let me know if you can see my screen, because I have shared my presentation.
Ma'am, presentation is not visible. Raman sir, please help ma'am.
Hello, madam.
Yeah.
I'm Raman, and top right में option होगा share का. Top right.
Yeah. I know that. So.
जी. And click on that screen.
Just open it.
क्यों click भी करना पड़ता ना?
Enable editing. Yeah, because I have been...
Hello, हाँ जी मैडम.
मैं ये कह रहा था अलका जी, इसमें कभी प्रॉब्लम आती है. आप ऐसे करो, साइन आउट करो तो फिर साइन इन करो.
Sir, actually, I have been able to share also, and so I don't know what the issue is.
Not visible here.
क्या sir?
तो आपकी presentation visible नहीं आ रही है.
इस पे क्लिक करना पड़ता है.
It's not being shared.
मैडम हेलो मैडम हेलो हेलो
yes.
मैडम टॉप में जो बटन दिख रहा है शेयर का उस पर क्लिक करो देन उसके बाद जो
उसी पे ही कर रही हूँ।
देन उसके बाद मैम उसके बाद जो स्क्रीन आ रही है उसके ऊपर क्लिक करो स्क्रीन पर क्लिक करो स्क्रीन वाले ऑप्शन पर क्लिक करो
Committee, College of Pharmacy, National University of Science and Technology, Muscat. She is a gold medalist with 39 years of experience in teaching and research, taught at Jamia Hamdard, India, for 21 years before joining the National University in Oman. She has worked as a chair and member of various academic and research committees for more than 35 years. She has been invited as a speaker at national and international forums. Research work has been presented in various international conferences held in India, Oman, and abroad. She is a recipient of Indian Drug Manufacturers Association GP Nayyar Award. She has also been awarded as a Career Award for Young Teachers by All India Council for Technical Education. She has worked on 20 major research projects financed and supported by ICMR, UGC, DST, AICTE, and Moheri, Oman. Over to Alka ma'am. Alka ma'am, have you joined with us? Please call her.
can you present my presentation? I've sent it to Shalini.
Yes, ma'am. Now it is visible. Your screen is visible to us. Your presentations is visible to us, ma'am.
It's visible to you?
Yes, ma'am. Pharmacokinetic modeling in drug development.
Okay, thank you so much. And first of all, I would like to apologize for this technical error. And this as Professor Khar, he already mentioned about the importance of pharmacokinetics in drug development. So I think this topic will be appropriate for our students as well as my colleagues to know how pharmacokinetic modeling is very important when we are developing the new drugs. Now, the outline of my presentation is just to know what is pharmacokinetic modeling and what are its applications in drug development and what are the important highlights. So as all of us know that drug research, it encompasses the disciplines which are all united by a common goal. And what is the common goal? It is the development of the novel therapeutic agents. Professor Khar, he highlighted the importance of so many new agents which are developed by our own pharmaceutical companies in India. So, and we really feel proud that, but it is not the work of one person. So many people have joined their hands in these development of the novel therapeutic agents. Now, when we search for the new drugs, it involves two stages. One is the discovery and one is the development. So I would be focusing on the development part today, and which is basically, it is focused on evaluation of the toxicity and efficacy of the new drug candidates. Now, when we talk about the drug development, I'm sure all of us know that to bring a new drug to the market, it takes about 10 to 5 years of the hard work. But as we saw during COVID time, that how even the Indian pharma company and the other companies like AstraZeneca and then Pfizer, and there were so many other companies, they brought these vaccines at a very, very short time. So for these drugs which are needed in the emergency process or their first available treatment, this approval process and drug development can be expedited. So, and or in the cases where we see that there is going to be a significant benefit over the existing drugs, then this time of 10 to 15 years can be shortened. Now, if you look at this slide, you can see that drug development process, it talks about this journey, how drugs, they move from 5,000 to 20,000 compounds, and then we narrow it down to 250. And the clinical research step only takes about 5 molecules. And finally, out of where we start from 5,000 to 20,000, we reach only 1 molecule, which is the approved drug. Because before that, we have to do a lot of safety studies, efficacy studies, lots of steps are required. But if you look at this picture, what is the major reason for failure in development of the formulations? It is basically because of poor biopharmaceutical properties, which account for almost 41% of the preparations which fail only because of this. That is why I find that this topic is of extreme importance to all these people who are involved in research, because they need to study the proper ADME principles, the absorption, distribution, metabolism, and excretion of the drugs. And but what is pharmacokinetics? It is basically quantification of the ADME processes. And what are the reasons when we talk about pharmacokinetics and we talk about drug failure? What are the basic reasons for that? What we really need to study when we are in the drug development stage? So one is the poor bioavailability, where the drugs, they might not have the proper solubility, or they might have undergone, or they go through first pass metabolism, or the drug might exhibit very high clearance and a very short half-life, or there might be unanticipated drug interactions, which are normally seen in phase 2B or in 3 trials. And all these things, they result in variable pharmacokinetic properties, and they result in the undesirable effects. So that is why we need to focus, and we need to take this bioavailability problems or the total pharmacokinetic properties very seriously. And as Professor Carr already mentioned about its approval from different FDA and the regulatory bodies, so already now we are getting so many patents from India also. But you can see that there are so many bodies like Food, Drug and Administration and other regulatory bodies worldwide. When they approve the drugs, what do they study? They see that the drug is safe, it is efficacious, and also it is ready, like he mentioned about from bench to bedside, because, and also he mentioned about from laptop to labs. So this is what we really need to see. But we have to develop, also reach the next level, not only till lab, but we also have to see that from bench to bedside, the drug should be available. And if it has to be available at the bedside, then we have to make sure that drug is safe, efficacious, and it is ready for use. So these are some of the main agencies which take care of the regulations and which take care of the approval worldwide. Like we have US FDA, that is US Food and Drug Administration. We also have European Federation of Pharmaceutical Industries and Association, European Medicines Agency, International Federation of Pharmaceutical Manufacturers and Associations. So these are some of them. Now, if you look at this picture, it is self-explanatory that if we are actually not able to achieve this minimum effective concentration, that means our entire step of the drug availability is of no use. And if we are exceeding this concentration, which is beyond the therapeutic window, what will happen? The drugs, they will cause toxicity. So we have to make sure that when we are administering the drug should be in the therapeutic window for its entire period of its stay in the body. Now, this picture, it is a very simple curve which talks about the processes and the how when we are developing the drug, we have to see that how much drug is reaching the blood over a period of time. And now we have to see that absorption step, which is very important. And there is absorption step, which is involved for all the formulations except for the IV routes, because there we are directly putting the drug in the blood circulation. And after we reach the Cmax, then we see that the drug's concentration, it starts declining. And how much time it takes, it all depends upon the half-life. That is what we say, that time which it takes to come back to its half of its original concentration. So if the drug has a very short half-life, we have to increase the frequency of the administration. So that is how half-life calculation becomes very important in the drug development process. Now, this picture tells us about the various steps which are involved in the journey of drug, that how when we give the drug in its dosage form, the drug is getting into the solution form. And once drug is in the blood, then how it is distributed and how it is getting stored in the tissues. And once it is at the site of action, then what is going to happen? If though there is a free drug, it will get bound to its proteins and to blood cells. And only the free drug, then it is ready for excretion also. So we have to see how much drug is bound, how much drug is free, because that determines the efficacy of the drug too. Now, this is the simple picture of the pharmacokinetic modeling, which talks about starting from the dose to reaching its administration site. Then it talks about absorption and bioavailability, distribution to tissues, excretion through various routes, and
Alka ma'am, you are not audible. Alka ma'am.
Hello.
Yes, ma'am, Alka, you are audible.
Yes, I'm back now. Can you hear me? Hello, can you hear me?
Yes, yes, ma'am.
May I know from where I got disconnected? Because I really don't know. Because I was speaking. So can you let me know from where did my presentation, where did it stop?
Ma'am, I think there were some network issue from our side. Everything was showing all right.
But from my side also it's okay. But now can you tell me from where do I need to start, you know? Should I start from the beginning?
No,
yes,
no,
yes, yes, yes. This side, from this slide only, pharmacokinetic modeling.
Okay, from this slide only. Okay, thank you so much. Now, when we are talking about the pharmacokinetic modeling, so we must know that we are talking about the route of drug administration, the drug, how it reaches the administration site, about its bioavailability, then about reaching the tissues. And this is the entire picture of the pharmacokinetic modeling. Now, the questions which we need to address at this stage is the applications of the pharmacokinetic modeling concepts. Now, when I'm talking about the different models, I must know that there are basically there are many models, but the models which we normally use in our research are either the compartment models or the physiological models or the non-compartmental analysis. Now, when I'm talking about the pharmacokinetic models, now if you look at the physiological models, now they are basically perfusion limited models, or they are the non-compartment models. Now, in non-compartment models, we only talk about area under curve, the mean residence time. When we talk about physiological models, they are basically perfusion limited or diffusion limited models. Now, when I'm talking about the compartment model, we are talking about the catenary model, mammillary models. Can you just let me know if you can see my slides, please?
Yes, ma'am.
Okay, fine. Now, under mammillary model, we are talking about one compartment, two compartment, and multi-compartment models. Now, when I talk about these models, like when I'm talking about the physiological models, we must know that these are basically the studies which are applied on animal species, and then they are extrapolated to humans. So they are based on either the physiological or the anatomic data. And what is basically responsible for distribution of drugs is the blood flow. And we can only predict some realistic tissue drug concentrations in this. So now this is what happens in the physiological model. Now, when I move to this more about this model, so I must know that in this no data fitting is required. And what basically determines the drug concentrations are the organ tissue size, its blood flow and drug tissue ratios and blood ratios, and any pathophysiological condition can affect distribution. If person is having problem with albumin concentrations, or a person is having any kidney disorders, liver disorders, they will all be affecting how the drugs they are having the journey in our body. Now, when I'm talking about the mammillary model, which you can see in this picture very clearly, it is basically used to study the kinetics of the protein metabolism. And there is this compartment Q1. So Q1 is the central compartment, which is surrounded by the peripheral compartments, which are Q2 and Q3. But Q2 and Q3, they are not connected with each other. So only these peripheral compartments, they are connected with the central compartment. So that is what happens in the mammillary model. And catenary model is also used, but not very commonly, because it is like a train. So what happens in the train, like you can see that there are different compartments. They are all like going from 1 to 3. So now 2 is only connected with 1 and 3. So otherwise these compartments are not connected with each other. Now, what we really need to know for our research in drug development are the compartment models. Now, basically what it talks about is that our body is consisting of several compartments which communicate reversibly with each other. Reversibly means it is not some physiologic or anatomic region we are talking about. We are talking about the tissues or the group of tissues which have the similar blood flow and drug affinity. So all organs which have high blood flow, they are put into central compartment, like we have heart, lungs, liver, kidneys, and all those organs which have poor blood flow, they are put under peripheral compartment. Like under peripheral, we again have some shallow compartments, we have deep compartments. Like deep is where there is minimum blood flow, like nails, we are talking about here. And shallow, we talk about our nervous system. So that is how to understand in the drug development process, we divide our body into several compartments. But when I'm talking about one compartment model, basically what we talk in compartment modeling is that any change in the blood concentration is happening because of which parameter. When I'm talking about one compartment model, I'm assuming that any change in the blood concentration in our body is happening only because of elimination. That means the distribution is same to all the organs. So that is one compartment model which we classify how we are administering the drug, whether we are administering through intravenous administration or through infusion or through first order absorption or through zero order absorption. But practically, if you look at in our lives, it doesn't happen because we have drugs which are moving to different organs and there is different distribution equilibrium. So depending upon the distribution equilibrium components, we have 2 compartments, we have 3 compartments, we have 5 compartment modular drugs. So this is the simple equation which we normally use to measure the concentration of drug using the compartment model at any particular time. Now, if you look at this, it is talking about the initial concentration. It is also talking about the elimination constant. So elimination plays a very important role when we are talking about the modeling concepts. Now, if you look at this, how even one compartment models, as I told you, then when we give drugs by different routes, how it is changing. I just give you very simple example. If you are giving in one compartment model, you are administering the drug through continuous intravenous infusion. Now, what is happening after intravenous infusion? After sometimes the steady state is reached. But now, if you are changing the rate of administration, now the time taken to reach the steady state is same, but you will be able to achieve the higher concentration in the blood. So that is what happens in the one compartment model. Now, in one compartment model, when we are talking about the infusion, now you can see that this is the equation which is used for the steady state, which is the zero order infusion and the first order elimination. Now, order of kinetics is again very important, whether the drug is following zero order or it is following first order. Although we know that most of the processes in our body, they follow first order kinetics. Now, 2 compartment model, as I mentioned before, it is the most common model and which divides our body tissues into basically 2 broad categories. Central compartment having all the group of tissues which have very instant equilibrium. The moment the drug is in the blood, we achieve fast equilibrium with all the organs, and the sample compartment is the central compartment only in this case. Now, when we talk about this central compartment, we should also know that we have a tissue compartment which has slowly equilibrating tissues, which includes again deep and shallow. Now, in this case, when we are measuring the plasma concentrations of the drug, we must know it is bi-exponential. That means there are 2 components which are involved in the change in blood concentration. Now, one is the distribution factor and second is the elimination factor. So we cannot ignore, because after the drug is in the blood, it also needs to be distributed. And we know that different organs will have different distribution equilibrium. So that is the most common model which is used for our drug development research. Now you can see that in the blood, there is initial decline, which was because of distribution. And then there is again decline because of elimination. But as the decline happens in the blood because of distribution or elimination, the concentration in the tissues, it starts increasing. So that is the journey of the drug, which we need to understand in our research process. Now, this is the picture which is showing central and the peripheral compartment. Why two peripheral? Because they're showing different equilibrium constants. Now, when we talk about this two compartment, just look at this picture, which is very, very important for us. This is before the drug is administered. This is immediately after you administer the drug. Now you can see from this picture, where is drug reaching immediately? Drug is in the heart, it is in the lungs, it is in the liver, it is in the kidneys. Why? Because these organs have very, very fast equilibrium because they're richly supplied by blood. Now, after some time, you'll find that the drug is now there in the entire body. Now, because slowly the drug is also reaching the slowly equilibrating tissues, and that is what happens to the drug after a certain period of time. Now, this is the picture which shows now the area under curve. Now you can see the amount of drug in the blood. Now you can see that in two compartment, you'll find that there is now the drug has moved from the blood and it has moved to the tissues. Now, drug is in the blood, drug has moved to the tissues, and any change in this concentration of drug in the blood is either because of elimination or it could be because of the tissue distribution. Now, let's talk about the 5 compartments. How are we talking about 5 compartments now? 5 compartment means that different compartments, they have different distribution equilibrium. Now, there are compartments which are very highly supplied with blood. So that is V1. V2 is some lean parts which are having a different equilibrium. V3, which are having intermediate. V4, we are talking about, again, very poor. And V5, we are talking about having the least blood supply and the least distribution equilibrium. That is how the compartment modeling becomes very important. We must know which compartment model our drug is following. So this is now showing the distribution from the blood to various organs, specific organs, general organs, which we must know if we are making any new drug. Now, which are the factors which are now changing the distribution? Person has low albumin. What is happening when person has low albumin? That means the binding, because most of the drugs, especially the acidic drugs, they bound to albumin. When albumin concentration falls, the binding will also fall. Now we have, what will happen? Now binding falls, we have more free drug. Either free drug will be eliminated, or if it has high concentration in the blood, it will cause toxicity. Now, again, if there is problem with the heart, cardiac output is changed, the drug reaching the various organs is changed, the circulation problem, diabetes problem, all these are going to change the distribution equilibrium. Now, if you look at this picture, it is talking about, again, various organs, and it is showing that how drug is now going to different organs with different distribution equilibrium. Now, normally we think that volume of distribution is same, but you must know that if drug has a very large molecular weight, it has strong binding to plasma proteins, then they will be having very low VD. Low VD means the drugs, they are confined in the blood. They are not moving to the tissues. But then we have drugs which are moving to the tissues, like we have drugs Can you hear me now?
Yes, ma'am.
Talka, you are audible. Yes. Okay, fine. So now this is the slide I was talking about, that drugs have different volumes of distribution depending upon their molecular weights. Like we talk about drugs with high molecular weight, they have very low volume of distribution. And there are low molecular weight drugs which are hydrophilic, which cannot cross the lipid membranes. They have medium volume of distribution. And that means they are either in the extracellular fluid or in the intracellular fluid. And then there are drugs which have very high volume of distribution, which is more than 42 liters. That means they are in the tissues, and they are the ones with very low molecular weight, and they are hydrophobic. So now we need to understand what type of drugs we are handling. And if we know what type of drugs we are handling, and when we will automatically know that what is their volume of distribution, in which volume they are contained, and that will help us to develop our drug products. Now, that is why we need to understand our compartment modeling. It will help us to understand how the loading doses and their plasma concentrations, they are happening. I'll just give you two examples before I finish, and how we can actually circumvent the problems of toxicity and adverse effects. Now, these are some formulas which we use for distribution, clearance, half-life, bioavailability, which we must know in the drug development process. And we should also know when the drug is reaching the steady state, which is the state when the rate of administration is equal to rate of elimination. And drugs which have very short half-life, they reach the steady state rapidly. But there are drugs which have very long half-life. It might take from days to weeks to reach the steady state. Now, if we do not know this, then we will not be able to develop the drugs. Now, if I have to calculate the loading dose is normally given in medical conditions where high concentrations are needed very quickly. We should know what concentration we are trying to achieve, and then volume of distribution. So already I mentioned that volume of distribution is very, very important factor in drug development process. Now we have initial volume of distribution when drug is in the blood. We have high volume of distribution when drug is in the tissues. Now, when I'm talking about the examples, I should know that where my drugs are going to exert their effect, like lidocaine, phenobarbital, procainamide, they will exert their effect on the organs which behave as if they were located in VI. VI means in the blood. Now, it is sometimes the organs are present in the central compartment, but they behave as if they were present in VT. Now, that is the example I need to mention now. Like suppose when we are giving potassium we know is a electrolyte fluid, but this follows the principle of two compartment modeling. Now, why? Because it has a parallel effect on the heart also. If we administer potassium and we do not control its rate of administration, it will cause immediately the effect on the heart, and person might even die. So what we can do, because we know potassium follows this two compartment modeling, and the organ is behaving as if it were present in the initial volume of distribution, what we will do, we will control its rate of administration. We will give it very, very slowly so that the person has not having bad effects on the heart. Now, I'll just show you a picture of digoxin. Now, digoxin has an effect on the heart, and heart we know is present in the central compartment. And the moment the drug reaches the blood, it should have immediate effect on the heart. But it doesn't happen. Why? Because digoxin follows principle of two compartment modeling. Now, this is the picture. Now, at time 0, this is only 0 concentration in the blood. Now, after 35 minutes, still the right concentration has not reached the heart. After 70 minutes, again the right concentration is not reached. After 105 minutes, again the right concentration is not reached. It takes about 3 to 4 hours, even after IV administration, to reach the organ, drug to reach the organ. Now, if we do not get the action and we start giving higher dose, what will happen? The person is definitely going to die. Why? Because we are exceeding the dose. We, under compartment modeling, we know we have to wait for 3 to 4 hours after even the IV administration, or to see the effect of the drug to happen on the organ. Now, this is the example of the 2-compartment modeling or the multi-compartment modeling. So sometimes it takes about 6 hours after the oral dose for digoxin to have effect on the heart. So we have to wait. And because it is the organ, although it is in the central compartment, it is behaving as if it were present in the tissue compartment. That is how the compartment modeling has helped to develop the drugs and to find out the right time and the frequency and the concentrations. Now, if you're talking about the maintenance dose, which is loading dose is normally double the maintenance dose. So in the maintenance dose, we must know about the clearance of the drug. How clearance is happening, whether there is any problem with the liver, problem with the kidneys, because clearance will change. And the time taken to reach the steady state will also change. So these are the factors which we all must consider when we are developing the drugs. And now there are so many pharmacokinetic modeling softwares, like non-MEM is a very common, then PHYSIC PK, integration of R within PHYSIC PK, then phonics with non-lin. So these are some very important these models which are available. So I would just like to end with some take-home points, that when we are searching for new drugs, it includes drug discovery and drug development. And drug development, we are talking about its efficacy and its actually therapeutic action. It also helps us to predict drug behavior and its toxic effects. It also helps us to design the dosage regimen, its route of administration, its proper doses which need to be given. Now, these are some of the publications which we achieved using this pharmacokinetic modeling concepts to develop the drugs. We actually could prepare some developed drugs for Parkinson disease with some of our PhD students. We did work on that. Then curcumin loaded nanocarriers, they were used to improve bioavailability using modeling concept. Then we could use some gastroretentive drug delivery systems. Then we could also prepare some chronomodulated drug systems. So these are just few examples. And thank you so much. Remember that the drug development process is a critical step which we must understand properly and must use the pharmacokinetic modeling principles. Only then we will be able to achieve a good drug product. Now, thank you so much. This is the place where I live these days. That's Muscat, Oman. It's a very beautiful country. So all of you are invited to visit. And if you have any questions, I'm ready to take the questions also. And I really apologize for the technical error which kept on happening. So I don't know what it was, but thank you so much for your patience. And I really appreciate your patience for this technical glitch. Okay, thank you.
Thank you, ma'am, for sharing your expertise on pharmacokinetic modeling in drug development. Now, may I please request our panelists, Professor Bharadwaj sir, Professor V.K. Kapoor sir, Professor Tejveer sir, and Dr. Shushma ma'am to begin the question and answer session. Over to Bharadwaj sir.
Just before Sir speaks, I would again like to thank the organizing team, and I would also like to thank Dr. Ravinesh, who very patiently was communicating with me and who was also part of the organizing team, and also my own batchmate, Dr. Sushma Sharma, and who has been with me through the journey which we had, and especially no words to thank my own teachers like Professor Khar, Professor Bharadwaj, Professor Kapoor, and many others, you know. Okay, thank you. Thank you so much.
Thank you, Dr. Alka, for your very
Thanks,
encouraging talk. In fact, pharmacokinetics studies give very good favorable directions with respect to the formulation specifically and the delivery also, delivery the systems and how to maintain the dose, what is the drug administration, eliminations, routes, and those kind of things. And many of the drugs have been again identified on account of the metabolites, which are again identified in the elimination steps, which have again thereafter they have been developed into very good results, very good drugs also. So thank you for being with us and encouraging our participants. Thanks a lot.
Thank you, sir. Thank you so much. Okay.
That's it. Hello, Alka.
Hello, sir. Sir, it's your real motivation and inspiration for all of us to see all of you working so hard to build up institutions. Really proud. We feel really honored and proud of that, sir.
Thank you so much. It was a real, treat to listen to your lecture, which was full of information and authenticity. And I feel a sense of pride because you have been a student of UIPS and you have been taught by us. So all the best wishes for the future endeavors. Thank you.
Thank you, sir. Thank you so much, sir.
Okay. I would like to ask a very penetrating question, which is what is the relationship between distribution coefficient? You said that distribution and the dose of the drug. So
yes,
how do you determine the distribution and the dose?
Okay, sir, that is a very good question. Basically, as I mentioned before, that distribution is dependent upon so many factors, including blood flow and the binding to proteins. Now, if a drug has a very high binding to proteins, that means there is very less free drug. And only if the drug is free, then it will be able to go to various organs. So first we have to study its binding properties, and then only we can see that where this drug is getting distributed, and then the nature of the drug and its dissociation constant. Because if the drug is ionized, then it will not be able to permeate through the lipid membranes. But if drug is in the unionized state, then it will be able to penetrate the lipid membranes, and its volume of distribution, all these, there is a study also that drugs which are having very high molecular weight, they'll be only confined to blood. But drugs which are having very low molecular weight, they will be able to reach the tissues. Like digoxin has a very high volume of distribution, and it is about 500 liters. So we know that drug is getting stored in the tissues. So that is how with the volume of distribution, we know that whether drug is in the blood or it is in the tissues.
Great. Now I would like that Professor Sushma should ask something.
Thank you.
Thank you, sir. Thank you very much. Hi, Alka.
Hi. How are you?
I'm good. What a treat it was. I'm sure my students, this is the subject which I'm teaching, and my students, they must be really benefited. You have really covered everything so beautifully, and something which I have learned, well, I'm always learning from you. You are quite ahead of me in this, is that what prospects we can have in research. But one thing I want to ask is, like theoretically, it looks very easy that we find the coefficient, the distribution constants, the distribution. But how easy it is to find the distribution, the volume of distribution in the whole body. Say even for digoxin, like you're telling, that it gets distributed but doesn't go to the heart to give its effect immediately. This is what I think you said. It takes a long time.
Yes.
So how
why
difficult it is to assess these things?
Yes, thank you, Dr. Sharma, for a very good question. Basically, when the drug reaches the market, all these studies, they would have already been done, because drug cannot reach the market till the time you have completed pharmacokinetic studies, and all these values are already reported in the literature. Now, if the volume of distribution is reported, that means it has been studied over a vast population. Now, only if there is a disease, pathophysiological condition, this volume of distribution will change. Like, I'll give you a simple example of edema. When there is extra cellular fluid, there is extra fluid in our body, any drug which is water soluble, we will have to give the higher dose for that drug. So knowing that this drug is soluble in water and person has extra water now, so, and there is edema, so there is a pathophysiological condition which is there, which is changing volume of distribution. Now there is obese person. Now obese person has more fat. Now, if the drug is fat soluble, we cannot give the same dose for the same drug. So we have to modify the dose. So already these constants have been studied. So in your development, you can also have variation depending upon their age. And because children, they have more water, they have less fat. And obese people, they have more of the fat component. So already these factors have been studied in drug development process. So we have to mention the entire profile when we are bringing the drug to the market. No FDA or no regulatory body will approve our drug till the time we have complete pharmacokinetic profile. And that is how they will study. Like for digoxin, if they would have studied, okay, they have heart. Heart means immediate circulation. But when they studied, they realized that action was not happening. And they started giving higher dose. And there were a lot of calamities, where the casualties which happened. Then they realized that it was reaching the heart even after 4 hours of IV administration. So that is how the modeling concept came. That is why we talk about 2 compartments, we talk about 1 compartment, we talk about multi-compartments. This is a must for us to report when we are preparing the drug. And this process happens because these studies are normally done on animal species, then it reaches the preclinical research. That is why it's a long journey.
This subject looks to be difficult and challenging so far as research goes.
Yes, but I have been using this in my research for almost, you can say, since 1986. So I have I have a lot of experience which I've accumulated in this area, and plus I'm teaching also these subjects to a master's PhD students. So I have really gone into depth, and I find this subject to be the most interesting subject because it really involves science in that. It involves science, basically, how we can bring drug from bench to the bedside, you know.
So it means we'll have a very long association, madam, and we'll keep getting your inputs into our research projects too. Thank you very much for being with us. Thank you. Very, very
much.
Always
welcome. Thank you. Yeah. Thank you, sir. So it was really a privilege to see you and interact with you, sir. Thank you so much. Thank you. Thank you. And
yes. Any other students? Anybody has any questions?
Yes, ma'am, I have a question.
Shashali.
Ma'am, which could be more accurate and better between animal modeling system or a software modeling system?
Actually, there are a lot of softwares which are available now. So, but for using the software, you should have initial data with you. So if you do not have initial data for already existing drug modules where you already have lots of data, you can use softwares to shortlist, but initial studies on animals are definitely required to study its toxic problems, to study its histopathological effects. How will you develop a model without studying those things, you know? So still we have to do all those studies before we start the using, and basically models are used for analysis only. We take blood samples, we study its concentrations, and then we analyze which compartment model does it follow. So you have to use a combination of both, you know. So,
okay, ma'am.
Yeah.
Is there any question from a panelist? panelists ne, sorry, our attendees.
What I feel is that this compartment modeling, if you read, looks very simple. But when you practically have to deal with it, I feel it looks to be very, very challenging.
Yes, it is. It is because you have to study so many factors. It starts with the simple physico-chemical properties, starting from the dissociation, its solubility, and then we are talking about its reaching the blood, then talking about how it is reaching the organs, and for how long it stays there, and then what are the different mechanisms. So there are different aspects, you know. Yes, it is. It's a very, very deep subject. Yes, you're absolutely right. Yes. Yes. Somebody is asking that how will we do pharmacokinetics of some, I couldn't read the question, of polyherbal drugs.
Formula. Yes, yes, ma'am.
Yeah. So if you are talking about the polyherbal drugs, we have lots of constituents in that. So first is we have to have a marker, and the marker has to be studied, you know, the marker, because there are so many constituents. So you have to have the profile of the marker with which you are comparing. So only then you can be studying about that, you know. So some are asking how are ADME studies different for vaccines and biologicals, right? That's what they're asking.
Yes, ma'am, that's the same question.
Now, first question for vaccines, there is no absorption step because it is directly going to the blood. And when we are talking about its profile, that for how long they stay in the body, you must have seen from your own experience that in COVID vaccines, they were also not given as one dose. They were given in also as a booster doses also, second dose, because they found that its efficacy was decreasing. So all these studies, they have to be done that for how long their efficacy remains in the body. Okay, so that is the study. If I've understood the question, you know, properly. So that will clearly tell you. That's why all these booster doses, etc., had to be given. Okay, now I can see somebody asked one question which I couldn't read. I did 150 compound something, no?
Ma'am, I am reading the question for you. It is Sibu Sen. His question is, I did phytochemical profiling and got 150 compounds showing good score. How to proceed further for pharmacokinetic studies for a polyherbal formulation?
150 compounds you got. Okay, 150
yes,
compounds is it is only the synthesis part, you know. So you have only done the drug discovery part. When we are talking about this 150 compounds, you have not reached the development stage. Okay, you have only talked about these are only shortlisting. So now you have to go about its pharmacokinetic profile. You have to study it in animals to see that whether the drug is actually getting into the blood also or not, you know. So that you have to study with complete its physico-chemical properties. Now there are so many softwares available for drug discovery also. So you have only talked about the first step, you know. Okay.
Ma'am, his question is, the marketed polyherbal formulations also contain 7 plants, more than 7, it can be more than 7 also in a polyherbal formulation that are already marketed in the market. So how can they proceed, or how can they did the pharmacokinetic studies before marketing?
Okay. Now I understood that. Now for polyherbal formulations, what is happening now, we all know that there is Ayush, you know. So now there are many drugs which are used in India, but many herbal formulations, they have still not gone through the complete studies. So that is why to market it globally, they have to do all these studies. So now even drugs come in the market saying that they are safe, but later on they are withdrawn. Now because they are found to have liver toxicity. So first step is to study your toxicity. When you are using any polyherbal formulations, first step is do toxicity studies, see its effect on various organs. Once you find that these drugs, they are not toxic, see that for how long they are staying in the blood, because their active constituents, are they really going to stay in the blood for a long time? or they are also eliminated fast. So you have to study if you want to market it globally. In India, yes, we are still doing a lot of shortcuts for polyherbal, but we cannot do shortcuts for our synthetic drugs. Our pharma market is not allowed to bring any drug to the market if they have not done the complete studies. But herbal, yes, still it is being done, saying that it is safe, but we know that kava, it is another example of herbal formulation which has been withdrawn because of its toxicity. So remember, herbal formulations are not free from toxicity. Okay, very important. And so many, I'm sure all of you have read this reviews and reports where people were found to have cancer because they were taking overdose of the herbal drugs. You know, there were also cases like that. So we have to be very careful that we are not even taking them like the food, you know. We think that, okay, we can chew them like food, but no, definitely no.
Sibu is saying that he is not satisfied yet. He want to talk to you.
Okay.
Sibu, can you raise your hand?
Hello, ma'am.
Yes, you can ask your question, Sibu.
Ma'am, actually, it's a good presentation from your side. And I wanted to ask that, like, I have a marketed polyherbal formulation in which I got 150 compounds from the phytochemical profile. So if I see the toxicity in different organs, so I have to see in different extract, preparing the extract or whole formulation I can giving the animal model to for seeing the toxicity, and how to going in further PK studies using like same standards I have. But as a separate standard, it will not show the same result as a mixture of the formulation. So how to proceed in that case for PK studies and toxicity?
Yes, you are absolutely right, because when there is a single constituent, sometimes they don't show toxicity, but when they are given in combination, they show toxicity. So that is why sometimes they extract those constituents which are safe. And also there are so many isomeric forms, etc., which you also need to study. But polyherbal, when you have lots of constituents, you have to study in the combination also, and you also have to study its individual constituents. Sometimes you have to extract a single constituent and then formulate in a dosage form. So many studies are done on that also. So there are some extracts with simple constituents which you have to extract and then use them in the formulation. You cannot use the entire, because one part could be toxic also.
Then I can't say, ma'am, like
I would also like to add here, a very common thing is drug interaction. When you're taking 7 constituents, maybe some 2 of them, they are interacting and becoming toxic. So maybe you have to then do these studies of 2 to see the interaction between any 2 of them. Am I right, ma'am?
Yes, you are absolutely right, because that's why I mentioned that sometimes there are certain constituents which are more active. You have to isolate them and use them in the formulation and remove the other constituents. So that is why, and also with one single constituent, you have to see which racemic form is effective. Sometimes lever form, sometimes rotatory form, also crystalline form, amorphous forms. It's a complete study. Now, if I'll give you a simple example of penicillin even, penicillin is active only in its crystalline form. Chloramphenicol is active only in its amorphous form. So we cannot use the amorphous form for penicillin. So you also have to go and see that which form is active, because it might not be toxic, but it might not be active also. So you have to see that that goes in the drug discovery stage. So in drug discovery, you have to study all these factors. You have an excellent mentor, Professor Kapoor, Professor Bhardwaj, who can teach you all these things. I learned only from them, you know. So what are these various isomeric forms, etc. So you can learn, because these are, you have to see which form is active, okay? And which form is safe also. You have to see activity and its toxicity also. Okay?
Okay, ma'am. Thank you, ma'am. And my later part is like drug interaction only, HDI. So like same as like PK studies, if we see the like single entity or compound, and from the profiling also I get to know same of having the mass same also. So from the fragmentation pattern, came to know that the two structures are different. So after that, like if we take as a single compounds or extract some of the compounds, so it will not like represent a full formulation. So how to like that I understand to separate and isolate the single compounds or two compounds interaction, like from the HPI normal drug or normal herbal drug interactions. So means if we are doing PK studies, so it will not justify for the formulation, only for the single compounds or a mixture of some certain compounds. So how will justify the formulation with the certain 4,3,5 compounds?
If you are having a mixture and you study and it is safe, it's fine. If any mixture is showing a problem, only then you will have to isolate it, Otherwise, you don't need to isolate. Isolation happens when you are finding that some mixture is showing some toxicity, So first do your toxicity on the polyherbal formulation or the extracts which you are getting, and only then you can go to the next step, Okay, so first study only the toxicity. If you don't find any toxicity problem, then you can go ahead with the formulation. See its solubility, because it will be also soluble in different solvents, So if it is very much soluble in the solvents, then you can easily make it in a formulation and study its bioavailability. It's not, it can be very simple also. You might not have any problem. If you have further questions, you can always get back to me. My phone number is with Dr. Ravinesh and with Dr. Sushma. You can always write to me. They have my email IDs also, and we can talk more about it also.
Okay. Sure, ma'am. We will share your details.
I can see a lot of interest of people. Yes. I can see a lot of people have. Yes. I have already my phone number and email ID are with Dr. Sushma and with Dr. Ravinesh. You can take it from there. I will be very happy and I'll be very glad to answer.
With all participants. We will share with all participants.
Thank you so much. It's fine with me. Okay.
Thank you, ma'am.
Thank you. Thank you so much. Thank you. I'm so glad that this topic raised a lot of interest, That's, I feel really blessed. Thank you. Thank you so much.
Our next session would be related by Kapoor sir on herbal drug interactions.
Yes, very good. So I think that will be good thing for all the answers to be, for those people who are having questions. I think Professor Kapoor, that's why I already said, he's a master. We learned from him. So you can ask all these drug discovery questions from him, He will tell you all the answers. Okay?
Yes, ma'am.
Okay. Thank you. Thank you so much. Thank you. Thank you. Bye-bye. Bye. Thank you once again. And my kind regards to all of you, especially to my mentors and my teachers. Thank you. Thank you. Thank you so much, sir. Thank you.
Thank you, ma'am. Thank you to all respected panelists and all participants to be with us. We will join again after lunch break. Stay with us and join at 2:30 sharp. Now it's time to introduce our next speaker, Professor Emeritus, Professor V.K. Kapoor sir, but School of Pharmacy and Emerging Sciences, Baddi University. Former Chairman, University Institute of Pharmaceutical Sciences, Punjab University, Chandigarh. Dean, Faculty of Pharmaceutical Sciences, Punjab University. Recipient of Professor G.P. Srivastava Memorial Award 2004 of the Association of Pharmaceutical Teachers of India, Bangalore. Recipient Shri Bhojraj Panjmul Life Science Time Achievement Award 2004 of the Association of Pharmaceutical Teachers of India, Bangalore. Recipient of Shri Bhojraj Panjmul Lifetime Achievement Award again in 2011. Awarded Commonwealth Academic Staff Fellowship by the Association of Commonwealth Universities, London for the year 1983 to 84. Recipient of Academic Excellence Award 2016 of the Association of Pharmacy Professionals. Awarded Shiksha Bharati Puraskar 2011 of All India Achievers Foundation, New Delhi. We welcome you, sir. Over to V.K. Kapoor, sir.
बस जैसे ए बोल के फिर आप नीचे करना
इसको
पहले इसका
Am I audible?
Yes, sir, you are audible.
Okay. Thank you. My greetings to all. Professor T.R. Bhardwaj, my colleague Professor Tejveer Singh, Dr. Ravnish Mishra, and all the participants to this conference. I'm indeed very grateful to the organizers of this conference for giving me an opportunity to interact with you. My topic is herb-drug interactions. Herb is related to botanicals and the products from plants, and drug here is indicative of the modern drugs, that is allopathic drugs. I'll be talking about this. More for centuries, the botanicals have been used for the alleviations of a number of diseases. In this respect, traditional medicines have been the integral source for health in household and communities. When we talk of Indian system of medicine, we mean Ayurveda. Ayurveda is the Indian science of healthcare and is one of the oldest system of medicine. And they have been in use right from Vedic period, that is 600 before Christ. The use of complementary and alternative medicines has increased in recent times, particularly the American and European countries. The World Health Organization has recognized the importance of integrating traditional system of medicine, particularly Ayurveda, with the conventional allopathic medicines. And WHO has taken several initiatives to promote this integration. The WHO Traditional Medicine Strategy was formed in 2014 to 2031. The Ministry of Ayush, which was earlier the Department of Ayush, and now it has been upgraded to the ministry, they have agreed to host 2 WHO working group meetings that took place at National Institute of Ayurveda in 2018 and at Institute of Teaching and Research in Ayurveda 2019 for developing WHO benchmarks in Ayurveda with the participation of 27 experts from the 6 regions of the World Health Organization. A very significant majority, up to 95%, they consider that Ayurveda can make important contribution to the conventional medicines and are in the favor of integration of Ayurveda and allopathic systems. The various benefits of Ayurveda in the treatment of human is that Ayurveda is the holistic approach. That is, it takes into consideration the physical, mental, emotional, and spiritual well-being of the individual, and this provides a personalized health care. It is used as a preventive measure. The drugs are less toxic as compared to the allopathic systems. The drugs are cost effective and they have complementary approach. You can combine with other system of medicine. This fundamentally the sorry for interruption. Recently, a number of herbal products in the form of dietary supplements are being consumed along with modern allopathic drugs. And this is particularly true for the geriatric patients, old age patients who consume a number of medications simultaneously. The consumption of herbal drugs is often associated with a general belief that these drugs are relatively safe and without side effects. This is an oversimplification and is not true. There may be many herb-drug interactions which has resulted in very adverse reaction and even death. And these herb-drug interactions can be synergetic in nature or antagonistic in nature. Fundamentally, the drug action is due to a number of chemicals present in the medicament, whether it is in allopathic medicine or herbal drugs. And these chemicals are metabolized in the body, particularly in liver, through a number of enzyme systems. And these enzymes are involved both for the xenobiotics, that is the chemicals which are given from outside, or endobiotics, the chemical substances which are present in body itself. And the herb-drug interactions can either be pharmacodynamic or pharmacokinetic in nature. In the first case, the constituents of the herbal products have synergistic or antagonistic activity in relation to allopathic medicines. And in pharmacokinetic interactions, the herb-drug interaction may involve inhibition of metabolic enzymes, resulting in increased level of one or both the drugs. And conversely, the herb-drug interaction may result in induction of enzyme systems, leading to underdosing of the drug to the patient. These are the fundamentals of drug interaction. Now, these are the enzymes involved in the metabolism: cytochrome P450 and its isoforms. These are the different forms of the main enzyme cytochrome P450. Now I will G-glycoprotein, PGP, also plays a role in the drug metabolism. I will now make you apprised of most common globally used herbs and their interaction with allopathic drugs, which may sound to be very alarming also. Hypericum perforatum is a drug which is used worldwide. It is commonly called at St. John wort, and the flowering tops or aerial parts are used in the treatment of depression as a self-medication. The two main constituent of Hypericum perforatum are hypericin, which is a complex phenolic compound, and hyperforin. Out of these, hyperforin is the potent inhibitor of serotonin, as well as norepinephrine and dopamine uptake. That is the mechanism of action of antidepressants. By suppression of the uptake of serotonin, the concentration of serotonin increases. With the result, there is an excitation or antidepressant effect. Similarly, the uptake of dopamine also results in increased concentration of dopamine, which has antipsychotic action. Now, I will discuss how dangerous is the use of St. John wort along with allopathic medicine. Use of St. John wort along with other serotonin reuptake inhibitors like allopathic drug, nefazodone, paroxetine, venlafaxine may result in this synergistic interaction with the herbal causing serotonin syndrome. That is not desirable situation where the excess of serotonin may produce some psychotic-like behavior of the patient who are taking both herbal drugs and the synthetic antidepressant. So those who are on the therapy of antidepressant, they should refrain themselves from taking this natural antidepressant, St. John wort or St. John wort. Second is a very common plant, Allium sativum, which is commonly called as garlic. The garlic is very widely used as a vegetable dietary item. We all consume along with our food daily allium. Garlic has been reported with several beneficial effects, like it is an anti-hyperlipidemic. It is given to reduce the level of triglycerides, to reduce the level of cholesterol. It's also a fibrinolytic. It helps in the cardiovascular disorder and is also an antiplatelet effect. These are the advantages of taking daily Allium sativum or garlic. There are two main constituents of garlic which are present in it. One is alene, which is a sulfur-containing sulfoxide, and diallyl sulfide. The smell of garlic and also onions is because of these sulfur-containing compounds. The garlic interacts with orally active synthetic anticoagulant drug warfarin. You see, warfarin is an orally active anticoagulant drug, and it is taken by all those patients who have the tendency of deep vein thrombosis or pulmonary embolism. Sometimes there is a coagulation of the blood in veins or in lungs. They coagulate and they form the clots within the veins, which is called as vein thrombosis. And when the clots are formed in lung, they are called as pulmonary embolism. Both these diseases, the patient has to take continuously some anticoagulant drug. Right? Anticoagulant drugs are those which prevent or prolong the coagulation of blood in vitro or in vivo. So warfarin is one of the most commonly orally active anticoagulant. And the patients, they have to take continuously, particularly the old patients which have tendency of the thrombotic embolism or pulmonary embolism. They have to take it regularly. Now, if along with this herbal drug or common food item, they are taking the warfarin, there is a increased bleeding. The anticoagulant effect of warfarin is neutralized, and there may be increased bleeding in the system. There is already a report of a 70-year-old woman, while being treated with warfarin, was admitted to hospital with multiple internal hemorrhage after having consumed garlic or chamomile, another herbal drug. This is the reference where it has been reported. Now, another common vegetable is Mamodica carentia, which is commonly called as bitter gourd in English and Karela in Hindi. So this has been reported quite extensively, and there is a general belief that the patients of diabetes who have high glucose level in their blood, they take lots of Karela or bitter gourd in their diet, and this helps in reduction of sugar in the blood. But there is a report that a diabetic patient which was on the anti-diabetic synthetic drug propamide, he took a lot of Karelas also in his or her diet, and this resulted in severe hypoglycemia. You should mean falling down of the blood sugar is a very dangerous sign. It is more dangerous clinically than even hyperglycemia. The person may go into coma if there is a severe hypoglycemia. And this incident has been reported by the British journal Lancet in 1979. So those patients who are on diet of warfarin, they should refrain from taking Mamodica carentia too frequently. Another plant is Glycyrrhiza glabra, which is commonly called as licorice, mulatti. And the dried rhizomes and roots of Glycyrrhiza glabra, they are used as a demulcent, expectorant, for ulcer healing, and mild anti-inflammatory properties. Whenever there is a mild cough which cannot be controlled by synthetic medicines or the syrups, it is recommended that mulatti is a very good demulcent. It will stop the unproductive cough. it will also have the expectorant action. So it is a very useful herbal drugs being used. It is also an anti-inflammatory drug. And besides these clinical features, it has a flavoring and sweetening agent also. It has a sweet taste, and in many condiments and in many agents, it is used as a sweetening agent. Chemically, the licorice contains saponins. Saponins are the glycosides of high molecular weight triterpenoids or steroids. They are the glycosides because they are linked with some sugar molecule. And they are called saponins because when you shake a saponin glycoside with water, it produces a lot of froths, foams. That's why it is called as saponin. It behaves like a soap. Its main constituent is glycinic acid, which is a hydrolytic product of glycerinatic acid. Glycerinatic acid is a saponin. It has the molecule of sugars combined with it, but if you hydrolyze it, you get the non-glucose component, which is known as glycerinatic acid. Now, glycerinatic acid has been found to be an inhibitor of controlled metabolism, resulting in concentration in the body. It is an anti-metabolic agent. And this has been reported that licorice interacts with prednisolone, resulting in its increased plasma concentration. You see, if you take a lot of licorice, it causes inhibition of the enzymes, metabolic enzymes of steroidal compounds, with the result, for example, cortisol is naturally produced in the body. And if you take licorice along with this, the level of cortisol may increase. And if you are using prednisolone as an steroidal anti-inflammatory agent, the concentration of prednisolone will also increase because of the inhibitions of the enzymes by glycerinating. So those who are taking steroidal drugs, they are advised not to take licorice or mulati either in the form of diet or in the form of cup suppressant. Another commonly used plant is Tamarindus indica, imli, which is commonly called as imli. It is a fruit of a large tree, which is Tamarindus indica, very commonly grows throughout India and particularly in South India. The imli is used very much in our dietary items. Those who take this southern food, they will observe that tamarind is present in South Indian food, in dosa and sambar and all that. It is a laxative. If there is a constipation, Tamarindus indica is a useful laxative. It contains tartaric acid and citric acid. And that is because it is a laxative and gives a taste, sour taste to the food items. It may be mentioned here that the medical statistic reports that South Indians, they have very less incidence of stone formation in uterus or in bile duct or in kidney. They are comparatively to North Indians. South Indians, they have less incidences of the stone formation in their body. It is because the South Indians very largely consume tamarind indica in their diet. They take, along with sambar and all their food items, they must contain imli. And because of the presence of tartaric acid, all the extra calcium which is present in the body is converted into calcium tartrate or calcium citrate. And this is the only salt of calcium which is water soluble. All other calcium compounds with inorganic acid, they are not water soluble. So formation of calcium citrate and calcium citrate, this because of being water soluble is excreted out of the body in the form of urine. So there is a lesser concentration of calcium, and stone formation is due to the formation of calcium salts in the body which cannot be excreted because of their water insolubility. So this is a very good use therapeutically for those who have a tendency. Some patients have a genetic tendency of formation of stones in the body, but there is again interaction with it. If one is taking aspirin daily and taking heparin along with it, it increases its bioavailability because it suppresses the enzyme metabolizing aspirin, and thus it increases the concentration of aspirin, which may be harmful to the body. It may cause ulceration of the gastric system. There is another plant which is Silbum marianum. The fruit of Silbium marianum is commonly known as milk thistle. The fruit contains the active principle silymarin, which is a mixture of flavonoids, and the isomeric 3 flavonoids are silibinin, silicristin, and silibinin. Silibinin is the major constituent. Silbium marianum is an anti-hepatotoxic drug. It is used in many of the Ayurvedic formulations as a hepatoprotective drug. And the beneficial effect of hepatoprotection is carried out by one of the isomers, which is silibinin. And is a very commonly used in United States as a dietary supplement as a hepatoprotective. In India, many of the hepatoprotective drugs like Liv 50, and there are a number of 17 or 18 syrups available in India which are used when the liver disorder is there. But in United States, it is cleared by FDA to be used as a dietary supplement for protection of the liver. And silibinin significantly inhibits cytochrome 450, isoform 3-1-P, and G2P gluconide transferase. These two enzymes are inhibited in the body, which are metabolizing enzymes. So if you are taking any drug which should be immediately metabolized and excreted out of the body, it will get accumulated and cause its toxic effect. Now, paclitaxel or Taxol is a plant product which has been isolated from bark of Taxus brevifolia, which grows only in America and is commonly called as yew tree. From it, the Taxol, which is a terpenide, was isolated, and you may be glad to know that one of the Indians, Mansukh Wani, was associated with the discovery of paclitaxel as an anti-cancer agent. Mansukh Lal, he was Gujarati. So, whenever you are taking the milk thistle or silymarin as a dietary supplement to give you a protective liver, one should be cautious that you should not be taking allopathic drugs, particularly Taxol or many other drugs which are likely to be metabolized and excreted out by cytochrome P450 isoform 3A4 or butyl gluconide transferase. So there are many examples available in the literature where serious drug interaction may take place, and some of them have resulted in fatality also, particularly in the case of warfarin, where there can be severe bleeding in the body, hemorrhage can take place, or the anticoagulant effect because of anticoagulant effect, and particularly when you are taking St. John wort and you should not take synthetic antidepressant. So after discussing these examples of the commonly used drugs and their interaction with allopathic drug, I pass on to another concept which is related to this concept of synergism and antagonism, which is very much related to the use of herbal drug or herbal synthetic drugs or synthetic drugs. Now, a herbal drug consists of several different classes of compounds in which the bioactive constituent represents only a small fraction of the whole extract. See, if you extract a plant, say berberis plant for the production of resort, right? And you extract the berberis roots or berberis leaves, you will get a extract, alcoholic extract, and in which the berberine will be present in very small amount. Now, when it is not only berberine which is present when you extract the berberis plant, along with berberine, other constituents are also isolated, which may have the same activity and may perform different roles. They may act as a diluent. For example, you take digitalis. Extract of digitalis is used in cardiac failures, and the sugars which are present along with digoxin, they act as a diluent. They reduce the toxicity and they also increase the oral bioavailability of digoxin. Or the other constituent might also potentiate the action of the main ingredients. Or reversely, they may reduce the desire and activity. That is, they may have the other constituents which are present in small amounts. They potentiate the action or antagonize the action, reduce the action. And the recent studies which have been carried out, they give credence to these facts. We can see some of the reports which have been cited in the literature. In Acacia angustifolia, which is used as a laxative. For constipation, the Senosides are recommended to relieve the constipation. Senosides are the active constituent present in Cassia angustifolia, senna. Though they contain to the extent of 3 to 5% of these Senosides for their purgative action. Now, another anthrone derivative, this is alloamidine. It's also a glycoside. It has no cathartic activity, but it potentiates the purgative activity of Senoside A in mice by 1 to 1.3 times. This is the reference where alloamidine present along with Senosides, they may potentiate the action. Recently, artemisinin is the second natural product which has been discovered in plant kingdom with anti-malarial action. The first drug with anti-malarial activity was quinine, and it was in China that the leader Mao Zedong had asked their workers in the scientific field to come out with an alternative of quinine or chloroquine or other synthetic anti-malarial agent, because it's related, it's very close country, Vietnam, is under the attack by United States. And the war, Vietnam War, was fought in jungles. It was a guerrilla war. They used to attack the Americans from the bushes, with the result that there is a high incidence of their suffering from malaria because of malarial parasites. And the Europe has cut off the supply of quinine or another anti-synthetic anti-malarial agents, and they were dying of malaria instead of being fired on. So Mao Zedong had ordered or given the suggestions to, those engage in phytochemistry, that you come out with an substitutive anti-malarial agents. And there was a lady pharmacist. She was a B.Pharm from University of Beijing, who was given the credit of isolating artemisinin, a endoperoxide from Artemisia annua, Ma Huang. There it was called as Ma Huang. I may tell you that Artemisia annua does not grow in India, although there are 50 other species of Artemisia which are found. And the attempts by the CDRI or other CSI agency to grow annua, Artemisia annua, have failed. So we have to depend on the supply of artemisinin on China. So that's how the artemisinin was discovered in 1972. And for the discovery of artemisinin, that lady, which was named as U22, was given the Nobel Prize in Medicine or Physiology. This is the first case where a pharmacist has been rewarded by the highest recognition, Nobel Prize. It should be in your knowledge and it should be in your records that a pharmacist got the Nobel Prize, of course, along with two others, for the discovery of artemisinin. Now, artemisinin suffers from the disadvantage that it is not water soluble. So it has been converted into semi-synthetic compounds like artemisinin artesunate, Artemis ether, ethyl ether, methyl ether, which was relatively soluble. Now it has been reported that trisophenol T and cryoplantin, the flavonoids occurring in plant, have no antimalarial activity, but they potentiate the antimalarial effect of artemisinin. This is the report made in 1992 that along with artemisinin, another flavonoidal compounds are present there which may potentiate the activity of artemisinin. As we have mentioned that again cyanosides which are present in Acacia angustifolia, the alloamidine, which is another anthraquinone glycoside, is present along with it, and it itself doesn't have cathartic activity, but it potentiates the purgative activity of cyanoside A mice. A very remarkable observation. Now, berberine is an alkaloid which is isolated from different Berberis species. The extract of berberine in India is used as a rasant. It is very bitter substance, and our grandmothers and others, old ladies, they used to give rasant in very minute doses for their antibacterial activity. The active constituent of berberis or salt is berberine, which is an quaternary alkaloid, water soluble. Now this has a potential antibacterial activity against Staphylococcus aureus. Now the plant also contains another constituent which is methoxyhydrocarpin. This component doesn't have any antibacterial activity itself, but is present along with berberine. And it is reported that the presence of methoxyhydrocarpin increases the antiprotozoal activity of berberine by a factor of more than 100. The antiprotozoal activity is increased 100 times when methoxyhydrocarbon is present. You may refer to this reference. Glycerida glabra, about which we have just talked. There is a plant, Echinacea purpurea, the chief constituent of which is chicory acid, the best-selling immunostimulant, very commonly used in Europe, Echinacea purpurea. And it was demonstrated that mixing Glycerida glabra root extract with that of Echinacea purpurea leads to potentiation of immunostimulant activity of the latter. This chicory acid. It may not be consumed in India, but is a very popular immunostimulant drug used in Europe and other Western countries. Then there are multi-component Ayurvedic formulations. So there has been a discovery made in RRL Jammu, which is now renamed as Indian Institute of Integrative Medicine. Professor Atal, who was a product of University Department of Pharmacy, was the director at that time. He made an observation that almost all Ayurvedic formulations, they have one common component, which is Piper longum. And he asked many of the practitioners that what is the reason that all the formulation, Ayurvedic formulation, they contain piper longum. So they could not give any logical answer. Then he started working on it. And it resulted in the isolation of a compound which was named as piperine as a bioenhancer. First of all, it was demonstrated that addition of long piper longum, it potentiates the anti-asthmatic action of leaves of Adathoda vesica, which is reported to have use in cough. And the substance which was reported as piperine, it was found to be a bioenhancer. When it is given along with other synthetic compounds, it reduces its metabolism and its potency increases several times. This discovery was purchased by Cadila, and they were marketing at one time the rifamycin for the treatment of the patients of tuberculosis. The dose was 400 milligrams. But when it was given along with piperine, the dose came down to 200 milligrams. So this was a new achievements in the field of natural products. Bioenhancer came. Later on, work started in many CSIR laboratories to find another bioenhancer, but they have not been successful. The only success story is with regard to cow urine. Cow urine has been reported to enhance the activity of other synthetic stuff. So thank you very much, participants, ladies and gentlemen, for your patience hearing. This is what I had to discuss with you. Thank you. Thank you all.
Thank you, Professor V.K. Kapoor, sir, for enlightening all of us. Your insights were truly invaluable. I may please request our panelists, Professor Bharadwaj, sir, and Professor Tejveer, sir, for Q&A session. Over to Professor Bharadwaj, sir.
It's a very good, informative, as well as, again, it tells us about the safety aspect of the drugs, as is otherwise considered that Ayurvedic or herbal drugs are always safe. So that may or may not be true. It has to be well documented and studied. Only then thereafterwards we can say with certainty that they are safe. Of course, as is said by Professor V.K. Kapoor, sir, that there can be interaction between the drug, herbal drugs, or the Ayurvedic drugs. If they are taken along with your allopathic drugs, modern drugs, there can be some interactions, and some of these are very, very serious, fatal also. My request to Dr. Kapoor, sir, is that how he feels about the safety of the herbal drugs or the Ayurvedic drugs. Are they really safe or always safe as compared to the other drugs whose toxicity has already been enlisted or is given in the literature? Yes, sir.
Thank you, Professor Bharadwaj, for this question. Although the common thinking is that Ayurvedic drugs are not toxic. Now, this is an oversimplification of the thing. But there are a number of plant drugs which have very high toxicity resulting in death also. A number of croton species, they have been found to have the croton alkaloids, which are highly toxic. In China, a formulation was made and it was selling as a hot cake in the Western countries to reduce the body weight or to reduce the obesity in women particularly. The drug was very popular and a mixture of many of the agents, in which that was found that one of the ingredients was crotalaria. And crotalaria presence was highly hepatotoxic compounds. That drug, which had earned millions and millions, was ultimately withdrawn. So, croton species, astrolochic acid containing drugs, or Cygenelia, there are some drugs, even Datura stramonium is a very toxic plant. So it is not true that all the Ayurvedic drugs are safe. They have their toxic aspects of the also. Strychnos nextvomica, it contains strychnine, which is a CNS stimulant, but the stimulant action is to the extent that there is a convulsion. There was a case, it may be about 40 years back, that in Moga there was a vet who was giving the drug for these small children who suffer from the disease of Suka. सूखा के लिए बड़ा बच्चे वेट रिड्यूस करते थे, खुजली होती थी. It was very popular. Ultimately was found that many of the children, they died ultimately by consuming that Ayurvedic formulation given by that Ayurvedic practitioner in Moga. And the drug was found to contain Strychnos nextvomica. So there are cases of this type. Strychnine at one time was official in British pharmacopoeia as a bitter tonic to increase the appetite. But since its CNS stimulant action is so profound that persons taking it may suffer from convulsions, so it was withdrawn from the pharmacopoeia. Now, the other related alkaloid with strychnine is brucine. Brucine doesn't have that anticonvulsant action, but to that extent, strychnine has, and is mainly used as a rodenticide. Both strychnine and brucine are used as rodenticide to kill the rats and other rodents.
There was one news few days ago. It was related to the Japanese nutraceuticals that 8 persons died.
Yeah.
And maybe 140 or they were hospitalized also. It was very popular nutraceutical which was being exported from there. And ultimately they had to withdraw that from the market as soon as this happened.
Yeah.
And it was considered by the proprietor or the scientist of that company that it contained some molds which were highly toxic. Mold, yeah. Fungal. So my question is that such type of nutraceuticals, they should be under prescription, not OTC. So that such kind of these things can be avoided. What's your opinion, sir?
Yeah, I agree with Professor Bhardwaj that many of the Ayurvedic drugs, they are being exported to Europe and to America as a dietary supplements without prescription, without their proper standardization. Few years ago, there was a report against the Ayurvedic formulations which was being imported in the European countries, And that report appeared in most prestigious journal, JAMA, Journal of American Medical Association, that all the Ayurvedic drugs which are imported from India should be banned because of the high contents of the metals, cadmium, arsenic, copper, other contents. But it was nullified by Ministry of Ayush that the metals contains in the Ayurvedic formulations, they are in the form of bhasmas and not the free elements. In Ayurvedic formulation, one of the form of drug is bhasma. You may have churna, you may have rishtas. One is bhasma. Bhasams, they are, in today's terminology, they are the nanoparticles. And these are prepared along with the mixture of herbal drugs, giving, which is in their own language called put in the furnace of their own, not in the furnace available in the market, but it was the underground furnace which was given 8 puts or 80 puts. And those bhasmas, they were used as gold bhasmas, silver bhasma, copper bhasma, and all those. So they don't have that metallic action. They are not metals, but now they are converted into oxides or sulfides. So this explanation was given by Ayush, and to some extent they were accepted by JAMA. And this, but nevertheless, all the Ayurvedic preparations, I mean, TCM, traditional Chinese system of medicine, there are 75 TCM formulations which have been approved by FDA. But none of the Indian Ayurvedic formulation has been approved by FDA so far, because they have given all the data, the complete data of the toxicity, the complete data on the carcinogenicity or mutagenicity, and they have given proof of their efficacy in certain cases. But unfortunately, Indian workers have failed to do that. Still, there is a strong demand from the minister that you should produce strict parameter for herbal drugs so that they can be accepted by American and European markets, particularly with regard to their toxicity, their reproducibility, and the safety.
So, thank you. Thank you. Yeah, over to Professor Tezvi.
Certainly, Professor Kapoor's lecture was very interesting. It raises various questions, but I cannot dare to ask him the questions. I can only have few suggestions. Suggestion is that in South India, if turmeric, imli is used, that must have a tartaric acid, a particular isomer, because whatever he has shown a mixture. He has not shown the particular isomer, because a tartaric acid must have at least 3 diastereomers, because there are 2 chiral centers in it. So therefore, your calcium salt of tartaric acid is water soluble. There should be the solubility difference in all the 3 calcium salts. So there should be 1 calcium salt that is water soluble, not all. This is one suggestion. The other suggestion is regarding the synergism and other factors that how come a other compound, whether it is a drug or vice versa, without having the stereoisomers, increases the rate of the reaction or that particular biological action. So my purpose is to suggest that in case it increases the rate of reaction, in what might be happening, that it might be increasing the binding of that drug to that particular. Certainly in those papers, those persons, they might have missing this point. They have just said that it is 1.3 times or it is 100 times or it is 10 times. But my suggestion to Professor Kapoor is that one should find out whether the binding is 1.3 times, 10 times, or 100 times. Increases because of the additives. So with these suggestions, I would like to thank Professor Kapoor a lot for his very interesting talk.
Thank you, Professor Tejveer Singh. His suggestion is well taken. He has given a hint of very good suggestion that a tartaric acid occurs in L form, D form, and a meso form. There are 3 diastereoisomers, and all the 3 must be having the different solubilities of their respective calcium salt. This is a very intelligent and a question which only a research-minded person can put to us. It is certainly a issue which should be, studied in the laboratories. One can take the problem. Even our own institute have the D-tartaric acid, L-tartaric acid, and mesotartaric acid. All these are available in the market, right? And then you make the calcium salts of all these 3 diastereoisomer and see its solubility. It will be a very good contribution, and a paper can be published in any journal of repute. So thank you, Dr. Tejveer Singh, for its worthy suggestion. And regarding potentiation or the antagonism, the only study so far made is on the enzymes. The drug which might be suppressing the metabolic activity of the enzymes will certainly potentiate the action of the other drug. Or if it suppresses, if it induces those enzymes, certainly the action of the other component would be lowered down. Both these suggestions are well taken. So thank you very much.
Thank you, Kapoor sir. There is one question from attendees. Attendees, can you raise your hand? Priyanka Kondal. Priyanka. Kritika, can you ask your question?
Yes, ma'am. Good afternoon, sir.
Good afternoon.
Thank you for sharing your valuable information regarding herbal drug interaction.
Thank you.
So my question is, if we use herbal drug formulation which contain extract of any part of plant with allopathic chemical moiety, can the interaction will be same as if we consume the single chemical quotient from the plant with same chemical moiety?
Can you expand a little bit with some example of your question? I mean, you say if there is a herbal drug, the constituent will be present either in the fruits, in the seeds, in the bark, or leaves, right?
Sir, I think she just want to know that, for example, if we consume curcumin with, suppose paracetamol, or either we take whole turmeric with paracetamol, then is there any difference between the drug interaction between the two?
Certainly, the dosage will be different. When you are taking source of the drug as turmeric, naturally the interaction will be with regard to the large quantities, right? If you are taking the curcumin, which is the extract, it will be lower down consistency. Are you satisfied?
Are you satisfied, Ms. Kritika?
Yes, ma'am. Thank you, sir.
You can discuss more when I am in the school, right?
Okay, sir. Okay.
I appreciate your inquisitiveness. Thank you.
Sir. Thank you, Kapoor sir.
Thank you.
Thank you to all panelists. Our next speaker for this session is Dr. Hussain. Dr. Hussain. Dr. Hussain has more than 20 years of experience in pharmaceutical industry as well as academia. He has published several books and research articles in the field of pharmaceutical sciences. Dr. Hussain has invited as a guest lecturer to USA and Iran to present their research papers. He holds life membership in Indian Pharmaceutical Graduate Association, approved manufacturing chemist and pharmacist by Government of India, attains life membership in Iranian Pharmacy Association, approved manufacturing chemist and pharmacist by Government of Iran. We welcome you, Dr. Hussain. Over to Dr. Hussain. Dr. Hussain, are you with us? Dr. Hussain. Dr. Hussain, have you joined with us?
Hello, hello, hello.
Hello, Dr. Hussain. You are audible.
Yes, yes. I'm audible.
Yes, sir.
Okay, okay. I will start. Hello and good afternoon, respected professors, audience, and viewer. Thank you very much, organizer, for inviting me on a topic of drug delivery in international conference. I would like to present in paper in related to telemedicine, a special focus on telepharmacy. Telemedicine was started long back in 1904 by electrocardiograms who published a paper on a telecardiogram. Modern telemedicine has a start from 1964 in US between two medical centers. And telepharmacy in India also is a start from the year 2020, means long gap. Telemedicine is a platform help streamline drug development process and clinical trials. It helps drug discovery in three ways. One is in clinical trial, another is in monitoring, another is monitoring, another is consulting with the patient through the pharmacist. Pharmacist is the first and the last one who deliver the drugs to patients who are suffering from any type of illness. Pharmacists play a vital role in a healthcare system of a country and in a global healthcare system. For example, during the COVID-19, it was the pharmacist who was in the first line to combat this unwanted virus epidemic disease and help patient and doctors to find out the best possible prevention and combination of medicines to cure COVID virus and subsequent infection caused by these viruses and secondary infection which are common with COVID infections. It used to facilitate drug delivery to the patient during the COVID epidemic viruses. Pharmacists are a best source. Pharmacist is the best source of information related to the drug and discovery of new drug in the society. Pharmacists can play an important role as a pharmacovigilance through the telemedicine, which I should call, now as a pharmacist, I should call it telepharmacy. Pharmacovigilance can be done very accurate and very cheap regarding monitoring dosages of a drug given to the patient in remote area or in chronic patient or in unable patient who are due to age problem cannot come and visit the pharmacist. Information with the pharmacy will be a vital information and vital platform for drug discovery, because in a modern pharmacy, as I have worked with Shiraz Medical University in Iran, we have a platform in pharmacy, and the pharmacy has a very crucial role in registering side effects, dosage, adverse effects, and drug interaction, and all information related to the drugs, and all information related to the patient. Means it's a bank of information which can be used by the researcher, by the government authority, and by the monitoring authority for checking the pharmacy activity. Pharmacy can help for immediate recall of malfunctioning drugs or drugs with the toxicity after marketing also. Because it is the pharmacy who is in the direct contact with the patient and with the industry. In one side, he has to deliver the drug to the patient. In another side, he has to be responsible to question asking by the authority regarding to the drugs and regarding to the efficacy of drugs after marketing also. If something wrong with the formulation or with the drugs, it is the duty of pharmacy who can immediately recall and send back the drugs, the mal-formulating drugs to the manufacturer. Telemedicine, in another way, helps industry also for better service as a selling drugs to the different healthcare systems, especially to pharmacy, to hospital, because online they can put the list of medicine available with the factory, and then the pharmacy will put the order and they will receive it in the pharmacy. In case before coming to the pharmacy can change, pharmacists can check the list of the drugs which he has ordered and check before coming to the pharmacy. And in this case, in this way, there is a cost reduction in transport and in hiring an employer as a sales person to visit the pharmacy in person. The system in the modern pharmacy is like that the patient will go to the doctors or online with the help of telemedicine, will contact the doctor. Doctor will contact the patient and give the time for consultation online. And after that, doctor will send the prescription through the telepharmacy app to the patient, and the patient has the right to choose any pharmacy at his nearest, as nearest to him. Patient only needs to contact the pharmacy online by telephone or by other means and just give his national identity number. With this national identity number, pharmacy is able to track the prescription which is given by the doctor to the patient, and the pharmacy will consult first the doctor and second patient, and after that, when he get the OPD from the doctor as well as from the patient, then he will dispense the prescription. Then he will send the prescription to the patient, and no need for patient to come and waste his time and money to collect his medicine. And all the information will be registered in the file of patient. Each patient all over the country, they have one unique medical file. This file contains all information regarding to the health of a patient from the childhood, when he born in a hospital, till the last day of his living in this world. That's why it is very easy to monitor the status, health status of the patients, and also it is a valuable source for a researcher, for the government organizer, for the industry to see the number of the patients for particular diseases, for a particular drug. For example, how many paracetamol is used per year in a special locality or by a patient or all over the country. All these pharmacies are linked to the Ministry of Health as well as the health insurance company. All the people are insured, they have insurance, health insurance, and therefore it's also pharmacy duty and helping insurance company not to overuse the drugs or waste the drug. For example, if a doctor is given 7 days antibiotics to a patient daily 3 times, the patient is entitled to get 21 amoxicillin as antibiotics for 7 days. Patient is not allowed to get any medicine without presence of the pharmacy in the pharmacy. Pharmacist has to be present in the pharmacy, and they are controlling the pharmacy. And pharmacy first will consult the patient. If the patient comes to the pharmacy, the pharmacist first consult the patient, will ask the patient what's his illness. Then if he cannot cure the patient or give the right medicine, then pharmacist's duty to refer to the nearest doctor. Means each and every items, every drug should be registered, and to whom it is given and for what reason it has been given to the patient, and all the items which pharmacists are received from the pharmacy industry or from a manufacturer, he has to register in the pharmacy database system. And this database system is used by the insurance company, by the government, and by the university for evaluation of the pharmacist activity. And online university or regulating authority can monitor whether the pharmacist is available in the pharmacy or not, or is there any malpracticing by the pharmacist or by any staff member in a pharmacy. That is connected online to the university and to authority agencies within the ministry. Then modern pharmacy can help the researcher in providing the vital information and doing the clinical trial of the drug with accurate and at a very, very reasonable price online. It is possible to provide to the authority online as well as in a store and forward telemedicine. This telemedicine is also acts as an accountant to pharmacists and help the pharmacist to have a correct idea about the availability of each and every medicine or item in his pharmacies and check the expiry dates and also daily pricing if there is a change in the price of the medicine, particular medicine or items in online on daily basis, it will be up to date. During the COVID viruses, pharmacists has played a crucial role in Iran, despite being sanctioned by the European and American in the field of providing to us medicines. But we pharmacists overcome this crisis also by using the whatever resources is available in our market. For example, I was working in a pharmacy, in my own pharmacy in a medical complex with daily visiting about 200 to 300 persons suffering from COVID viruses. I with the hospital which were connected with my pharmacy and with the doctors in the medical complex, we came to know that we have to find some alternative to cure the patient also. And through the telemedicines, we search different resources, different doctors all over the world. And fortunately, for example, we didn't at all give remdesivir injection to any patients. We replace it with a drug, brand name is PowerFit. It is a herbal drugs manufactured by Holistica, France, and is from the shiitake mushrooms. Shiitake mushroom, it is rich in HSCA and proteins, vitamins, minerals, which is HSCA is an active hexose correlated compound. It is alpha glucan compounds, and it is also have a good anti-cancer activity also, as made in Japan, but we manufacture it locally in Iran. Also, we cultivate and manufacture it locally in Iran, and we replaced the drug remdesivir, which in practical work did not give any benefit to the patient. Moreover, those vaccines which have been launched during the COVID viruses, they just sold their vaccines and they gained the profit. But these vaccines which have been manufactured in hurry, they didn't consider their side effect and after marketing effect. Nobody is responsible. But I face many patients suffering by receiving this vaccination. Especially hair fall is a very common. Then impotency also, heart problem, and kidney problem, and many other problem side effects, which only the pharmacist knows what they have done this people to the health of the patient during this COVID epidemic virus infections. And fortunately, with a minimum facilities, we just give the advice to the patients to give precaution, to take precaution, prevention. We told them that prevention is better than treatment at this stage, because treatment is very limited, and whatever is available, it is just they are using you as a animal, just to animal in a laboratory to test those vaccine on you. And it was really true. We lost many people, over 700 to 800 and more, due to receiving this vaccine also. But nobody disclosed it, except it will remain in the heart of pharmacists, who are the empathy, have a true empathy, as Professor as mentioned, as a Professor Kha and another professor, he mentioned that there should be fear of God in our hearts, and it is with pharmacists. Pharmacists, through the telemedicine, can do three more important functions also, service they can provide to the patient. One is consulting that I mentioned. Another is monitoring and monitoring. Monitoring means dosage for side effect and all. We will be in contact with the pharmacists. Pharmacists, they are all the patients, they have our contact number. We are on call 24 hours in Iran. They even know I'm in India also, they are calling for some medicines and for some consultation they are regarding to their diseases. And other is monitoring. We guide them how to take the medicine, how to take the precautions, and how to protect themselves from the different diseases. For example, we give the good diet to them also. We also serve as a dietitian to the patient also, because they expect from us. First, the patient come to the pharmacy, and then they go to other healthcare providing agencies. With the help of artificial intelligence also, pharmacists nowadays, they succeed in synthesizing new type of antibiotics which are not showing resistance to bacteria. Therefore, based on store and forward information, we target the patient to the researcher for clinical trials of a drug. We tell the researcher, we give the information through the health ministry to the researcher that, okay, in this particular season, in this particular region of the country, this disease is epidemic, and in this particular time, and these particular people in genetically, for example, those people who are in the south, they are suffering from such a disease. And this all information will be provided daily online or in a store and forward mode to the concerned authorities. And another main duty of the pharmacist is to provide the consulting to the patient. In modern pharmacy, we have a separate room for consulting. When the patient arrives in the pharmacies, it is the right of patient to consult pharmacist before prescribing before they dispense their prescriptions. And one of the most and very effective rule of this telepharmacy is that we avoid mistake by bad handwriting of the physician, because those physicians, especially super specialists or other physician, other healthcare system, their handwriting are not good, or they used to do monopoly and write in such a way that only the patient should go to a particular pharmacy for dispensing their prescription. But now with this system of telemedicine, telepharmacy, this problem also has been eradicated, has been deleted, and now patient with full confidence will get the best healthcare through the pharmacist by using this telemedicine app, which each and every patient in Iran, they have this facility to contact pharmacists, to contact doctors, and they get best facility, best healthcare at the nearest place to his residence. Then it's during the COVID viruses, I said it was an opportunity for us, for a pharmacist in Iran, to develop this system of providing medicine and delivering the healthcare to the patients in a remote area also. And moreover, patients are very happy, and they are this type of telemedicine or telepharmacy brought more and more confidence between the patient and the pharmacist. And regarding to drug design, researcher can use this information through the pharmacist for any diseases. For example, if there is some problem related to formulation, we have the information, what is the problem, what's the side effect, why the patient complain, what is the side effect, if it is related to dosage form. For example, if the patient cannot take the medicine, for example, we had to prepare omeprazole suspension for the kids who are suffering from a stomach problem, or we have to also prepare the cosmetic products and in the pharmacy as per the patient's desire, because some patients they are allergic to some ingredients, because those who are available in market, some patients they are allergic to excipient also. We prepare the medicine for a few days, means for 1 week, then again another week we will repeat it. We just do in a smaller scale preparation of drugs in pharmacy. These facilities are with all the pharmacists in Iran, in all the pharmacy shop. We do this type of work, and we extend this service to the patients, because some combination may not be available in a cosmetic or in some drugs. For example, some patient cannot take a medicine, for example, with such of, especially kids with paracetamol, they want a special taste, because they come to the pharmacist and they ask about, for example, they want with the tutti frutti taste or good flavor, so that their kids can use it. We in the pharmacy, we do extend these services also in dispensing the prescription which are coming from the skin specialist. Also, we do the preparation, we manufacture the preparation, and we also do the vaccination also. If the patient come, and if we teach them how to use also in presence or offline and online, we give to the patient the method of using the modern preparation. For example, this insulin injection in the pen form. Many patients, they are, especially old ages, they don't know how to use it. As a duty of pharmacists, online or in person, the patient comes to the pharmacist, and pharmacy, it is a duty of pharmacy to teach them how to use, especially for other drugs also, aerosol in the form of encapsulated in the form of powder, because before this telemedicine, telepharmacy in modern pharmacy, the patient didn't know how to use, and the doctor are so busy with their business, and physician are so busy with collecting the money, and they don't have a time also to spend on the patient. It was on the shoulder of the pharmacist to provide even the method of administration to the patient. And this is as also we are doing nowadays, and patients are very pleased. They are very happy, and I'm also thankful for this organizer, all the professors who are in the panel. Thank you very much and give me this chance. And I'm ready to extend my all services to all the people, not in my country, but all over the world. And I will be thankful to all of you again and again. Thanks a lot.
Thank you, Dr. Hossain, for delivering such a beautiful talk. I can say practical talk, or means practice that used to follow in Iran. So the best way to keep the feedback mechanism updated by correcting the procedure of dispensing or doing the right practice of pharmacy. That data is helpful in designing or development of new molecule. So that kind of system is going to improve our drug discovery process, and we can accelerate the drug discovery portion.
Thank you. Thank you, Professor Mishra, for your comment. Thank you very much. Any question? I'm ready.
Now over to Madam Salni.
Thank you, Dr. Hussain, for sharing your knowledge on telemedicine.
Thank you very much, Madam. Thank you for
extending
your cooperation to organize such a nice and very informative conference. Thank you, Madam.
Thank you. Thank you so much, sir. Now it's time to introduce our next speaker, Dr. Jitendra Madan. Dr. Jitendra Madan is Associate Professor and Head Department of Pharmaceutics, Sniper, Hyderabad, India. He obtained his PhD from Bundelkhand University, Jhansi, and also worked as postdoctoral fellow in Georgia State University, Atlanta, USA in cancer nanobiology. Currently, Dr. Madan is associated with Department of Pharmaceutics, Sniper, Hyderabad, and continuing academic research in the area of topical drug delivery system, vaccine delivery, and cancer nanobiology. We welcome you, Dr. Madan. Over to Dr. Madan. Dr. Madan. Mishra sir, kindly unmute yourself. Sorry, mute yourself. Unmute kuch. Dr. Madan.
Hello.
Hello, Dr. Madan. You are audible.
Good afternoon, ma'am. Yeah, good afternoon to everyone.
Good afternoon, sir.
Should I share the presentation, ma'am?
Yes, sir. Please share your presentation.
Ma'am, is it visible?
Yes, sir. It is. Maximize it.
My presentation is the critical role of computational tools in drug delivery. Ma'am, am I audible?
Yes, sir. You are audible.
Okay. So introduction, drug design and development is a complex, costly process which involves the identifying and developing new chemical compounds that can be used as drugs to treat disease. So the basic aim is to aware about the role of in silico drug designs in drug delivery. So traditional drug design methods are time consuming and labor intensive. So that's why modern drug discovery approaches such as in silico methods have emerged as a way to limit the cost, time and manpower required in the drug discovery process and hence overcome the drawbacks of traditional approaches. So here I will present the case studies in which we have used the in silico methods, how in silico methods can be helpful in drug delivery techniques. In silico drug design, also commonly known as computer-aided drug design, involves using computational techniques and models to identify drug-like molecules using bioinformatic tools. In silico method analyze and predict the biological activity of the potential drug candidates and also predict their physicochemical properties. So same thing we have also applied in our case studies. So basically we can classify them as structure-based drug design or ligand-based drug design. If there are three-dimensional structure of the protein is known, then structure-based drug design may be preferred approach, which allows the design of molecules that fit precisely into the protein binding site. On the other hand, the ligand-based drug design, if the protein structure is known, then ligand can be docked with the protein molecule and that have the similar properties to known ligands that bind to the protein. So the basic applications helpful in drug delivery, which saves the time and resources required for drug discovery, optimize the structure of potential drug candidates to improve their binding affinity, safety, efficacy, and other pharmacological properties, design compounds based on the chemical structure and properties of known ligands that bind to a target protein. It also helps in predict the binding mode of a drug candidate to the target protein. So this is our case study 1, where we have identified the potential lipid molecule which can be helpful in the topical drug delivery of our molecule, that is the meloxicam. So in this case, we have studied about the impact of different lipids on the performance of lipid nanoparticles loaded gel. So in this case, meloxicam loaded lipid nanoparticle, initially meloxicam was loaded in the lipid nanoparticles, and the lipid nanoparticles were optimized on the basis of the functional group, which can be helpful in the maximum permeation of the molecule inside the skin. So to optimize the structures, we have used the in silico docking tools to and in silico docking tool may guide to screen the lipid core for augmenting the penetration of therapeutic entity into the skin. So we have used the stratum corneum lipid model. So we have used the ceramides, which is the major component of the skin lipids, free fatty acids, and cholesterol. Hence, the in silico docking studies are performed to construct the lipid layer of the skin that mimics the human skin. So these are the different lipids with their chemical structures. So we had selected the lipids based on the lipid chain length and the functional group. So palmitic acid having the C16 carbon chain length, stearic acid C18, arachidic acid C20, behenic acid C22, and styramine having the functional group amine group, stearyl alcohol with the hydroxyl group, stearic acid with carboxylic acid, and astatine with triglyceride. So we have used in silico organic studies. So molecular modeling studies were performed with the aid of master Schrödinger suite to simulate the interaction between the lipid core and ceramide. So initially interaction between the lipid core and ceramide were carried out. Ceramide was extracted from the protein crystal coordinates with PDB ID 2E3R from the protein data bank. Energy minimization using the OPLS3 force field of the LickPrep module from the Schrödinger suite was carried out. And later receptor grid generation was carried out. Ceramide was docked using the lipids docking light. The docking score represents the interaction of skin lipids and lipids. To formulate the dosage form, initially we have carried out the solubility of the drug in different lipids, as we have mentioned previously. So a fixed amount of the lipid is weighed, which is heated to the melting point above the 10 degrees Celsius. So in this way, we have initially we have carried out the solubility of meloxicam in different lipids by using HPLC method. So different process parameters like screening of surfactant. So we have taken different surfactants, twin 20, twin 80, fluoronic F60, fluoronic F127 based on their HLP values. And later we optimized the homogenization speed so that we could formulate the lipid nanoparticles with minimum size under some micron size. And sonication time was also optimized. In addition, surfactant concentration screening was also carried out. So lipid nanoparticles were prepared by homogenization method. Initially, the meloxicam was mixed with the chloroform, which is an organic solvent in which all the lipids are soluble. And then they are heated to up to 70 degrees Celsius near to the melting point of the lipid. Later, they were homogenized to formulate the submicron size particles and sonicated with the sonicate. So these are the compositions in which we have used different concentrations of the lipids having different chain lengths and different functional groups. And surfactants were added to make a solution of different with the different HLB values. The meloxicam lipids loaded solid lipid nanoparticles were characterized for particle size, polydispersity index, Gita potential, and tap-in efficiency, and unloading capacity, in addition to the surface topography by transmission electron microscope. After synthesizing the lipid nanoparticles of meloxicam, they were amalgamated with the gel, and the gel was characterized for color, homogeneity, grittiness, pH, so that it should simulate the skin pH, rheology, using a rheometer, texture properties with a texture analyzer, and dosier distribution in the skin was carried out with by using the confocal laser scanning microscope, using cuminant dye as a fluorescent dye.
So, excuse me, sir. Your slide is on the first page only, so you are requested to match your talk to your slides.
Okay, I reshare the slide. Now they are moving.
Yes, sir. Now it's good. I think you put it on the slide show mode. That is going to be easier.
Okay. So we have carried out the characterization of the optimized meloxicam loaded solid lipid nanoparticle gel. So texture properties we have carried out with the texture analyzer. Later, dermal distribution in the skin was carried out by using Combinant 6 dye, which is a fluorescent dye, using confocal laser scanning microscope. The in vivo study was conducted in the monosodium iodoacetate induced osteoarthritis model in rats. So these are the different groups in which we have given the meloxicam oral dispersion, meloxicam intravenously, meloxicam solid lipid nanoparticle gel. In addition, we have also taken the standard, that is the etoricoxib and daclofenac marketed gels, and comparison was made with the disease positive control group. We have measured the different parameters to showcase the efficacy of our gel. So in this case, we have measured the knee joint thickness, thermal hyperalgesia, and motor incoordination tests. In addition, last we have also carried out the X-ray evolution study of our to showcase the potential of our gel, which was optimized using in silico docking techniques. And also we have carried out the histopathological studies using histotoluidine blue dye. ELISA testing was performed to measure the cytokines, pro-inflammatory cytokines, that is interleukin 1 alpha and beta and TNF alpha. So this is the results, and we have seen that we have carried out the in silico docking with palmitic acid, stearic acid, arachidic acid, behenic acid, stearyl alcohol, and stearylamine with the ceramide. A ceramide is a skin lipid. So interaction between the ceramide and our selected lipids will enable us to optimize the particular chemical structure of a lipid, which will show maximum higher penetration ability. So this is the docking score we have measured. So we have seen that we have measured the different docking score and glide energy with the C18 to C20 lipid chain length, in addition to the different lipids having the different functional groups like amine, alcohol, acid, and triglycerides. So based on the docking score, lipid displays an affinity in the increasing order of stearic acid, palmitic acid, arachidic acid, and behenic acid. Ultimately, it is a parabolic relation. So C18 stearic acid had the highest binding affinity than the lipids with lower and higher chain length. So it is a parabolic relationship. Following this, lipid chain followed a parabolic relation with the binding affinity. And on the other hand, concerning the functional groups, alcohol, that is the stearyl alcohol, exhibited a high docking score, followed by amine and acidic group, that is the stearic acid. So we have also carried out the solubility study, as you mentioned earlier, by HPLC method. So based on the solubility study, stearyl amine, because of the charge, it has made the complex with the meloxicam and shows the higher solubility as compared to rest of the lipids. Stearylamine was taken as a lipid in optimizing the processing parameters. So tween 80 was chosen among different surfactants to scale up the solid lipid nanoparticles and produce the lower particle size with polydispersity index below 0.3%. So we have also optimized the homogenization speed. So lipid emulsion size was reduced from around 1 micrometer to 347 nanometer at 10,000 RPM with increase in the homogenization speed. Particle size was also reduced with 2% of the surfactant. After that, there would be no change. This indicated the optimal concentration of surfactant has reached to 2%. So with the 2.5% concentration of the surfactant, we obtained the minimum size 218 nanometer with polydispersed index of 0.259, which is good for the topical therapy. Hence, the optimization of the different lipids indicated that styramine was adopted for the preparation of solid lipid nanoparticles by hot homogenization method for all the lipids with 10,000 rpm for 5 minutes and sonication for 50 minutes were the optimized conditions. So this is a particle size which is measured by the Jita sizer in our institute. So this is about the Jita potential, that is the surface charge of all the solid lipid nanoparticles of meloxicam. So surface topography was measured by transmission electron microscopy, which indicated that the particles, there were particles in the size range of 150 to 250 nanometer. So this is about the entrapment efficiency and tug loading capacity. So sterile alcohol we have used given the maximum entrapment efficiency. Later we formulated the meloxicam loaded solid lipid nanoparticles amalgamated dermal gel, and we have used 3% chitosan. Chitosan is already approved by US FDA for the topical application. So 3% chitosan was selected for tailoring the SLS gel, and the assay was found to be 95% for meloxicam. So this is the skin permeation efficiency. So lipid with amine functional group, that is the styramine, showed highest permeation than hydroxyl group, linked sterile alcohol, carboxylic acid group, stearic acid, and triglycerides. So which is according to our in silico docking. So this was synchronizing to the in silico docking results. So in this way, sterile alcohol and sterile amine demonstrated high affinity towards the ceramides of the skin. So in this way, in silico docking techniques helped in optimization of the different among the different lipids, which saves our time. So sterile alcohol disclosed highest docking score in silico docking studies. Sterile amine exhibited highest penetration in ex vivo experiments, as well as it also shows the slow release, which is also helpful in formulating a depot effect. So this is about the organoleptic properties and textural analysis and visualization of dermal distribution of the skin. So this is about the rheology, and which is comparable to the marketed gel. So this is the textural analysis in which we can measure the work of shear, spreadability, gel strain, firmness, force of adhesion. This is all about the stability studies. We have carried out for 3 months at the room temperature. So this is the visualization of dermal distribution of the skin. We have seen that our cumulin loaded solid lipid nanoparticle gel of meloxicam shows higher permeation as compared to the fluorescent dye suspension through transfollicular pathway. So this is about the in vivo studies. In vivo studies indicated that meloxicam loaded solid lipid nanoparticle gel has reduced the thickness of the knee joint as compared to the marketed gel and positive control group effectively. Similar results were also obtained in our thermal hyperalgesia data. And this is about the motor and coordination test, in which we use the Rotaro test apparatus. So this is the X-ray evolution of the knee joint. So here we have seen that our meloxicam loaded solid lipid nanoparticle gel have shown the equal efficacy as compared to the marketed gel and meloxicam oral suspension. So pro-inflammatory cytokines, because osteoarthritis is allied with the induction of pro-inflammatory cytokines. So I measured the TNF alpha and IL-1 beta, and our formulation have effectively reduced both the cytokines as compared to marketed formulation and a positive control group. So this is about the histopathology. So our formulation has also offered the chondroprotective effect as compared to the meloxicam oral dispersion group and meloxicam intravenous injection. So we can confidently say that our formulation was also effective as compared to the oral treatment group as well as the marketed gels. So this is about the ORC score, which is also indicating the efficacy of our gel. So this is the publication from this research work, which is published in the Journal of Drug Delivery Science and Technology. Second case study is with the naringenin. We have also performed the in silico docking technique in this research work. So we have developed the naringenin, which is an antifungal drug, but it has weak antifungal activity. So we have complexed the naringenin with the silver nanoparticles, and then later this complex was fused with the chitosan gel for the treatment of cutaneous candidiasis. Cutaneous candidiasis is a fungal infection in the skin. Candida can be the primary cause of this skin disease and may arise secondary to other skin diseases like atopic dermatitis, psoriasis, or existing diaper dermatitis. According to WHO, an estimated 20-25% of the global population is affected with cutaneous fungal infections, including candidiasis. However, the limitations of the current therapy is the reoccurrence and resistance, as well as cytotoxic or hemolytic effect. On the other hand, pharmaceutically, lower skin permeation and shorter skin retention is also associated with the commercial formulations. So we have formulated the silver nanoparticles using naringenin as a capping agent by precipitation technique, in which silver nitrate was used as a template. Later, naringenin solution was added in a drop-wise fashion to synthesize the naringenin complex silver nanoparticles. So these are the different characterization parameters. So we have used UV visible spectrophotometer to characterize the synthesis of silver nanoparticles. And absorption was recorded in the range of 300 to 600 nanometer. In addition, we have measured the pH, particle size, surface topography by transmission electron microscopy, and assay of naringenin and silver with ICP-MS method. After formulating the naringenin cap silver nanoparticles, they were amalgamated with the topical dermal gel, and we used the Carbopol Ultras 10 NF, which was gifted by the Lubrizol Incorporation Limited. So these are the different characterization parameters. First, we evaluated for the organoleptic properties like color, feel upon application in terms of grittiness and phase separation. The pH was measured by Mettler Toledo pH meter. Rheology, in which we have measured the viscosity, and we have prepared the curve between the shear stress and shear rate. Texture analysis, and naringenin and silver content. In silico docking was performed. Molecular docking studies were performed in Mastro version 13 of the Schrödinger suit to unwind the binding affinity of naringenin and naringenin silver nanoparticles towards sterol 14 alpha D methylase which is an important enzyme responsible for the ergosterol synthesis in Candida albicans or any fungal microbe. Permeation efficiency was also predicted by in silico docking technique with ceramide. As we have discussed earlier, ceramide is an important lipid in the skin, which is a major barrier for the different therapeutics. So sterol 14-alpha-D methylase and ceramide was retrieved from the protein data bank. Using the Schrödinger suite, the protein was prepared for molecular docking. Energy minimization of protein using the OPLS-4 force field and converging the heavy atoms to 3 angstrom. The molecules were subjected to the Lick-Trap module. Receptor grid generation molecular docking studies were performed. The interaction between the ligand and the receptor was studied, and a 3D structure was generated using the Schrödinger suite. The gel was characterized for ex vivo skin permission, retention, and skin distribution analysis. So friend diffusion cell was used for ex vivo skin permission study and retention study. And the skin distribution was also analyzed by confocal laser scanning microscope using cumin 6 dye. In vitro antifungal study was performed against Candida strains with MTCC number 183 and for Candida glabrata with MTCC number 3019. Both the strains were grown in the Sabot dextrose agar medium, and turbidity was adjusted using the McFarland standard. So in this case, we have measured the MIC 50 value, that is the minimum inhibitory concentration, and comparison was made with the commercial 2% weight by weight miconazole gel, miconazole cream. So this is the calibration curve we made with the ICP-MS method for the silver. So here we are showing the characterization of silver nanoparticles with UV visible spectrophotometry. So UV visible spectrophotometry was employed to notice the color change that occurs due to surface plasmon resonance. The peak was obtained at 420 nanometer post scanning between 300 to 600 nanometer. The silver nanoparticle exhibit conduction band and valency band close to each other, which leads to the free movement of the electrons called SPR, or surface plasmon resonance. So this is about the particle size, zeta potential, and surface topography. So all the silver nanoparticles were showing little bit aggregation, but they were in the range of below 50 nanometer. So assay for naringenin and silver was carried out with HPLC and ICP-MS method respectively, and they were in the range as prescribed in the Indian Pharmacopoeia. So this is about the rheology and texture analysis. So this is about the in silico docking. So molecular docking of sterol 14-alpha-D methylase with PDB ID 5FSA at the active site with different ligands was performed. Similarly, we have also performed the in silico docking of miconazole at the active site of the sterol 14-alpha-D methylase enzyme. So sterol 14-alpha-D methylase, as we have discussed earlier, it is an important enzyme for all the azole groups based drugs. So this is a docking score which we have measured between the interactions, docking score interactions of naringenin, silver naringenin complex, and miconazole at the active site of sterol 14-alpha-D methylase with PDB ID 5FSA. So we can infer that the interaction, the binding mode at the active site of sterol 14-alpha-D methylase were similar for naringenin silver nanoparticles and miconazole. Consequently, this strongly suggested that naringenin silver nanoparticles may exhibit potential antifungal activity. So these are the optimized structures of molecular docking of naringenin with ceramide and silver naringenin complex with ceramide. So this is the docking score of naringenin and naringin silver complex with ceramide. The naringenin interacted with ceramide via 2-point hydrogen bond interaction through the 7-hydroxy group with a distance of 2.2 and 1.91 angstrom. However, complexation of naringenin with silver ions induced the conformation alterations, which subsequently led to the interaction of the 4-hydroxy group with a distance of 2.07 angstrom. Additionally, it also showed a pi-pi stacking interaction with the ceramide. So this is about the ex vivo permission analysis of naringin silver nanoparticles loaded gel, in which our synthesized gel, that is naringin silver nanoparticles loaded gel, showed higher permission as compared to the naringin gel only. So this is about the in vitro antifungal activity. So it indicated that our gel was close to the commercial marketed gel in terms of MIC 50 value. So this is the basic interaction, how our silver nanoparticles, naringin complex, interacted with the candida albicans, and they have affected the multiple targets, and in this way they induce the synergistic activity. So this is the publication we obtained from the this research work. So this is the case study 3, in which we explored the synergistic effect of COX-2 inhibitors and hyaluronic acid in osteoarthritis therapy through in silico docking studies. Hyaluronic acid is important, which acts as a shock absorber in osteoarthritis. So it is already working in clinic, and it is induced through intra-articular route in case of osteoarthritis. This study focuses on the suppression of human p38 MAP kinase and junction and terminal kinase in the signaling pathway of osteoarthritis. So both the enzymes are responsible for the production of pro-inflammatory cytokines and MMPs, which are the important targets in cartilage protection. So in silico studies were conducted to analyze the interaction of COX-2 inhibitors and hyaluronic acid with human p38 MAP kinase and junction and terminal kinase, which exhibited moderate to high binding affinity of HA towards human p38 MAP kinase and junction 3 terminal kinase when compared to other selective COX-2 inhibitors. Hyaluronic acid showed a high binding affinity with human p38 MAP kinase and junction and terminal kinase. Alongside selective COX-2 inhibitors showed a high binding affinity with both human p38 MAP kinase and junction terminal kinase. Thus, the docking results concludes that a combined formulation of COX-2 inhibitors with high molecular weight HA may provide a synergistic effect in the management of OA, that is osteoarthritis. So this is the binding score with human p38 MAP kinase and terminal kinase, and both the enzymes are responsible for the production of pro-inflammatory cytokines and MMPs. So we have carried out the study for the celecoxib, lumaricoxib, hyaluronic acid, meloxicam, nimesulide, pericoxib, rofecoxib, valdecoxib. So we have also got this publication. So idea of developing novel therapeutic strategies for OA treatment has gained considerable attention along with discovery of new molecular targets. So in this review, we have summarized that the viscosupplementation and chondroprotective mechanism of hyaluronic acid and its synergistic action with the COX-2 inhibitors may helpful in designing of new therapies for the management of osteoarthritis. So a few case-to-case studies have revealed that hyaluronic acid along with COX-2 inhibitors, that is the selective COX-2 inhibitors, has a disease-modifying impact. However, only few clinical studies have been reported to date. So we have a lot of opportunities in this case. Hence, we presume that unboxing of the signaling mechanisms involved in hyaluronic acid anti-inflammatory action may lead to the development of innovative techniques which can prevent degradation, degeneration of articular cartilage as well as inflammatory arthritis. Hence, this research work And this research work highlighted the potential beneficial signaling cascade of crosstalk between the hyaluronic acid with COX-2 inhibitors and opened new avenues for drug discovery and drug delivery in the management of osteoarthritis. So these are the publications which we have received from using in silico docking tools in development of drug delivery systems. Thank you very much.
Thank you, Dr. Jitendra Madan, for sharing your expertise in critical role of computational technique in development of drug delivery system. Thank you, sir. We have panelists Dr. Tejveer Singh, Professor Dr. Ravneesh Mishra, Ms. Shushma Sharma, Professor Dr. V.K. Kapoor, sir. Over to Tejveer.
Good afternoon, sir. Good afternoon to everyone.
Dr. Madan, you have mentioned several things, but when you talk about the COX-2 and
yes, sir,
you have studied the interaction with silico and non-silico, whatever. So what did you find out really?
Sir, in osteoarthritis, sir, there are two approaches. First, we can use hyaluronic acid as a viscose supplement, and COX-2 inhibitors can be used to prevent the inflammation.
So you could see it with binding that the inflammation has been stopped?
Sir, we did not perform any experiment by taking these two molecules. Only we have performed the in silico docking techniques. But in future, we will also go for it, sir.
Okay, you did the calculations only.
Yes, sir. We performed only in silico docking, sir. But in case of meloxicam, we have also performed that experimental part. And we have reported the good results with meloxicam loaded solid lipid nanoparticles in which we have optimized the lipids with different chain lengths.
What about the standard drug?
Sir, we have taken the meloxicam, sir.
So how much percentage of inflammation could be stopped?
Yes, sir. Inflammation was reduced as compared to the meloxicam intravenous and meloxicam oral dispersion group. Sir.
No, I know. I want to know the percentage.
Sir, it was almost equal, sir.
It was.
But we can only claim that we have reduced the opportunities for the gastric side effects, sir. By applying the topical therapy.
Okay. Thank you, Dr. Madan.
Thank you, sir. Thank you very much.
I would have appreciated if you had drawn the structure of naringenin and its complex with silver, so that we could see which are the coordinating atoms.
So this is the structure of naringin silver complex, sir.
Yes, all right. It is the
so it interacted with basically the OH and that
model.
carbonyl.
I was looking at the structural drawing, and the structure of the complex with silver. I mean, in this, we cannot make out which are the atoms involved in the coordination.
Yes, sir, I can understand, sir.
It would have added additional information. It doesn't matter.
Sir, sure. Yes.
It does not lower
yes,
the merit of your publication.
Yes, sir. Yes.
Thank you, sir, for delivering such a informative talk and its role in the drug development. So, can you elaborate about this NRG silver nanoparticles, or do you have done any comparative studies with the other polymers?
No. In this case, we have formulated the naringenin complex with the silver nanoparticles. Silver is itself is an antimicrobial agent, a strong antimicrobial agent. So our aim was to increase the antimicrobial activity, that is the antifungal activity of naringenin, which is a weak antifungal agent.
So, is this method is your green synthesis method?
It is near to yeah, we can say it is the green synthesis method, because naringenin here is working as a capping agent.
Okay. Thank you so much, sir. Over to Madam Priyanka.
Thank you, sir. Thank you, Ravneesh, sir. Thank you, Dr. Jatinder Madan, for sharing your knowledge with us. Now, we are moving to our next speaker, Dr. Shrikant Vijirao Joshi. Sir, are you there? Shrikant, sir? Am I audible?
Hello.
Hello.
Am I audible?
Yes, sir.
Yeah.
Okay, sir. Welcome, sir. Our next speaker, Shrikant Vijirao Joshi, sir. He completed his B Pharm from SN Institute of Pharmacy, Pusad, Maharashtra, M Pharm from RSC Patel Institute of Pharmaceutical Education and Research, Shirpur, Maharashtra, and PhD from Veer Narmad, South Gujarat University. Currently working as Associate Professor at Maliba Pharmacy College, having total teaching and research experience of 17 years, 5 months. He published 2 books, 4 books chapter, and 36 research articles in peer-reviewed journal. He guided 28 students for MPharm, and 1 student completed PhD and 1 PhD ongoing under his guidance. Research interests are in silico drug discovery, repurposing of drugs, anti-cancer study, cardiovascular study, traditional and herbal medicine, etc. He had research skill and studies handled in silico drug discovery software like Marvin Sketches, ChemDraw, ChemSketch for drawing the chemical structure. He had publication regarding anti-cancer study, cardiovascular study, toxicity study. He handled zebrafish for various CNS and CNS studies. Thank you, sir, and welcome. Over to you.
Thank you. Thanks a lot for the nice introduction. I would like to thank, first of all, Dr. Ravneesh Mishra, sir, for inviting me for this specific talk. I would like to share my screen. Hope my screen is visible.
Yes, sir, visible.
And first slide is there on the screen.
Yes, sir, it is.
First slide is visible.
Yes, sir, visible, sir.
Yeah. So first of all, I will like to thank the management and Dr. Ravinesh sir. This is what I'm presenting today is my journey, how I learned this in silico drug discovery and in silico techniques, tools in drug discovery and development, and whatever studies we have done, we have completed in this specific our institution. I belong to Maliba Pharmacy College, Uka Tarsadia University, which is situated in Bardoli, Gujarat. And I will like to throw some light on this in silico tools techniques. What I found in some of the lectures which were delivered in some of previous such conferences and seminars, that the lacking part is how these all tools these are being used, because most of the times presenters or researchers, they show their work, how they have done it and all the things, but how to use these tools, it is something like which is missing from there. And most of the researchers, what I feel is most of the researchers and our students, mostly I'm associate professor over here, so I deal with various students over here, like post-graduation students and graduate students, and they are lacking this expertise in handling these various tools and techniques. So rather going behind this, presenting my work and how I got the results and what results I got, and all this can be available through my various research articles. So I'm not going to specifically target on that, but I will like to target on various tools which can be easily used and interestingly, which are freely available, or which we can say these are available free of cost, or freewares, or we can say available for academic institutions so as to use it. So starting with, can say, first thing, that is drug discovery and development. All of us know that drug discovery, it is nothing but finding new chemicals which could act as a, which could have pharmacological effects and required pharmacological effects, and we need to develop it into drug. So all through this process, we can go through this in silico tools. We can use these in silico tools so as to find out newer chemicals and so as to develop them as a drug. Silico word, it can be from silicon chips which are used in computers. So these are nothing but various computational tools or softwares which are available, and these are used for discovery of the drug. What I found, as I told you, my objective behind presenting this is to help our researchers or to help our students. So what I found, the challenges which are there in front of our researchers in Bharat, that is, I don't think it is only the condition of Bharat, because we are now 3 trillion economy, and we are moving towards 5 trillion economy, becoming, we will become 5 trillion economy in future. And in such condition also, we are still lacking in some points regarding research. And I feel this is the condition in most of the underdeveloped countries or developing countries. So there is lack of scientific training in the methodology of research, like how to perform the specific activity, or how to perform the specific research. So the scientific training, it may be because of lacking of sufficient time for the researchers to train others, or if it is possible, it can be possible through such type of workshop or seminars which are being conducted. And one-to-one interaction, it may not be possible in various research settings. Another thing is, it is the research process whole, it is a slow and time consuming one. So it is like slow and steady races the win the race, like that only, that research, it can only be done with this. We believe in that slow and time consuming process it is. And it is difficult to getting grants and funds in Bharat or in India. The reason is, we know many such applications are these funding bodies, they are getting many such applications, and they need to provide limited funds. So difficulty in getting the grants and funds. So planning their research, it will become difficult for the researchers. Lack of active networking and collaborations. What I found is, and what my experience says is, most of these researchers who are working over here,
they are like they are expert in their own domain, but they are lacking knowledge in some other domains, like in bioinformatics. I am basically a pharmacologist, so I may have knowledge in pharmacology, but the thing is, I am lacking the knowledge in bioinformatics. So I can collaborate with some scientists from the background, and this is what lacking. So lack of active networking and collaborations. And important thing is, in most of the places there is lacking of physical facilities, and this physical facility lacking, it may lead to restriction in the research. Our students, they are also facing various issues, like generation of the concept for any research. We know that from experience we learned this, that generating a concept, it is something Herculean task, and that too innovative work if we need to perform, then it will be difficult for students on their level, like UG students or post graduation students. Generation of the concept, it is a tough task for them. Guidance which is available, I will not like to criticize any of the guide, but guidance which is available, it may be it may not be sufficient to the children who are doing this various studies. Then facilities which are available, so again, in facility aspect, as we have discussed, students in academic institutions, facilities are not appropriate or may not be available, and they need to perform their research in some limited facilities which are available. Funding issues are also there, and pressure of research publication, it is also there with the students, that each and everyone now, NAC also and NBA also, they are also they also want this research publication. So students are also burdened with these research publications. During COVID, what I found is this in silico techniques, these are, or in silico studies, these are seen upsurge in these studies during this COVID or after COVID. Before that, some of the researchers, they were developing various this in silico techniques and using those techniques in research. But during COVID or after COVID, what has been seen, if you see the these publications, the number of publications involving in silico tools, it has been increased many-fold after COVID. So in COVID, the challenges were urgent need of novel antiviral drugs, and the need was there. Along with that, lockdown was there, non-functional labs were there, unavailability of resources, facilities, unable to develop newer facilities, difficulty in facility development, difficulty in assay development, unavailability of funds, even funding agencies, they were shut down, or they were not providing funds at that time, and accessibility of available resources, it was also become difficult in those era. But it has been said that where there is a will, there is a way, and we could find various such articles, research articles, which involve this in silico studies, and it has been exploited most in the year 2020 and later on. So they have used these techniques for various establishment of various chemicals, even from natural origin, like citrus medica, zingiber officinale, even from Ayurveda, various botanicals, these are being tested. So wherever structures were available of the chemicals, these are being tried, and these are being tested against this COVID-19. Even multi-target approach, it was tried with the help of this in silico studies, and various phytochemicals from the traditional Indian herbal medicine system they have used. Even one of the article I found, which I found interesting, is steam inhalation therapy. So they have also used such type of therapies which are being used, and tried it or tested it by using in silico tools and techniques. So advantages, we'll go through these advantages of in silico testing. Like we all know, I think, less time consuming it is. So the time required for completion of the research is less. No state of the art lab is required for performing any pharmacological activity. We need animals, we need various instruments, equipments, we need chemicals, and then we can go for in vitro or in vivo analysis of that chemical. But when we use in silico approach, we do not require all that state of the art lab. not much facilities are required. Whatever facilities I said, we may not require it. It will be so no huge investment is required, and we can access all these softwares or all these in silico tools from the safety of our home, which was important at the time of COVID also, and later on even if I'm not carrying my system, my laptop or my any PC or anything, I can use cloud system. Okay, because our my whole softwares we can install on the cloud, or whatever that space it is been provided on the cloud. So all the softwares and all my tools, these are available on cloud, and I can access it from anywhere throughout the world. So this is also, and I can say there is no restriction of a specific lab or physical lab. It will be there if we this in silico technique, they provide us this freedom. Do not need a huge setup. So within a small setup, we can perform this. An interesting thing is many of the researchers, they don't like this lab work. They they don't want that wet lab experimentation, and they even some of the pharmacologists I have seen, they are not they restrict themselves from using these animals even. So this avoids dirty lab work also. And one of the important thing which is nowadays is important for each and every researcher is publication with decent impact factor and recognition by peers. So many of this in silico testing or in silico tools which are being used by researchers, and they have published their research articles in some good reputed journals. Even my publications, our team also published in some good reputed journals with the impact factor of more than 4 or 5. So which tools? Tools are easy to understand, easy to use, and available free. Either freewares are there, or academic licenses are there, or these are available as open source softwares. Hardware requirement, not much. Even when I started working in this in silico, working with in silico tools since last 8 years, and I started with using the laptop with Core 2 Duo. C2D system, you probably know. Now we are having i5 and i7 processors. So I started with that, and even I could perform better studies by using that C2D system also. If we can work with Linux, it is paradise over there, because most of these freewares or academics licenses softwares or open source softwares, these are available with a Linux system. And one of the interesting thing is Linux, it is the performance of Linux is better than Windows. So what I feel like personally, that Linux system, they work fine with this all softwares. So this is not an exhaustive list, but some of these tools I have included over here, which are easy to use, and even our students or our researchers who are exploring this, who start exploring this field, they can use this. Like nucleic acid and protein sequence analysis can be done with BLAST and FASTA, which is available with this NCBI website, or which is available with this ebi.ac.uk website. And these tools are available for use free of cost. Then pharmacophore modeling is there. Pharmit, which is available, which uses various databases, which allow us to use various databases for searching the chemicals. Zinc Farmer, it is nothing but developed by the Zinc database, and it is having some limitations that it only find out the chemicals from their database only, Zinc database only. And Ligand Scout is there, it is paid version, but it is also an interesting software which can be used and which provide us better results. Pharmacological activity prediction. So these are the software, these are the basically web servers. Pass Online is there, Swiss Target Prediction is there, or Mole Inspiration is there. So all these web-based servers, they provide us pharmacological activity prediction. Means whatever structure we are having with us, we just provide the if we provide the structure to this online platform, they will predict the pharmacological activity for us. How we will see in some later slides. Pharmacokinetic properties prediction. As we know, if pharmacodynamics we can go for this pharmacodynamic study. We can say prediction, pharmacodynamic prediction, or activity on specific receptors and how much activity it is being produced, and even we can go for quantification, estimation of quantification quantity of the drug also. But the important parameters which will restrict the use of these chemicals into animals or in human being is pharmacokinetic parameters. So pharmacokinetic properties prediction, it can be possible with Swiss ADME, PKCSM, or ADMET, SAR. These are also the online tools or online web-based servers which are available, and which are again, as I said, free of cost. QSAR, quantitative structure activity relationship. So this is also the two tools which I found interesting, and which is the QSAR INS, which is available as an academic license, and which is generated by Dr. Paula Grammatica, and she is providing it to academician who is having some background of this in silico testing, and this is also an important, or we can say good tool for doing this QSAR. DTC lab, it is available for all. We just need to visit this website and we can download it, and good result can be obtained with this QSAR also. I haven't even tried to use this QSAR INS and DTC lab on the same database, and nearly similar results I have got, and both of these softwares, they work on the same, basically same principle, or same methods they are using. Molecular docking, various docking softwares like Autodock tools are there, Autodock Vina. Then NRG suite, also interesting one, and AM dock, which provides multiple type of these docking protocols executed through this AM dock. So molecular docking softwares are there. Molecular dynamic simulation, this is what something, the extension of this molecular docking, where the molecular dynamics is been, or the movement of various groups which are there in the structure, protein structure, or in the ligand structure, it is been provided, and by using that movement, the exact fit of this ligand into the receptor, it is been found out. So the techniques which are being used, like BLAST and FASTA, it is nothing but basic local alignment search tool, and FASTA is FASTAll tool, which is nothing but sequence alignment tool. It is just find the matches between the query string and the database string. We can use DNA sequence, or we can use protein sequence, and in the database, whatever sequences are available, we can match with it. Here it is been shown on the screen that one sequence which is available in the database, and another one which is test string or query string, which has been aligned into aligned with that specific available database string, and the best fit, it is been found out. Okay? And depending on that, we can have various conclusions. Why BLAST or FASTA? DNA and protein sequence database, it is now used. A large protein sequence database or DNA database is available in public domains, and by using this BLAST or FASTA, it is now become easier to search through this database to provide the query, and this query will be aligned with the database, available database. And this BLAST and FASTA, these are two similarity searching programs which uses heuristic algorithm for performing these database searches, and they align these available sequences with the query sequence. Talking about application, various applications are there, like the person who are working in this genetics, they may find this interesting, that functional and evolutionary relationship can be established between the sequences. Even we can look for the species, we can identify the correct species, or we can identify homologous species by using these tools. Looking for known domains in protein, this known domain will be helpful for identifying the functions, maybe the protein category or the functions of the protein, or how that folding of the protein will take place depending on whatever domains which are present. Then looking at phylogeny or phylogenetic tree, whatever organism, microorganism, or whatever sequence we are having, we can arrange it into phylogenetic tree. Then mapping DNA to a known chromosome, that can be done. looking for common genes in two related species so that we can identify which type of protein can be synthesized. So these are what related to some genetic studies. Talking about pharmacophore modeling, it was established, or we can say the concept was given by Ehrlich in 1909, and he rightly said that the activity, biological activity, which is being produced by any chemical or by any drug, it is nothing but it is nothing but because of its essential features. And IUPAC definition of this pharmacophore modeling, it says that it is nothing but the biological activity is nothing but the feature of, nothing but the outcome of the steric or electronic features or various features of the structure of the compound, and we can predict, depending on the steric or electronic features, we can predict the biological activity of a compound. So two approaches are there, like ligand-based pharmacophore modeling is there, receptor-based pharmacophore modeling is there, and various attributes are there, various features of the compound can be used. On right side, it has been shown that the feature of various these compounds. If we know the ligand which are active, we can identify the features which are responsible for the activity, like H bond acceptor groups are there, H bond donor groups are there, anionic sites, cationic sites, or hydrophobic or aromatic sites are there. So depending on that, we can go for virtual screening. Like we are having chemical databases available nowadays, and through these chemical databases, we can found out the compound which will fit into these specific features. Like in between the middle of the page, we can see the features. These are being shown along with this various this diagrammatic representation. It is there of the various feature. So by using this feature, we can search various chemicals which are having these features in the chemical databases, or we can go for de novo designing, designing the chemicals by ourselves by matching various properties, various features. So this can be done with the help of Farmit. We just need to visit this website, farmit.csb.pitt.edu, and we can go for, see here, various databases. These are being shown. This Farmit allow us to use various databases like ChemBell32, ChemDiv is there, ChemSpace is there, MQL is there, MMolport is there, Zinc database is there. So we can find out various chemicals from these databases with the properties, with the features. On left side of the screen, we can see various features which are been selected. We can select or deselect this. We can switch it on or switch it off, like hydrogen donor, hydrogen acceptor, some hydrogen acceptors are there. Various positions of this hydrogen acceptors, it is been given below that. Okay, so depending on these various properties, and by using this chemical database, we can found out various chemicals. Here I have used this Molport as a chemical database, and one of the query which I have been given, I was working on this X-link apoptosis inhibitors of apoptosis. So XIAP is the protein, and on that I found this various chemicals or various 15 such chemicals from Molport database, which matches my query, and this is the structure which has been shown over here from of one of that chemical. So by using this simple approach, we can find out various chemicals, and we can get a list of chemicals, and then our actual work will start. This is what the guidance which has been given by this software, Molport software, or by this Farmit, that such type of compound which is already known, and which can be used. We can go directly for the activity prediction, or we can go directly for in vivo or in vitro screening of this compound, or we can develop some newer compound from this specific structure. So such type of approach I have used, my students are using nowadays. Next is Pass Online, which is also one of the we can say online tool which is available free of cost. Prediction, the long form is prediction of activity spectra for substances, and this specific database, it is having around more than 250,000 different compounds they are having, and their biological activities they are having. They are having the database of SAR, structure activity relationship, and depending on this database, they predict over 4,000 different biological activities if you provide the structure to this specific system. They predict the pharmacological effects, even mechanism of action due to similarity of chemical structures. Even they produce adverse effect, toxic effects of that compound, interaction with metabolic enzymes or even transporter proteins, and even they can predict the influence on gene expression also. So all these things can be possible or can be available within with some clicks, and it will see the mean accuracy of prediction. It has been claimed by this specific pass online portal is around 95%. They say that 95% accuracy it has been there, and many experiments they have performed, and on the with the help of these known compounds, and they say that their prediction accuracy is around 95%. So in research, I feel if mean accuracy of prediction is about 95%, then we can say that it is much useful web-based application for us. So it will reveal new effects and mechanism of action. Just now we have seen some repurposing. We might have heard this word, repurposing of medicine. Whatever drug available in the market, we can search their newer effect, newer pharmacological actions, or some newer mechanism of actions. Finding the most probable new leads with required activity spectra. We are having some various compounds available, and we need to find out, we need to segregate it depending on its prediction. So that can also be done. Selecting the most prospective compounds for high throughput screening. While performing high throughput screening, if I am having 9 different thousands of compound. One study we have performed, I was having my students, they have collected a compound database of around 9,500 compounds, and which are available and which are likely to be drug, can be used as a drug. Their pharmacokinetics, predicted pharmacokinetic as well as reported pharmacokinetic parameters were good, so they can be administered in the human being. Not directly in the human being, but administered in the animals, and we can perform these pharmacological various activities on that. So if 9,500 compounds, if I am having,
sir?
Yeah.
Sir, your slide is not moving. It's stuck.
Still stuck?
Hanji, sir.
So where it is been stuck?
Sir, pharmacophore modeling. Ligand based, receptor based. So that flow chart.
Just a minute. I'm sharing it again.
Okay, sir.
Is it visible now?
No, sir.
Still not visible?
No, sir.
Now?
No, sir. It's not visible. Still not visible, sir.
Now?
No, sir.
Oh. Hope it is visible.
No, sir, it's not visible.
I'm repeatedly sharing my screen, but still it is not
visible.
Can I do this thing? I will left and again rejoin.
Yes, sir. Sure, sir.
Still it is not allowing me to share the screen.
Sir, still it's not visible.
Yes, that's what. Just a minute.